On behalf of myself and Harris Berman, the Dean of our medical school, I want to welcome all of you to what I think will be a wonderful and informative day. Indeed, I wish I could spend the day with you. For me, to see this come together in such an exciting way, it's really particularly gratifying. Many of you know that back in the day, it's becoming sort of back in the day, I devoted the majority of my career when I was doing research to working in the cancer area. It was an area that I was passionate about at the time. It still means a great deal to me. It's been so gratifying to see over what, unfortunately for me, is now a very long number of years. Tremendously exciting to see the advances. For those of you, particularly, almost everybody here is younger than me, I'm sure, I actually vividly remember hearing for the very first time that oncogenes in retroviruses were part of the cellular genome, and remembering how amazing and exciting that seemed. And when I look at the program that you're all going to enjoy today, I realize how very, very far we've come, although we still have a ways to go, certainly. I think you'd all agree. But it's amazing to be able to see and understand the tools that all of you can bring to bear on this both challenging, extremely important, and fascinating problem. So I'm extremely excited that you're all here at Tufts to have this wonderful day. It really looks as though it's almost perfect. Maybe it will be perfect. I hope so. It brings together outstanding people from across Tufts, from across Boston, and indeed the US, and I believe there's international representation here as well. So it's a wonderful day for Tufts and the cancer efforts at Tufts. I want to particularly acknowledge the sponsors who helped to make this day possible: Cancer Research Technology, the AACR Boston group, the Raymond and Beverly Sackler Convergence Laboratory, the Breast Cancer Alliance, and of course the School of Medicine. Particularly also want to acknowledge the people who spent a lot of time putting the program together that you're going to enjoy. Phil Hines, Charlotte Cooperwasser, Carl Munger, Larry Sterenka, and Amy Yee. And you're all going to be the beneficiaries of a wonderful day, thanks to our sponsors and thanks to the hard work of those individuals. I think you're all anxious to get the day rolling, so with that, I'll ask Phil Hines to come up and actually start the program.
So without further ado, I would like to introduce our first speaker, Charlotte Cooperwasser. Charlotte is a professor in the Department of Developmental, Molecular, and Chemical Biology here at Tufts, and she's the director of the Raymond and Beverly Sackler Convergence Laboratory. And you'll hear a little bit about that from Charlotte's talk. So without further ado, Charlotte, if you come up and tell us about strategies and mechanisms to prevent disease.
Thank you, Phil, for that introduction. And thank you all for coming and joining us this morning for our first annual symposium. I hope you'll learn a lot about what we're doing here, what we're trying to achieve, and take away with you some of the exciting developments and engage in us in their collaborations or convergence, because that's ultimately the goal of what we're trying to do here. So as Phil mentioned, I am the director of the Raymond and Beverly Sackler Convergence Lab, and this really sort of came about because we right now, life sciences, is in the midst of a revolution. As you can appreciate, the last decade has really brought forth technology-driven innovations, massive data, wonderful advances in the physical sciences, in the life sciences, and the engineering sciences. But at the same time, with these massive expansions and innovations, there's actually been a contraction in the funding mechanisms and in the funding of this kind of work. And traditionally, we as researchers have been working sort of in silos. We have a problem, either in biology or engineering or chemistry. We work in our labs to try to solve that problem, and we're generally funded to do that. But because of this contraction, the silo model is becoming more and more challenging, simply because we can't innovate and produce in the same amount, the same quantity, given the amount of funds. And so one way to sort of overcome this has been to think about branching out and forging interdisciplinary collaborations. And that's really coined this term convergence. This is really coming about, and you're hearing a lot about it maybe in the zeitgeist. Washington and in DC, there's lobbying to try to bring this to the forefront. And really, it's just the notion that to make impactful collaborative research in this environment, in this climate, and to sort of bring in all this new technology, we need to start working together. And so really it's the idea of bringing multiple fields, particularly what I like to call the physical and hard sciences, the chemistry, the physics, the math, the engineering, to the biology and to the biomedical research, to really take the fundamental discoveries we make in biology and rapidly transform them and rapidly translate them either into commercial ventures, into therapeutics, into ways that we can't even potentially predict because of our silo group. And so in 2013, the Raymond and Beverly Sackler Foundation endowed the laboratory at Tufts University with the idea of really trying to make impactful research that would actually be collaborative and have a large impact across society. And they really kind of said at that point, do what you wish with it. And given our focus on cancer, and given that cancer right now is really at a precipice of really making a fundamental change in the diagnosis and the treatment of the disease, and our efforts, we came together and created a group of individuals that span multiple disciplines to tackle cancer as the first project. Obviously, we'll see how this goes in the future. We might expand to other diseases, but given our focus, the group has assembled to really start to battle cancer. And so the vision that we set forth was rather than say, okay, here's one cancer type, let's go work on it. Let's take the expertise of the individuals in the group and actually use that to then sort of step back and come up with new ways to think about cancer, to model cancer, to develop therapeutics or test cancer, and then to rapidly translate that into the clinic. And so how are we doing that? Well, for example, we have a group of individuals that we call the data health team. And Andrew Beck, Anthony Philippakis, and Piyush Gupta are actually brilliant computational mathematical geniuses in their respective fields. And they basically take bioinformatics and machine learning and artificial intelligence, and they're using this in ways to develop new wellness products, improved therapeutics, and creating sophisticated algorithms to determine therapy efficacy. And so if you interact or chat with any of them, you'll see that they in themselves have their own labs, but they're actually taking this technology and these ideas and bringing it a larger forth. So for example, Andy has pioneered artificial intelligence to sort of revolutionize pathology. And he's created wonderful machine learning algorithms to be able to scan through slides, very much as pathologists do, but the machine is doing it and then can be able to rapidly determine whether or not it's a malignant disease or whether it's normal. Anthony leads a group of 80 coders at the Broad who is taking very large databases and sequencing data and expression data and really trying to mine in new and sophisticated ways. And one of the challenges we put forth to him was whether or not he could take his team and his ideas and see whether or not there's any information to be gleaned in the area, for example, of immunotherapy. When you collect information, large quantities of data from a single individual, and then you look at that across multiple individuals, can you determine what it is about that, the mutation or the gene expression or the genetics of an individual that allow it to, for example, immunotherapy, respond to it or not respond to it? And Piyush Gupta has actually undertaken a really interesting idea, which is looking at large data sets to determine whether or not embedded in that information is information about therapy efficacy and which patients might respond to a therapy prospectively rather than having to embark on a clinical trial to determine that. And so that's just giving you an idea of what they're doing. Other ideas that they're thinking about is can any of this information and technology they're developing be used as in wellness apps and actually make it into the palm of your hand and convert your iPhone into a diagnostic reader in that sort. So, that's sort of one area that the group is looking at. We have another group, which is the diagnostics and therapeutics team. And really, these are individuals who have a real close tie with the clinic. They have their own research interests, but they also see patients, treat patients, and understand the ins and outs of clinical care for cancer care. And so Dejan Jurek, Vicky Seawald, and Gaurav Gupta are all oncologists in one sort of another. And so they rapidly allow us to access their clinics and their clinical samples and their diagnostics and use some of the technologies and ideas that we're thinking about to rapidly translate them into the clinic. And we have a very large group that's focused on what we're calling broadly as technology development. And really, these are individuals such as Josh Kritzer at Tufts in the chemistry department, Irene and David also in the Tufts at the engineering department, to really use their expertise and their backgrounds to come up with new technologies and new ways to think about cancer. To, in Josh's case, to maybe come up with small molecule inhibitors or novel drugs to inhibit cancer growth. Irene, to image it. She's a very sophisticated bioengineer that focuses on imaging, and are there new ways and new tools that one can develop to image cancer beyond our standard mammography or MRI or CT scans? And she's really coming up with some innovative ideas. And David Kaplan, one of our stars at Tufts with 3D tissue engineering, can we use some of his technologies to create tissues in the lab and use those tissues as a means to create cancers, to test therapies, to look and understand progression? And then we have another group of technology developers who create model systems, and you'll hear about Steve Elledge, his pioneering technologies that span so many different areas, so I don't want to talk about that. But having a group like this also embedded in the Convergence Lab really brings together different expertise, different perspectives, and hopefully will make a larger impact. And so what are we focusing on as a group? Well, there's a pretty short list, and it's not the exhaustive list, but really, using our strength, and that is tissue engineering and hydrogel fabrication to create human tissues in the laboratory and see whether or not these tissues can be used as platforms for understanding disease, for testing drugs, for predicting drug efficacy. Personalized diagnostics, peptide mimetics and library designs for either chemical screening or target identification and target therapy. The creation of various molecular tools for custom libraries for just general biology understanding, CRISPR libraries, expression libraries, and a lot of these really pioneered by Steve Elledge's group and Josh LeBaire's group. And then NAPA protein arrays, that's also a really interesting and wonderful technology that Josh has developed. And using these to understand and probe even some fundamental biology as well as transformative translational biology. And so I'm happy to speak with any of you individually one-on-one to give you more details of any of the specific projects. But what I'm going to use the rest of the time to talk about today is really how we're using some of this convergence work and some of the folks in the convergence group and how we've collaborated to understand something that my laboratory is interested in, I'm interested in, which is the origins of breast cancer. And while there is a lot to appreciate with the idea that therapy, treating and defining drugs that will treat breast cancer or cancers in general is a very honorable and daunting task. One of the things that I think is clear is that if we can prevent cancer in the first place, we're actually going to save a lot more lives. And prevention is one of these areas that's really difficult to study because you have to understand the disease in its infancy before it's even pathological. And so to do that, the first thing is, well, how do you model something that you can't see in patients? And so one of the things that the Convergence Group, and in particular collaborations between my laboratory and these folks, have been trying to do is develop models to study early disease, early cancer, and then use those models to then understand and define specific mechanisms that are involved in fueling those cancers, with the ultimate goal potentially then using that information to treat existing cancers that may have relied on those pathways to form in the first place. And so one of the areas that we've been looking at is how do we model early stage disease? And in breast cancer, an early stage disease is what's referred to as ductal carcinoma in situ. It's really when the cancer cells grow inside the lumens of the duct and fill in the ducts. And at this point, the disease is actually curable. I mean, women show up with ductal carcinoma in situ, they get it removed, and they're cured. However, once it progresses to invasive ductal carcinoma, then the horse is out of the gate, and then you're basically treating the disease like any other cancer. And while there's been a lot of improvements on detection and treatment, we don't understand this transition. And if we could actually stop this transition, we could actually stop a lot of women from having to go through cancer treatment and possibly life-threatening disease. And so the human breast, and my laboratory focuses mostly on human, primarily because the mouse models and the rodent models really don't recapitulate the human breast and the disease quite accurately, either anatomically or genetically. And this is just showing you what the human breast looks like, and it's very complex, it's very heterogeneous. At any one time you have structures in various forms of maturation. And what you can see here is with cell lines and with other sophistications, our goal is to try to recreate these structures in 3D through tissue engineering. And so the way this is done is we isolate cells from women undergoing reduction mammoplasties in the clinic, and these are either Vicky's patients or patients here at Tufts. These are normal tissues, presumably disease-free. We isolate the epithelial cells into little organoids, and then we actually embed these organoids in these extracellular matrix hydrogels. And so these are engineered fabricated gels that compose of matrix that's very similar to what's found in the human breast. And what's really interesting about these hydrogels, as opposed to the standard commercial collagen or Matrigel or other ECM scaffold that you see, is the complexity of outgrowth that you see when you embed human cells in these hydrogels. And this is just showing you today's standard technology, Matrigel or collagen, you really have stunted growth. But in these hydrogels, you really get much more elaborate, complex structures that are actually very reminiscent to what you actually see in the woman's breast. And so you have these immature lobules and these much more elaborate mature lobules. And additionally, these hydrogels really offer a level of resolution to understand cell biology and morphogenesis that's really been unprecedented. And you're looking here at just some phalloidin staining, so staining of the cell membranes and structure of these hydrogels growing in 3 dimensions. And I think you can appreciate the complexity and heterogeneity of these structures that's never been before seen. And you can stain these structures with markers that you see in human breast tissues. In this case, we're looking at green cells, which are the myoepithelial contractile cells of the breast. In red, you have the luminal cells, the milk-producing cells. And so you can see in 3 dimensions in these scaffolds, you can really appreciate and see the local. And so what you can see here, and hopefully at the level of precision and resolution that I don't think we've ever been able to see before, is you can actually see inside the cancer cells were actually injected within this lobule, and they've clearly migrated into other lobules of the structure. And then you can actually see now on a single cell level microinvasion into the hydrogel structure. And this is really amazing now because we can study this disease and we can isolate these cells and single cell sequencing, and you can imagine the things that we can do now that we have accessibility to this kind of structure. Moreover, we're studying the nature of what's the difference between a non-invasive cancer cell versus an invasive cancer cell, and what does that do to the structure. And so you're looking here again at the 3-dimensional reconstitution of, again, here in red, you're looking at normal human breast epithelial cells from reduction mammoplasties, microinjected with green cancer cells. And in here, this non-invasive cell line, it actually only migrates within the ducts from lobule to lobule and stays confined within the structure. But what's really remarkable is if you look at an invasive cancer cell line injected into the structure, not only does it invade out into the matrix, and through the different lobules, but it also fundamentally transforms the structure, the normal structure of the breast tissue. Maybe potentially leading to the idea that this invasive phenotype that one sees in the clinic could be actually coming from the inside out. And not necessarily just from the cancer cells that have invaded out. And so again, we're at a time now, and the Convergence Lab is in a place now where we can start to study some of these questions, use this platform to test therapies, look for inhibitors and drugs that prevent that. And to sort of tantalize the idea of looking at inhibitors, what you're looking at here is the same, these are static images of the human breast tissues that have been microinjected with cancer cells, and you can see over time how the cancer cells invade out into the hydrogels. And in this case, we've inhibited a specific gene that we've identified to be involved in that invasive switch, and you can see that when you do that, genetically, you can actually prevent the invasive progression. And so I think this now may even serve as sort of a rapid, high-throughput fashion to test therapies that would prevent progression. And so that's one model and one system that we're using to understand this disease. Another model and system that we're using to understand breast cancer, and particularly the origins and the etiology, is the human and mouse model that we developed many years ago in my laboratory, and have since then been using it for a number of different applications. And so the human and mouse model is really a similar approach to the hydrogel system, with the exception that now we're actually looking inside of an animal, a mouse, and so we can include the physiology and the histology on that level. And so this enables one to take an immunodeficient mouse, clear its endogenous mammary epithelium, and then humanize the fat pad so it actually resembles and recapitulates human breast tissue. And then we can isolate the human breast epithelial cells, implant that into the humanized gland, and recreate normal tissues, pathological tissues, cancer tissues. We can also make milk in these animals, but not nearly as well as we can in 3D. And so just showing you here these human and mouse tumors that we've created, and we've created these from genetically modification of human cells. Although you've probably seen the literature, patient-derived xenografts, you can actually take tissues from cancers and implant them, but we've actually been making cancers de novo from normal cells. Human tumors are very heterogeneous, and you can appreciate that we get the same level of heterogeneity and hormone expression that one sees in the clinic. And so other uses for these models that we've been using them for has been to screen for genetic drivers of breast cancer, oncogenes, tumor suppressor genes, looking at epigenetic and chemical libraries and perturbations and how that affects early progression and cancer formation. And then one can also use this platform for drug discovery and testing certain therapies that are targeted for the genes that we've created in the model in the first place. And so just showing you one example of an experiment and a strategy that we're using this platform to understand, and that is one of the things that we don't know about cancer yet, although we've sequenced the genomes, we don't know what genes actually are causing and driving the cancer. We can catalog what they are, but we don't know cause and effect. And so we've been taking advantage of Steve Elledge's, among his many libraries, he's created a CRISPR library that targets 500 putative tumor suppressor genes. He's also created another library that targets oncogenes. And we've been using these libraries to infect the cells and then do an in vivo screen to identify which genes cooperate together to give you a cancer. And do those genes have any role in the cancer behavior, the cancer phenotype? And so we take his library, we introduce it into mice, and then we can isolate the tumor cells and sequence the genomes of these tumors and identify the guide RNAs or the expression construct or whatever we've put in to identify the genes that are causal in the cancer forming process. We've also used these models to identify the cellular precursors to human breast cancer, and the reason this is valuable is because if one thinks about the progression and the evolution of cancer, it's likely the case that that initiating cell, whatever it is, its state, its differentiation program, its sensitivity to cell death, its response to proliferation, gets carried along during the cancer progression process. And ultimately, when you want to treat it, some of that information might still be retained. And so you could use that as a foundation for targeted therapies. And so at the time before we did this work, and in some cancers this is still the case, it's thought that different precursor cells in the tissue gives rise to different cancer types. And so in the breast, the idea was that a luminal progenitor cell would give rise to luminal tumors, and a basal progenitor cell would give rise to basal tumors. And it's really the intrinsic precursor cell state that is responsible for tumor heterogeneity. The other model at the time that existed was, no, it's actually the precursor cell is the same for all cancers, but it's really the constellation of genetic alteration that that cell sustains that drives the heterogeneity. And so in breast cancer, you could imagine that there's a luminal gene set that gives you luminal tumors, a basal gene set that gives you basal tumors. And there's actually examples that support both of these models in the literature for both breast as well as other diseases. And so it might not be mutually exclusive. And so about 10 years ago, we decided to look at this in the context of breast. And so we used the human and mouse model, where we either sorted out different populations of cells and put in different cooperating oncogenes and looked at the resulting tumors. Or we started with the same pool of precursors, and we looked to see if we modified the genetic combinations where we get different types of tumors. And the results from a lot of work is really the idea that both of these models in breast cancer are playing an effect. The vast majority of breast cancers, about 95 to 99% of breast cancers, are arising from the luminal progenitor population. And it's really the constellation of genetic alterations that are cooperating with each other that drive tumor heterogeneity. But there's an exception with a rare population of breast cancers that are referred to as metaplastic breast cancers, about 1%. These tend to arise from a different precursor population, a basal progenitor cell, and that no matter what genetic combination you put into this cell, you always get rise to a metaplastic tumor. And so it could be the case in other diseases and other cancers, you also have this third model where both of these are in operation, and the key will be to identifying what the originating precursor cell is. So for breast cancer, if this is the case, if the vast majority of breast cancers are coming from a luminal precursor cell, then it begs the question, how do you get such tumor diversity, heterogeneity, differences in response to therapies? And so one of the things we've been looking at is the role that specific mutations and specific genes play in this. And we studied BRCA1 because it is primarily involved in this basal gene program that gives you these basal-like tumors, which are very poor prognostic tumors, very little therapy. And here what we discovered was that actually the genetic predisposition, so the initiating genetic mutation or alteration that that precursor cell receives, sets the stage and sets that cell on a path to becoming a tumor of a specific phenotype. And in the case of BRCA1, that was the basal-like phenotype. And so why was that? Well, when we looked at women who carried BRCA1 mutations, and we looked at their normal breast tissue, what we found was before there was any evidence of cancer, and so these are essentially the precursor cells to cancer, we already saw evidence of defects in differentiation. And most notably, we saw that in these normal cells, that they had upregulated the transcription factor slug. And at the time, we'd also gone on and shown that this transcription factor slug was also expressed in basal-like breast cancers, those from BRCA1 patients as well as others, and that if you actually inhibited slug, you could actually shift the differentiation state of the cancer, going from a basal-like tumor to one that expresses more luminal-like features, which is much more treatable and has a better prognosis. We then went on to show that slug, the transcription factor itself, was actually required for cancer formation in the first place. And this was pretty unexpected and profound. And so what you're looking at here is survival of mice whose genetics background is either wild type or slug deficient. And they're just on a mammary tumor prone background. And so wild type mice develop tumors very quickly. Mice that no longer have slug actually fail to develop tumors at all. And what's more interestingly is they also fail to transplant. And so what the role of this protein was suggesting was that it actually is involved in the early stages of stem cell factors, stem cell programs that are required to initiate a cancer, to go from a normal cell to then one that's poised to become cancerous. It has to turn on a stem cell program. And so using mathematical modeling, again, the convergence idea of bringing other disciplines, we were able to model the probability and the stochastic transitions by which cells go from different states, from stem states to differentiation states, and quantify that, and use that information then to help us understand that the role of slug. And fundamentally, it is a transcription factor whose role is to allow cells to acquire plasticity and stemness so that they can continue to replicate in the background of mutations and DNA damage. And what was most profound is that that initial mutation, in the case BRCA1, was really the trigger in this familial group of cancers to be poised to go into that direction. Mechanistically, we showed how this worked. It involved modifying chromatin, and if you want, we can talk more about this offline of the details. And so this really begins to set the stage in the understanding of in early disease and in early cancer, you really have this state where the precursor cell and the fate of that precursor cell is really important for whether or not it will be destined to form a cancer. And in the case of BRCA1 and slug, that really suggested the idea that a precursor cell, when it activates a factor involved in stem cells, can actually acquire this plasticity and give you a poor prognosis tumor type. But is it the case that in other cancers, you have a different type of plasticity, and that might also give you different kinds of tumors? And so here, collaborating with Josh LeBaire's group, he created a library for us, 1,000 transcription factor libraries, human transcription factors that we pooled together, and we used to identify genes that were involved in that plasticity that would allow luminal cells to acquire basal characteristics. And after doing this screen with 1,000 genes, we actually got a very short list of 50, and one that really came out and was tantalizing was this protein called TAZ, WWTR1. I'm going a little quickly here because much of this is published, and I'd like to leave room for questions, and we can talk offline. TAZ is becoming a really hot and exciting protein because it's part of this Hippo pathway, which is really turning out to be essential for cancer formation, for stemness, for a variety of physiological processes. And what we went on to show was that TAZ was a really important mediator of switching fates of cells. And so luminal cells in the breast could switch and become a basal myoepithelial cell simply by expressing TAZ. We went on to show mechanistically that this was because TAZ was binding to critical components of the SWIFT-SNF complex, both Brahma, BRM, and BRG1, and that this function of TAZ was involved in controlling lineage gene expression. And so in a cancer perspective, this becomes an interesting and valuable target because TAZ is amplified and overexpressed preferentially in basal-like breast cancers, again, the cancer that has very little targeted therapies. And so this gene that seems to be involved in conferring that basal phenotype is also selected for in cancers of the basal subtype. And moreover, its expression is actually statistically significantly correlated with poor prognosis. And so tumors that have high TAZ expression do much worse clinically than those that have lower TAZ expression. And so this sort of is illustrating the point about how some of the fundamental biology of understanding cell fate, stem cells, and early disease can be used and propped up with the convergence group. And so now that we're learning this biology and something we're going to continue to do, some of you in the audience are really interested in how is this going to benefit patients? How are we going to be able to use this in therapies? Our lab has been really identifying transcription factors, which actually are useless commercially because you can't target these. And this is where the help of individuals like Josh Kritzer, who have innovative ways of targeting transcription factors, might be one way that we can find ways chemically to inhibit them. But another way we can actually inhibit these transcription factors is understand how they're regulated in the first place, and maybe their regulators can be targeted. And in the case of slug, what we've discovered is that the deacetylase SIRT2 is actually a critical regulator of slug protein levels and its stability. And that if one looks at protein slug turnover, when SIRT2 is expressed, it maintains very high elevated levels of slug. And when SIRT2 is inhibited, slug rapidly declines. And what's more interesting about this is just as TAS was genetically selected for in basal-like breast cancers, SIRT2 is genetically selected for in basal-like breast cancers. And you can see that there's an amplification of SIRT2 in the basal-like breast cancers, high expression by gene expression, and even by protein, the triple-negative breast cancers have much higher levels of SIRT2. And finally, is this even a therapeutic option? Well, one in the lab showed that if you inhibit SIRT2, you can actually profoundly reduce tumor growth and tumor formation, suggesting that potentially that this could be a novel way to achieve slug inhibition by affecting SIRT2 level. And so we can get away around targeting transcription factors by finding the enzymes or kinases that regulate them and then targeting those. And so with that, I'd like to acknowledge the members of, current members of my lab, prior members of my lab, collaborators, and obviously some of the Convergent folks that were involved in this work. The current members of my laboratory here, I spoke about some of the work that former members, Patty, Sarah, Adam, Lisa, carried on. Wen had done the really lovely SIRT2 work. Anya and Janelle are doing some of the CRISPR and oncogene library screens. And obviously the Convergence Lab members who were involved in this work, Piyush Gupta, Andy Beck, Vicky Seawalt, Josh LeBaire, and Steve Elledge. And we're hoping that other groups are going to converge around other areas as well. And then finally, I'd like to end with the funding source for my laboratory, the Raymond and Beverly Sackler Lab, obviously, but then the other grant funding sources that have allowed me to continue this line of investigation. So with that, I'd like to thank you for your attention. I'd be happy to take any questions. Yes?
Quick practical question. What is hydrogel? How is it different from gel? Is it less expensive?
Not less expensive. So Matrigel is essentially an undefined extracellular matrix preparation that's actually created from tumor. So in mice, they're injected with a tumor. This tumor secretes a lot of matrix, and that matrix is then harvested, purified, and bottled as Matrigel. It's primarily laminin as its major component. The hydrogels are actually, the base is actually collagen, but then it's actually infused with various molecular weights of hyaluronan, various other extracellular matrix proteins like fibronectin, escaping me all the details, but various specific defined extracellular matrix components, and then on top of that, there's actually certain growth factors that then are added to it. So it's a very defined, matrix. The other thing about it is Matrigel, you just kind of stick it in your well and you let it harden. The hydrogels have to be fabricated, so it goes through multiple steps to actually go from collagen 1 to these hydrogels, and then you have to actually scoop them out and they contract. So it's a little bit more sophisticated than just Matrigel or even just collagen for that matter. But it's highly reproducible because you can define everything and everything sort of specific concentrations, specific times, and so it's Matrigel. It's lot-to-lot variability. You're kind of at the whim of the mouse, and here everything is really systematic and reproducible. Yeah.
Do you use the hydrogel both in vitro and in vivo, or just in vitro?
So for what I've been showing you right now, it's all been exclusively in vitro. We are taking some of the structures we've created and putting them into mice and seeing if putting them in animals in vivo allows or alters progression, transformation. So those experiments are ongoing. So you can actually do both because it's such a amenable method. But right now, most of what I've been showing you has been in vitro. Yeah.
Ask questions from the back. Charlotte, great talk. So the organoid model is beautiful, and I presume it's growing in some kind of matrix. Have you looked at invasion into that matrix or changing factors in that matrix to enhance or increase invasion into it?
So we haven't yet, so the short answer is we haven't yet focused on the factors in the matrix, with the exception, and this is where Irene's going to come and help us out a little bit, with the exception that we can visibly see something happening to the collagen. So there's actually a physical force and dynamics that's taking place in those hydrogels that we can presumably image and quantify, and so Irene's going to try to help us do that. We've been focusing more on what the cells are doing, and whether or not there's been changes in cell fate, cell differentiation. We've looked at the invasion process we know is MMP, so we've done sort of active MMP assays to look at degrading the matrix. But we haven't yet really focused on whether or not there's other matrix components being deposited and sort of what's the fate of the collagen and the fibronectin and the hyaluronan and all these other components that we've put in. So much to be done.
Really, really nice. I have a question. Maybe I missed this. Do organoids have any immune cells? What cells are?
Right now, everything I've been showing you has just been epithelium. So it's a combination. So I should also mention we've done this with single cells, so it doesn't have to be organoids. But we find the organoid seems to preserve the niches of the stem cells, and so you get much more elaborate, complex structures. They're primarily enriched in epithelial cells, luminal and myepithelial. We are working on incorporating some fibroblasts. I mean, that's what's actually really great about these gels is you can put in immune cells, you can put in any cell type you want and start studying the heterotypic interactions of those. But right now, it's really just been at the level of just the epithelial cells.
That was I was going to do a follow-up question. Caroline's touched on this. So going back to your bioreactors, thinking about something that's not cancer, but to make milk,
Yeah.
right? I mean, one of the major components of breast milk is
Immune.
the immune
Yeah,
component.
yeah.
So can you envision how that would work on a more industrial scale? Or?
So this is going to be one of our next convergence meetings. Is when we talk to the engineers. First of all, how do we scale it up? And
Is there a role
second,
for silk there?
how do we scale it up? And then how do we also access the milk? Right now, it's kind of confined in the lumens. The physiology of getting it out in animals is well defined. But this is convergence. I mean, right here is a beautiful example of how you would get convergence to solve those questions by asking the engineers and the physicists to come up with ways. And so maybe next year, the year after we've met more, we'll find those bioreactors. Yeah, they'll be on.
Well, it would seem to me you wouldn't necessarily have to do all of those things at the same time in the same bioreactor either.
Right.
I mean, if you were making a material like breast milk with multiple components.
Right.
You could do multiple.
But yeah, immune cells is something we've definitely been talking about because we're going to need that at some point. But do we need the cells or do we need the factors that they produce? And so that's things that we can tweak and sort out.
Yeah, as you well know, everything affects cancer, right? Cytokines and all that.
Right.
Further questions? If not, if you're shy, you can catch Charlotte at the break after the next talk. So I think we'll move on now to. Oh, let's thank Charlotte.
So this is the big picture philosophy. I think most of you know why 3D tissues are important by now, and I don't want to really have to restate all that. I'll just say that we think about our in vitro systems very, very carefully, as you'll see. And we care a lot about what cells, what combinations of cells. We care about the matrices. You heard a little bit about that. I'll try and take that even to a more detailed level. And we care a lot about the environment. And the point of tissue engineering is really building a system and have all those components talk to each other to then get something that is truly relevant to the human response. Because if it's not, I don't see the value for the kinds of goals this symposium is about. And so we use our systems not just for disease, which is where I'll focus today, but to look at drug effects, look at toxicology, issues of nutrition, and many other things, as I'll show you briefly. So the real value here is getting away from 2D cell cultures as your only metric and only using animal studies on the other side as your only metric and trying to marry the best of all worlds. And so that's why I think 3D tissue culture based on human cells offers something quite unique. And you can read this, I don't want to really repeat it, but if you know, everyone talks about personalized medicine today. Well, personalized medicine can even come in the form of tissue screening tools now. That's the point here, right? You can build these tissues from each one of your own cells through the techniques many of you know already. And as long as you do this properly, you have a system that can be very valuable for many things. And so we care about all this here. I can't cover all this today, but we have systems that we vascularize, we have systems that we innervate. I won't deal with those details, but the point is we're starting to get very, very complex and sophisticated about what we can do with the cells to give us readouts that are relevant. So for our lab, all I care about are these red highlights at the bottom. If it's not human physiological relevant, I don't want to deal with it, right? Number two, I care a lot about not just acute, but also chronic effects in our tissues, and that's really important to me and something that's appreciated, I think, in the field, so I'll harp on that too many times. And as always, feel free to disagree with me. And then the last bit is imaging interfaces, and Charlotte touched on that with the work with Irene, and I'll touch on that just at the end because of time on what we're doing there. Because if you're going to long-term 3D human cultures for the readouts we want for both acute and chronic effects, you can't have tissues that you have to take down, destroy, do immunohisto and all the other things. You need something online that you can image in real time and determine what's happening metabolically, toxicologically, structurally, and so on. So that's where the work with Irene comes in. All right, that's philosophy. The other philosophy is here's the field today, and I do this sort of as developmental biology. So on the left is where the field started 15, 20 years ago with some really nice work that simply said, I should point, I guess, you can take some cells, you can pick your cell type, it didn't matter at that point, take some cells, find some spaghetti with high surface area, which is a biomaterial in fancy terms, put the cells on the spaghetti and have them attached, grow them enough, and then reimplant them in the human body and you have a cure, right? And of course that wouldn't work well. In fact, the only thing that evolved out of the 20 years ago work was all the work on tissue engineered human skin, and that's a commercially viable technology. But if you look at that technology, and organogenesis is one of many fields that sells that to help a lot of patients, it's a very, very simple system. It's not complex by any means, but very successful. But there is no other example of a successful tissue engineered product today. And so what did that mean? That meant the field took a step back and said, what are we doing wrong and what do we need to do better? And over the last 15 years, as you all know, the developments and insights into stem cell biology, sophistication in biomaterials, so not just spaghetti anymore, now we can sculpt and control all the features you need for a biomaterial to make the niche what you want for those cells in that tissue. And we can get really fancy with bioreactors to create the environment around those cells. And now we can do things that 15, 20 years ago you only dreamed about. And that's why I think we're at this sort of revolution now on the right, because we have the tools. Whether we talk about them in the context of disease today, you can use those same tools applied to many other areas of interest in both biology and engineering and so on. And again, anytime happy to talk about these other areas because we do have some ongoing work in some of these as well. So for today, really we're just after how can we propagate those tools towards the study of cancer, and I'll focus only on that with the three systems. Again, the underlying theme here, though, is the right cells, the right matrices, and the right environment. It sounds easy. It's not, as you just heard. You know, you start with Matrigel, and then you go running away from it because it's so complex and undefined. And then you have to rebuild from first principles what you want. And sometimes you guess right, and sometimes you don't. And then ultimately for us, what we're after for our disease models is creating the niche to give you the relevant human responses. We started out with cells thinking we'd be simplistic with one cell type, and it turns out that it's never a good answer. You always want multiple cell types because cell-cell signaling is so important, as many of you know. And then again, to continue to beat on the time issue, we care about chronic more than just acute effects. So 2 overview slides I like to use a lot when I give these kinds of talks. One is if you look at the field of tissue engineering, and this applies in many areas, you can go from the left to the right at the top in sort of pictorial fashion. You all know how to do 2D cell culture or probably have been exposed to it, and that's sort of the tried and true approach to start. And as you work from the left to the right, you're just adding complexity, right? So you're getting more complex systems to the right. You're dealing with longer ability to cultivate your tissues over time. You're adding physiological complexity, and I would argue you add relevance. Okay, so that's great. Why doesn't everybody do what's on the right-hand side? Because it's very costly, it's very time-consuming, you're prone to contamination, and the students have to graduate somewhere between 3 and 5 years. So what we all do is start at the left and screen, right, and work our way to the right, just like in drug discovery, and there's nothing wrong with that. My message today, though, is don't stop until you get to the right. Okay, because if you stop before you get to the right, you'll get some useful data, but you're not going to get all the kinds of data you can potentially get out of your systems. So our mantra in our group is get to the right, okay? And we don't always get there right away, but we usually do. And a good example is we've published, and again, this is with Irene, on systems that we've grown for 6 months in continuous culture in the laboratory. And if you think about most diseases, most drug effects, most nutritional effects, it's not a short-term effect, right? You're after really slow developing effects. So you need these systems to run for long periods of time. And I know it's a pain, but it's worth thinking about. So I put this into biological terms since I figured most of you are biologists, right? So I used a growth curve. This is more from prokaryotic literature, and you'll probably be familiar with this, right? So A is your bacteria starting to grow, and just think of this as your human cells in your tissues, right? B is your logarithmic growth. The cells are starting to get comfortable and doing what you want them to do in whatever system you set up. C is what we call steady state, right? That's where you want to get your readouts. That's where your tissue is stable, your disease model is stable, and you can start to generate data that may be more relevant to the human condition. And then if you wait too long, D is where things start going south, right? So the cells are starting to die, overgrow, whatever it is, and now the readouts are less relevant. So my last point of emphasis on the chronic versus acute is almost every system people use today is on the left-hand side there, whether it's 2D cell culture, transwells, microfluidics, tissue on a chip. They're all wonderful systems, but they end somewhere around 2 to 3, if you have a really good system, maybe 4 weeks. Great, but just keep in mind you're limiting what you can then use those systems for because they're not steady past a few weeks, and again, if we're looking for long-term effects, we need to get to perfuse cultures that we can go out for months and months at a time. So it's good to keep all that in mind if you're really interested in the kinds of things both this symposium is about, as well as what I mentioned before, nutritional issues, drug effects, and for us also just tissue regeneration. Can we damage our tissues, watch the repair, and learn how to improve those repair modes? Even all in vitro, we learn a lot by doing that. All right, so finally we're done with my philosophy. Let's go on. First tissue will be really quick. I just want to talk about fat. And this will be in the context of what Charlotte already spoke about in terms of breast cancer. But first, the system, because as I said, I want to tell you about the tools, not so much the disease. So we spend a lot of time studying 3-dimensional fat tissues. This is led by Roz Abbott as a postdoc in the group. And the simple way to describe this is we get lipoaspirate samples from patients. And then we used to go through the process of isolating single cell types from that lipoaspirate through all the FACS sorting, all the mess you go through, seeding our tissues, generating these tissues. And they looked good, but they weren't great. And then we took a step back and just took the whole mess that we get from the patient and we seed that into our biomaterial scaffolds and grow our tissues. And now the results are outstanding. So it's a lesson in how important cell complexity is in your systems, and once you seed and understand what grows well, you can work backwards and figure out what cells like the niche and how to then exploit those kinds of insights. So one is the cell sourcing is important. In the middle is the scaffolding, the biomaterials, the matrix that's going to create the artificial environment in the laboratory, the niche, if you want to call it that. So it doesn't matter so much what we use here. What matters is all the designing that goes into choosing the best system for that given tissue. And it'll be different for every kind of tissue. It's even different if we're growing white adipose tissue as opposed to metabolically active brown adipose tissue. So it's not a one-stop shop here. So I gave you a list of things we sort of have to go through for every new tissue in the lab, and we've developed a lot of tools to do this, but you have to worry about all that. You know, the chemistry, structural morphology, the biomechanics, the degradation profiles, all those things matter if you're going to get optimal tissue outcomes. And I'm sparing you all those details for each tissue type today, so I'm sure you'll be happy with that. But then we take that and we go ahead and cultivate and look at what we get. So just for the white adipose tissue, here's an example of what Roz gets on the right-hand side. And if you know anything about fat, you should be excited to see this, right? Those orange globules are humongous, right? So we've got really good in vitro mature human adipose tissue. That's quite remarkable for those of you who study this in the field. And I won't walk you through all the details, but there are lots of different cell types in here because they all survive from that lipoaspirate. They figure out, they self-select, and then you get a tissue that has what you want with all the different cell types. You can see the scaffold is kind of the purplish blue in the background. I'll skip all that, but the scaffold is there obviously to keep things open and porous so you get good mass transfer and you don't have necrosis in your tissues over time. And we're using these kinds of very functional tissues for all kinds of metabolic studies, all kinds of tissue repair, and regeneration studies and patterning studies, which I won't go through today, but these are all done in collaboration with both Irene, who you've heard about already, as well as Mike Levin in the biology department up on the Medford campus. But what we're trying to do is establish this system as a sort of a physiological reactor that we can perturb in different ways and look at how it responds. And Roz is most interested in things like obesity and diabetes, and with a sustainable 3D adipose tissue culture, you can actually start to make progress in understanding drug effects and how overdosing with glucose and so on has an impact on the structure of the tissue, the size of the globules, and the metabolic profile of what you're seeing. And this is all being fed into models as we make progress to try and understand how to predict the best sort of inputs and outputs for the system. I know somebody asked a question, well, where are immune cells? So as many of you know, whether it's obesity and diabetes, immune cells play a huge role. So we also introduce immune cells into our system as part of the process of understanding how low levels of inflammation affect the readouts that we get. So this is very active, ongoing work in the group. In terms of disease models, really what we're doing, and this is really just started because of the convergence lab that you heard about from Charlotte, is to start to take those 3D tissues, and instead of lipoaspirate, now we use reduction mammoplasty sources, but the same idea, feed the whole material mess into our scaffolding and start to grow tissues, and you can see what we have on the left there. And this work was started both with Victoria's lab as well as a little bit with Charlotte's lab. And we can already show so far with fairly short-term studies, we get fairly stable cell responses and we start to see metabolic responses, whether we're stimulating with epinephrine or with insulin. So the tissues are responding the way you would expect as a starting point. And then obviously we can now start to introduce cancer cells and so on, and that's the next step in the plans as we move forward. All right, so that was the adipose tissue as a first example of how we go about building 3D tissues. I want to talk secondly about brain tissue because number one, it's probably my most passionate tissue these days. I'm intrigued to always learn more about the brain because we know so little. And so we have a group that's worked on developing a three-dimensional cortical neurological cell model. The current postdocs here, Olga and Yuting, are very active in continuing to improve the model that we already have. And the model was based on something very simple. If you look at the cortical region of the brain at the top, you see a layered structure and you see this really important connectivity among neurons to get proper cell signaling and obviously brain function. And so we recapitulated that design in our tissue model at the bottom. If you just look at the center, we call it affectionately the donut model. And the idea is the gray matter is that sort of porous sponge ring around the outside, and that's where the cortical neurons sit when they're seeded into the brain model. And then the green region in the middle, which is the white matter, if you will, the analogous white matter in our in vitro model, is where those cortical neurons have sent out their axons to connect in the middle and hopefully make a functional tissue model. So that's the system that we initiated a few years ago, and it's continuing to progress very, very well in many directions. And so just a few slides to show you what that looks like. Here's one of those sponges at the top left. You can get a sense of scale here. This is about 12 by 2 millimeters. The center hole is about 2 millimeters where the axons are going to create the connectivity. You can see what that center gel looks like. Lots and lots of axons here that have been propelled into the center by the neurons growing around the outer sponge ring. And just like before, you have to optimize the mechanics of those sponges, you have to optimize the chemistry, the matrix molecules, all the things I mentioned before. I'm skipping all that. And then in the porous ring on the outside, you see the axons, the neurons sitting there as well along the edges of the sponge layer, waiting to send out their projections into the center gels. And I always like to use this slide because it's instructive of how you have to optimize the matrix you're going to use. If we only use that outer porous sponge for these models, we get really nice cell growth around the rims of the sponge, around the porous domains, but you don't see axons really be projecting into the center regions where there's no solid substrate to hold on to. And if you only use the collagen gel, and this was mentioned a little bit even in the first talk, collagens are wonderful, but they are not very stable materials in vitro culture. They tend to contract over time. And so the cells get in there, they're alive, but they don't really send out projections. So they're sort of in steady state, not doing much. They're not going to give you a functional model. And then if we go to the composite system, which is taking the porous sponge in combination with a gel, now we get the best of all worlds, and we have a functional set of structures we can start to at least pretend look like a brain structure and go on from there. So the designs are really important if you're looking for a physiologically relevant set of outcomes. To the point about time and culture, just to make this point here, we go out about 8 weeks in our initial cultures. Our goal is 6 months. And we can see with the 3D systems, the red, we're able to maintain good viability as well as get good readouts of many markers of neuron activity, whether it's synapse-related, adhesion-related, and so on. So the 3D systems seem to be sustainable for reasonable periods of time. We're busy integrating a blood-brain barrier now with Yuting's work, and that should get us out now to hopefully the 6-month time point, but that's work in progress. So one thing is, do you have the right structure or something like the right structure? Another thing is, can you sustain it? The third is, do you have function? So for a brain, for us, function means some kind of electrophysiological reading, some calcium signaling, some biochemical damage response. And a little bit of that is on this slide. The answer is yes, we see all of that. So for example, if we take the brain tissue right there and we drop a weight on it to simulate brain injury, if you will, we see a change in the electrophysiological recordings with local field potential that we measure in 3D structures. We can actually measure the spiking, and it's different depending on the severity of the damage we apply to the model. So that's encouraging, and then things return to steady state after some period of time. And then also we can monitor the biochemical changes over here, and one of the best indicators or markers is glutamate production. And we see glutamate secretion from the damaged tissue, and again, the level we see depends on the degree of damage and so on, and that's something we continue to look at. So you know, the system seems to be functional, at least at this early stage. One of the things we had to do was make sure we converted our system, which started as rat neurological components, into human, and that we've done more recently. And this is the work of Dana Carens, who's a postdoc in the lab, and she developed a really beautiful human-induced neural stem cell line that we use in all the models. And you can see the beautiful axon networks that form in the gel center as the cells take shape and do what you want. There's really nice calcium signaling. I'm not going to run the video, but it's there as well. So we have a system now that's relevant to the human condition. That's the point. So where are we going? On the cancer side, these are the systems we're running now. These are the collaborations that we have going. using the tools that I've talked to you about, but now working with other labs to try and bring this to some relevance for cancer treatments. And you know, some of you, I'm sure, know the severity of some of these cancers. I don't have to tell you that, glioblastoma on the left and the medulloblastoma. So on the left side, we're starting with cell lines. We're in the 3D system. We have a really nice reporter strain here, so we can see if we compare now our niche system. Here we had to switch from just the protein gel porous sponge system to a protein plus hyaluronic acid system to get better readouts for what we think we need to see. And the cells seem to be responding very well, much better in 3D than you see in monolayer culture, as you might expect. And they seem to be doing very well in terms of upregulation in the system. And this is the work we're doing with Columbia here. On the right side with University of Connecticut, we're looking at patient-derived primary cells from resected tumor systems. And here in 3D culture, you can see over a couple of months now, we're getting very good, robust astrocyte response in our systems, indicating that the niche that we've created is supportive of these cells in the model, meaning we start to have a system we can use for screening and so on as we move ahead. And all this work is being done in collaboration with the folks below, but also led by Disha. Disha is a PhD student about to finish in the group this coming year. All right, so that's two samples, the adipose tissue and the brain models. The last one I'll just touch on very briefly is our human intestinal model. And this is led by Yin Cheng. Yin is a postdoc in the group. And I don't need to review what the intestine does and is, but it's clearly a critical component in all of our bodies. And so a few years ago, we started a system to generate a 3D human intestine system in the laboratory, taking in mind all the sort of design criteria I've already covered with you. So we wanted something sustainable, relevant, and so on. So we built both a small and a large human intestine, and the design is complex, but I'll just say it looks something like this. So think of the green regions here as a hollow tube, like the intestine, right? But that tube is a spongy structure, and then there's a center hollowed core, so the epithelial cells will line that core, and then there are channels within the sponge itself where we feed in other cell types, immune cells, and then now we wrap on the outside our neurological cells to get innervation into the 3D model. I'll just touch on a few of these aspects today. We also built a model with ridges versus no ridges, so we can simulate both the small versus large intestine, and on we go. All these models were first built with cell lines to make sure we could get things working well, and now they're all converted over to primary cell sources using organoids and so on. Okay, so do we have a system that looks and feels like an intestine? So the first answer is yes. Here is some data to show that. So I'll skip both lines just to say here are sort of tight junction evidence that the epithelial cells on that inner lining of the tube are closely associated, and you get beautiful microvilli forming on those cells as things are cultivated over months. You see digestive enzymes produced as well. The staining in red shows you that. So many of the things you want to see for an intestinal model are viable. The bottom line for an intestine is mucus. If you're not getting lots of mucus produced, you don't have a good model. And so you can see here, we get lots of mucus. Here's a cross-section for the 3D patterned lumen, and you see multi-layered mucus, which really recapitulates what you see in the human condition. And if you care about quantitation, you can see the 3D pattern lumen gives us the most copious amounts of mucus compared to the 2D transwell and the non-patterned lumen. So function in terms of mucus is also very, very prominent here. And then the bottom line for us is if you don't have control of oxygen, you also don't have a relevant intestinal system. And that's what we show you here. We use here reporter bacterial cells that light up depending on the oxygen environment. But the bottom line is we measure this. If we're just using transwells, we get down to, I think it was like 10 to 15% oxygen levels, not down to the level you want with a human intestinal system. If we had the non-patterned system, we can get down to microaerophilic conditions, which is what you want to see. And with the pattern conditions, we get down to all the way down to anaerobic conditions. So we really have a model that has the structure, it has the kind of mucus and other responses, and it has the control of oxygen to allow you to move forward. Ultimately, the best readout is a biological one, so in collaboration with Honorine Ward here at Tufts Med, she's been able to take our tissues now and infect them with one example of an infectious disease. This is Cryptosporidium, which many of you may be familiar with, and it's very gratifying to see that when she infects with the spores, they go through the entire life cycle, and then they can be collected and reinfected into the intestinal model again. So it's a nice example of how these tools can now be used for new discoveries, because now that we have a system like that, we can start to look at interventions. And we're very fortunate, we just got a joint Gates Foundation award to start to look at therapeutic treatments to prevent the spores from sitting in the intestine and going through life cycles. All right, so, and then again, we switched over to enteroids now, so everything's done. You get all the cell types in there, so you have immune cells, you have the things you want, and as I mentioned, we've already now been innervating the intestinal model as well, and so we have quite a system that I think is going to be very useful for a lot of studies. So we're using this, and we're just getting started. This is Terrence Rowe. He's a PhD student. He's interested in colorectal cancer, and so having a good intestinal model like this, we can start to switch out different cell types in terms of the different kinds of cell sources for these kinds of cancers. And so he's working to collect these cells and build the model, but now infusing the cell types in to look at how the cancers develop. And then long-term, he has some ideas on how he wants to study their responses. So we're excited about this because now that we have the relevant model, we can start to make some progress on the cancer side here. All right, so let me close real quickly just to say this is where I started. I showed you 3 tissue types that we work on. Hopefully get a sense of what we care about in terms of the highlights at the bottom there. And I think this is just the tip of the iceberg. I think the tools are continuing to evolve very quickly, both in our lab and many other labs around the country and the world. So take advantage of them. These are good tools to use for the kinds of questions we're addressing in this symposium. I don't want to forget to I put one slide in to mention Irene's work. So Irene is also in our department on the Medford campus, and she does all the imaging that both Charlotte and I talk about in terms of support for these models. And just to give you one sense here, there's a lot of data here, so don't worry about it, but she can follow for us in a non-invasive way. So no dyes, no perturbation to the system, just non-invasive imaging. She can track the metabolic profiles over time. She can track the structures of the matrices, collagen organization, all the things you care about as you study these models. So the collagens here, here's the scaffold as we start out. This is all with 2-photon emission fluorescence and second harmonic generation methods. But here is the work we published together on the 3D adipose tissue. And you go out to a half a year in continuous cultivation. And if you take nothing else away from that picture, it's how it changes over time. So these are not one stop, one time, you have a picture of what's happening. The cells go through changes, and you need to be able to capture that, understand where your system is to know what the readouts are and how significant they are. All right, let me close. I want to first acknowledge the NIH because we've been very fortunate. We've had support continuous since 2004 for our P41 center that we run here at Tufts, and our partner organization is Columbia down in New York, Gordana Vunjak-Novakovic. And so the whole center is about what I talked about today, building tools like this and getting those tools out to you guys. So like I said, if there's something we're doing that can be useful, let me know. I talked about 3 of our tissues just in some aspects of what we're doing with them. You can see the list of other tissue types we have actively going in the group. So if there's something there I didn't cover, feel free to talk to me about those as well. I also want to just thank our students. Number one, students and postdocs. I have an amazing group. Faculty collaborators all around the world who I'm very thankful for. We have support from many organizations. I won't go through all that, and obviously from the NIH to do what we do. So I'm very grateful for that as well, to keep things moving. And I'll stop there and be happy to answer a few questions. Thank you very much. Great. Yeah, great question. So we haven't looked at that in our model. The focus has been more on mechanical damage so far, and also we're starting some work in collaboration with MGH on, I don't know what I'd call it, but flooding with blood to see how it affects cell functions, particularly the astrocytes. So we haven't looked at seizure-related issues. But certainly if that's something of interest, that could be explored using the model in a way you'd like.
I mean, because for epilepsy, the only thing you can use is animal models, because the dogma has been cells don't seize for many years too.
But you can condition them, right, with electric shock and so on, I think. I mean, they do that with animals, right? They predispose them. So I suppose you could shock the model and then start to look at response.
So that and then the second question is a 180 degrees. So do you have any plans for your colon model to, since you can have immune cells in there as well, to look at colitis as opposed to the sporadic colon cancer? So colitis is a major risk factor for the different kind of aggressive colorectal cancer.
Yeah, great point. We have talked about it in the group. We have done nothing along those lines. This model is very, very new. We just published the first paper last year, and more are coming. So not yet. Happy to chat more and see how to approach those things. Yeah, thank you.
Now, just my ignorance will probably be reflected in this question, but is there any evidence of angiogenesis in any of these models?
So the answer is yes. Again, keep in mind this is all in vitro, so it's a little bit odd. Okay, but if you go back to our early work on 3D adipose tissue culture, when we do the seeding, like I told you, you can easily find assembled blood vessel-like structures. So the endothelial cells certainly pre-organize over time. I wouldn't argue those are functional blood vessels, but certainly those structures form. And then it's a question, can you hook them up to plumbing so they're relevant? And those are things we do look at and study. So the answer is yes, if you have endos in there. Yep.
You mentioned that one of the overall goals for all these 3D tissues is drug screening, but I'm curious what you think are the major barriers to implementing a drug screen using them. For instance, you have very complex tissue, you might have a lot more issues with kind of the narrow reproducibility you really need for drug screening, tension between kind of miniaturization and still getting the complexity that you need, the length of time to look at chronic effects versus the length of time of, so what are the major factors that are going to help with those tensions?
So it's a great question, Josh. So you hit the nail on the head. To me, the biggest issue is to take the tissue. So I say microfluidics aren't good because they're too simplistic, right, for a longer-term screening. But in fact, they're good because they're scaled at a level you can hook up and run online. So that's the advantage they have, and that's why it's an easier system to start with. Ours have to be scaled down, but you don't want to scale them down to the point you lose relevance. So it's really, to me, an engineering problem that needs to be solved, which is where can you scale them to still be relevant, and then hook up multiple tissue types to each other, and then to integrate the imaging online, as well as flow through downstream metabolic profiling with mass spec and so on. So there is nothing that can't be done. It's just a matter of the resources to engineer all that into a flow-through online system. And then you can imagine everybody would have their cells harvested, 50 tissue types grown, online screening of every drug, and then you have a database, you're ready to go. So that's the future as I see it. Okay.
Okay, go ahead.
How similar do your tissues remain to the tissues that you're trying to model after several months? So you mentioned that they're very dynamic. Are you able to track those changes so that you know that it's changing the way that normal human tissue would, or is it separate?
Interesting question. Do you mean do they age, or do you mean that they look the same as when we started? So I can say for the fat, it looks the same, unless we've overdosed or underdosed by design, so the fat globules shrink or swell. Those are things we can track. But otherwise, I would say those tissues look pretty much the same. It's hard to tell with the others yet, I'd say, too early. Check back in a year and we'll compare notes. How's that? Okay. It's a great question, though.
Well, that's some additional questions, so let's go ahead, Evan.
Cells and tissues live in a pulsatile, stress-stricken environment. You look at that dimension
Yeah, so again, I had to just cut down today. So for example, the intestine model, we have a whole bioreactor with flow through for the lumen, and it has mechanical flow, so it's like peristalsis. So that's all functioning up in the lab. You can come take a look. So yes, we consider that for every tissue type.
The idea about moving any compartments to the right, have you considered about putting them, making part of your model for the colon because of new construction? Some of the other stuff actually taking it and translating it back into animals, immunocompromised animals, doing drug screens that way, or alternatively even, yeah, I guess just even the drug screens alone is something that you can do. And also actually the influence of the immune system, because that's going to obviously be part of the in vitro setting. We'll use a meal like a fish animal in that case, basically putting them back into the animal so they can actually interact with the environment.
Yeah, it's a really interesting point. When we, I'll tell one short story. So when we started both the brain and the intestine work, there was never intent to go in vivo with them. It's simply in vitro models. But in fact, we have a collaboration at UPenn on the brain system, not the intestine yet. And it's very instructive from what you just raised because they wanted to look at repairing damaged brain tissue by implanting good brain tissue. And so we prepare the tissues, we get them down to UPenn, they implant them in the animals, and they all fail. At least the first iteration, and they failed because of immune response, right? And it was very instructive because we had to go back for thinking about transplantation. That's not our goal, but for them, it was their goal. We had to think about how to redesign the donut model to control inflammatory responses at the interface so that you could give the tissue a chance to take. And so we've redesigned for that in mind to see how well it will take in this round of in vivo studies. That's the extent that we've used. There's no reason you couldn't expand in the directions you're thinking about, but I'd rather work with others who want to do that because that's not something we really specialize in.
I think it would work in the cancer setting, certainly, especially if you want to get the animals, frankly. Can I ask one more quick follow-up? So with all those silk fibrine scaffolds, are you coating them with anything before you put your cells on them?
Almost always, yes.
Yeah, is it just matrix or?
It depends on the tissue type, right? So in the brain systems, it might be a fibronectin combination with a few other things, but it depends on what you're using, what tissue you're after. Yeah.
Okay, one last question.
In your adipose model, have you either intentionally or spontaneously come across malignant transformation?
A great question. Not yet, but we haven't really, I mean, none of the students have mentioned seeing it. It's a great point. It'd be really interesting over time to stimulate and see if you can create this as a spontaneous mutant. I like that idea. Yeah, thank you.
Okay, let's thank David again.
The next speaker we're going to have is going to continue the convergence theme, and that speaker is Josh Kritzer, who's an associate professor of chemistry in the Department of Chemistry, of course, at Tufts University. And he's going to tell us about different targets require different molecules, chemical approaches to unlocking cancer therapies.
Hello, everybody, and welcome. Thank you very much for coming out to listen to kind of a smorgasbord of different areas of research. I really love the theme of convergence. Convergence to me isn't just a buzzword, it's kind of how I live my life. I started out as a chemical engineer, and then I went on to get my degree in biophysical chemistry and synthetic chemistry, and then changed gears and did a postdoc in genetics. And so, this is how I see convergence in the new century is not just convergence in terms of different specialists need to get together to work on difficult problems, but actually that you shouldn't see yourself as a chemist or an engineer or a biologist, that you should figure out what problems you want to solve, get the education and the training you need to solve them, and then go out and solve that problem. And hopefully today I'll convince you that I've done that. And if not, you can just pretend I've done that because I did the training. And so, I've also actually, I'm a believer, obviously, in convergent approaches to problems. I'm also a believer in convergent approaches to teaching. We just published a perspective about some changes we've made over at the Medford campus in how we teach chemistry to make it more convergent, but that's not the focus of the talk today. Okay, so the title of my talk is different targets require different molecules, and as Charlotte implied earlier, is that if you want to start targeting things that we're are currently considered undruggable or can't be targeted by small molecules, you have to start throwing new kinds of molecules at them. And that's where the chemists come in, is trying to find new areas of chemical space, new types of molecules to throw at these targets to make them druggable. And so here's a classic example of a currently druggable target, very cancer relevant, obviously, but I would like to flash back to the time earlier that Phil was talking about when RTKs were first discovered, and they were, there was a lot of disappointment because they were viewed as undruggable targets. Because how are you going to target an ATP pocket, right? There are ATP pockets everywhere, right? And nowadays we consider it one of the great successes of targeted medicine, in that you can actually figure out what RTKs are expressed in a certain cancer, and we actually have small molecule drugs to target them. Okay, so why do we love small molecule drugs so much? It's because of their small size, the general hydrophobicity, which allows them to slip through cell membranes, slip through our intestines and our liver unscathed and get to the target, right? So we love orally bioavailable small molecules. Definitely the workhorses, even ugly ones like amanitin. It's still a beautiful thing that you can take it orally. But if you want to target other interactions, like, you know, RAS and SOS, an example that was also given by Charlotte, as something that we would like to target, but generally was viewed as undruggable, right? Undruggable until somebody discovers a molecule that can target it, of course. You know, what is the problem here? The problem isn't that it's some magical force field around it. The problem is that you'd like to block this interaction, but this interaction is very large and much more polar than the active site of, you know, an RTK. So you can't just wedge a greasy small molecule and expect it to block that protein-protein interaction or PPI with very much affinity, right? You need to be able to cover a lot larger part of that surface. And so, you know, the molecules that are currently FDA approved to do these kinds of things are largely antibodies and proteins and other things that aren't orally bioavailable and can't access intracellular targets. Right? So if your target is on the outside of the cell, you can target it using an antibody. But if your target is RAS or something else on the inside of the cell, there's no current chemical matter that bridges that gap. And so there's a lot of interest in trying to look in that middle space of molecules that have some of the features of small molecules, but have some of the molecular recognition features of larger surfaces, more polar surfaces. And that's where chemists like me come in, right? So we want to target these new types, new classes of cancer targets, but we need new molecules that can balance what we love about traditional small molecules and what we love about proteins and antibodies. And that's where my research comes in. This is a classic example, probably the most successful example of a protein-protein interaction that's been targeted, the interaction between P53 and MDM2. And so there are, you know, FDA-approved drugs after 20 years of trying to inhibit this protein-protein interaction. And so there are FDA-approved small molecules for this protein-protein interaction. However, it's also a breeding ground for new strategies on the basic science level for targeting PPIs. And I have a couple of examples here, not randomly chosen. These are examples of conformationally constrained peptide scaffolds. Here is a small beta hairpin, and here is a stapled alpha helix. Stapled because two of the side chains are locked together. And the virtue of being able to use these constrained scaffolds is that you get the benefit of a protein in that you can use any sorts of side chains you want in a very complex three-dimensional epitope to bind your target, but you get some of the advantages of small molecules because you're conformationally restricting the shape. And so you don't have this floppy, unfolded peptide that's going to have low affinity or get metabolically degraded as easily. And that's the purpose of this head-to-tail cyclic peptide or this side chain to side chain stapled peptide. The virtue of these is also that they become more cell penetrant, although the mechanisms by that are very poorly understood, and I'll get back to that at the end of the talk to tell you how we're applying this. And so this is from a review in 2014. I will tell you that my graduate work was also mimicking P53 to target MDM2. This is a classic sandbox for targeting protein-protein interactions. But you'll notice what we're trying to mimic here is a helix. And so the question is, have people generalize this to other types of interactions? Because we aren't lucky enough to have every single protein-protein interaction involving cancer involve a helix. Certainly, BH3 domains and P53 are well-publicized examples. But can we do this you know, if this is 2016 and we can just staple using creative chemistry a peptide into any shape we want, why are we still doing helices? And the answer is very few people are even trying to apply this to non-helical structures, and that's where my lab comes in. We want to be able to take any 3D structure and use chemistry to lock a small peptide into that structure to target a protein nobody's ever targeted before, hopefully make them cell penetrant and useful for chemical biology. My lab has a bunch of different projects, most of which, thankfully, you guys aren't going to hear about today, but I will focus in on some of the really cool ones. One of the ones I'm not going to talk about today are conformational mimics of phosphotyrosine. It turns out that when you conformationally constrain a peptide, you can have ways of putting together a recognition epitope that you wouldn't be able to do if you were just trying to mimic one chemical group. For instance, phosphotyrosine, if you take a benzyl phosphonate, which perfectly mimics the phosphotyrosine, you can inhibit any SH2 domain or any phosphatase you want, but you won't get any selectivity, right? Because it just looks like phosphotyrosine. And so we have ways of mimicking phosphotyrosine using a discontinuous epitope that when you lock the peptide into the correct three-dimensional orientation, it can bind SH2 domains, it can bind phosphatases. We can even take the 4G10 phosphotyrosine antibody and use our phosphorus-free molecule and fool the antibody into thinking there's a phosphotyrosine there. So that's a really cool place in which conformation can get over some of the medicinal chemistry barriers to that field. And I'm really happy to find out that we're going to hear more about phosphatase inhibitors in the next talk. I have another project in infectious disease looking at inhibitors of proteases. Kritzer, you might say, proteases are druggable, so why are you working on them? We know how to drug proteases. Well, I'm working on a class of proteases called IgA proteases that are secreted by mucosal pathogens, where here's the protease domain, and then there's all this other scaffolding that's involved in molecular recognition that is very, very poorly understood. And so this is a class of proteases that has not been amenable to traditional protease drug discovery, and that's why we're interested in it. But what I will talk about today are two other projects. One is a general strategy for trying to find protein-protein interaction inhibitors. Again, instead of stapling helices or sheets, which is really where the last 20 years, the birth of this field has come from, I want to generalize this to any three-dimensional loop. Right? This is 2016. We have clever chemists over at Medford. We should be able to take any 3D shape and lock it into the correct shape, and not just helices and sheets. Okay, so I presented this challenge to my students, and they said, great, what should we be targeting? And I said, I don't know. I was in graduate school, and I was mimicking helices, and everybody knew if you were mimicking helices, you'd want to mimic a BH3 domain and target BCL-XL or MCL1, or you'd mimic P53 and target MDM2. There are established protein-protein interactions for helix mimics. There are no established protein interactions for trying to mimic a loop, right? It's just, not an area that people have really dug into. And so in the theme of convergence, I want to include this bioinformatics project that we did to take the entire protein data bank, the entire PDB, and look at every single protein-protein interface and pull out all the loops that mediate protein-protein interactions. So we've had to figure out what the most promising targets were before we even did this. Now, I'm not going to go all over this. I will say that one of the benefits of being in Medford is I have very talented undergraduates who know how to code and write programs, which is great. I actually did some coding in my undergrad, but it was in Fortran, so it's not applicable to these kinds of problems. And so in work that was published in 2014 and a follow-up just very recently, we took the entire PDB, filtered it using our own programs and using Rosetta-based computational alanine scanning, and got out what we like to call hot loops. These are loops that are at protein-protein interactions that are hotspots for that interaction, meaning most of the delta-delta-G of binding is in that hot loop. And the assumption there is, okay, great. If we can mimic this loop, we'll have something that binds almost as well as the original protein, only it'll be much, much smaller and a little more drug-like. Okay, so that's where we got our targets. There's a lot of bioinformatics under the hoods here. For instance, if you do this with helices, you get a bunch of helices that all look like each other. If you do this with loops, you get a bunch of loops that don't look like each other, right? There's many, many different kinds of loops. And so we could use sophisticated clustering algorithms to try to find some that kind of look like beta hairpins, right? And then others that don't. And that was not clear what we would find before we applied some statistical analysis to that. But of course, there are lots of drug targets too. You don't need a program to tell you that this hot loop on NRF2 is probably a pretty good target for macrocyclization, but maybe some of these others, it's not so clear exactly what part of that protein-protein interaction you'd want to mimic with a macrocycle, right? And that's what I think the true benefit of this was, is that we pulled out some obvious targets, we pulled out some non-obvious targets. And a lot of these are, I think every single one on this page is relevant to cancer. Okay, so now we had a sandbox to play in, right? Now we actually figured out what targets we could target using this approach. But the answer is, how do we actually make inhibitors? And so the answer is the second part of this graphic, which is once we identify the sequence of this loop, how do we actually make a macrocyclic inhibitor? And we're gonna use some clever chemistry for that. I'll give you an example of work that was just published from a graduate student who just graduated, Tim Siegert, who works now at a pharmaceutical firm in the Boston area doing peptide therapeutics. This is a protein-protein interaction from the PDB. It's stonin 2. This is a portion of stonin 2 bound to an EH2 domain of EPS15. And it's involved in endocytosis. If you genetically disrupt this specific interaction, you see decreased clathrin-mediated endocytosis, slows down things like Ebola entry, and also cancer invasion. And so our program took out this hot loop here. This is an amino acid sequence and predicted the hotspots. And so we had kind of the three-dimensional structure that was required for binding EPS15. And so you would think that this is 2016 and I should be able to computationally just figure out how to lock it into that three-dimensional shape. And I do have I collaborate with David Baker at University of Washington and Yushan Lin in our own department at Tufts Chemistry, who does molecular dynamics simulations. And from talking with them, I will tell you that in 2016, we can design entire proteins that nature, never thought of, right, and have them work, right? And that is an easier problem than designing a 10 amino acid macrocycle and getting it to fold in water, right? Because the tolerance is much, much less forgiving, right? You don't have secondary structure and tertiary structure to get you down into a well-folded protein. Okay, so I can't just model it with wooden balls and sticks or with pixels, right? What can I do? Well, I'm a creative chemist, so I'm gonna make a library, right? I'm gonna make dozens or hundreds or thousands of these, and I'm gonna take this recognition domain, and I'm just gonna lock it every which way. And then I'll do a screen to figure out which one is locking it. So instead of screening random chemical matter in this space to try to find a hit, what I'm doing is taking a biological recognition epitope and saying, I have no idea how to lock this into the correct shape. Let me try 500 different locks. Okay, this is the chemistry. I have to show some chemistry. I teach chemistry, I love chemistry. Suffice it to say that there's a lot of different ways to cyclize peptides, and most of them are terrible. And do not work at very high throughput where you can make dozens at a time or hundreds at a time. This method it allows us to make an arrayed collection of stapled loops, which we can then screen for target binding either using a biochemical assay or in cells. In this case, here's our winner. Again, there was no chemical matter other than the native protein that was known to bind this protein-protein interaction. We started in a sandbox where there were no other pails or shovels, right? And so we had to invent our own shovel. And here, right off the bat, we find a sub-micromolar ligand, which is great. I'm not showing you all the structure-activity relationships, but you'll have to take my word for it that if you change the shape of this linker, you ruin binding. If you alter some of these computationally predicted hotspots for binding, you lose binding. That was all published, so you can keep me honest by going and reading my paper. And we have selectivity for this particular EH domain over other EH domains. And that's another thing I really like about the macrocycle approach is, if you're making a molecule, very small molecule to wedge into a target site, right? And that target site is similar for all other targets, like a phosphatase or these EH2 domains. You're not gonna get very much specificity. But if you have a larger surface of shape complementarity, right, then you're gonna have a much more specific lock and key interaction or an induced fit interaction. We also do some structural biology, so we're growing crystals of these guys, but we also have an NMR structure of just the peptide, which is shown in beige here, and it overlaps very well with the target that we pulled from originally started with the PDB, especially the hotspots. And so, those of you who know are in the biophysics field, hopefully you had an audible gasp just now when I told you how well structured this is, because a cyclic peptide of 10 amino acids in water should not be very well structured. This is not something that generally forms well in water, and yet we have a very, very tight structure, and we can model what we think the complex looks like, and even propose a role for the staple in molecular recognition over here, which was unanticipated, and then test that and try and grow crystals so we can see that firsthand. So that's an example of this approach, and I also hopefully give it as an example of the type of targets that I'm interested in. I'm interested in going after targets that other chemical biologists have kind of ignored. I mean, one of the things that jumped out at me when I looked at my database of loop-mediated protein-protein interactions is how many of them were involved in vesicle trafficking. And any of you who have used compounds to try to block vesicle trafficking, you know, you have your bifilomycin, you have your chloroquine, you have these molecules that biologists use to block different pathways. And I have a dirty little secret, or maybe it's not such a dirty little secret, which is those are not selective molecules. They block every single thing. They're pump inhibitors. They change the pH of all these different compartments. They're not selective tweezers that you can use to block one of these vesicle trafficking pathways and not the others, right? And these vesicle trafficking pathways are supremely important in cancer, especially when you're talking about cancer invasion and metastasis, right? Where you have a cell that spontaneously polarizes and starts to push through into surrounding tissue. So my vision for this is to actually go after a lot of these interactions which are not mediated by helices, which is probably one of the reasons why they've been ignored by chemical biologists. They're mediated by loops, and so to apply this to a lot of different pathways, to give biologists and cancer researchers the tools to block one of these pathways without affecting any of the others. Okay, so just to hit that home, when I learned about cancer biology, and in fact, when I teach about cancer biology, we talk about the different circuits that have to get interrupted in the cell. You know, proliferation circuits, tumor suppressor, survival checkpoints, metabolic circuits, all of these things that we think, okay, you have to accumulate defects in all these circuits before you can get cancer. And that's a great way to think about it. But there's another way to think about the cell, and that's as a transport network, right? And that's, in that case, you should be able to block specific vesicle trafficking pathways and have similar effects on the overall biology of the cell. So that's what I'd like to do with a new, more convergent approach to this chemical biology problem. Okay, so in my remaining time, I would like to talk a little bit about a project related to autophagy. Everyone, if you haven't heard about autophagy, now has heard about it, so it's good timing to work in autophagy because Wasumi just won the Nobel Prize. He discovered it in yeast, but it turns out that autophagy, it's one of those things that you discover in yeast, and then you're like, holy moly, this is happening in every single one of our cells and has for billions of years, and nobody realized it until you looked at yeast. And what it is an alternative to ubiquitin proteasome recycling pathway, where material is recycled by just engulfing it in a membrane. We call that an autophagosome, and then that's shuttled to the lysosome for degradation. So very simple idea. Turns out cells are using this all the time. And one of the master regulators here is Beclin-1. And there are, you can't really, it's very hard to inhibit autophagy because it's such a critical pathway for a lot of different cell types. But inducing autophagy, we induce autophagy under caloric restriction, under exercise. So there's a lot of interest in inducing autophagy. And you can induce autophagy through upstream regulators. But of course, mTOR, AKT have a lot of different effects, right? So we'd like to find selective inducers of autophagy, things that induce only autophagy and not everything else that mTOR signals, right? So can we find selective inducers of autophagy? The answer, of course, is yes. Otherwise, I wouldn't have this in my introduction. How does autophagy relate to cancer? It's been known for a long time that autophagy will support actively growing metastatic disease, just like chaperones and the proteasome support it. You know, basic housekeeping is necessary for these. But in carcinogenesis and progression, it's found that actually losing some of your autophagy regulation, including Beclin-1, accelerates the accumulation of transforming mutations. And so there's a theory that if you can add autophagy activators at this point, that you might be able to slow down or stop tumor growth. The problem is, and I specifically took a review from 10 years ago because this has been in the ether for 10, 15 years, and nobody's been able to test this because there are no good selective pharmacological inducers of autophagy. And that's how this really relates to cancer. I don't have a lot of time left, so I will tell you that I'm collaborating with Beth Levine, UT Southwestern, one of the researchers who first discovered and described Beclin-1. And she quite accidentally discovered a peptide derived from Beclin-1 that induces autophagy. The problem with this peptide for using it in vivo and using it as drug development is that it only gets into cells because of this purple sequence here called TAT. This is a polycationic, positively charged sequence that kind of cheats peptides into cells. And so it's not going to be really useful as a basis for drug development unless we can get rid of that. And so I was foolish enough to decide to try. And so I started with that hot loop that she had. And I did this same strategy where I stapled it every which way. And then here we're using cell-based assays to look at whether these stapled peptides can induce autophagy. And what we find is a hit here that can induce autophagy using 2 D-cysteines and this orthoxylene linker. And it turns out if you use 2 L-cysteines, it doesn't work anymore. So you have this beautiful stereoisomer control that tells you that it's probably doing something pretty darn specific. This is to show that we can see autophagy being induced in vitro and in live mice. These are LC3 GFP mice, so each of these puncta, hopefully you can see, are autophagosomes being trafficked to lysosomes. So when you induce autophagy in these LC3 GFP mice, you start to see these autophagosomes form in large amounts, and that shows you that you're inducing autophagy. So everything in these green dots is being targeted to a lysosome for degradation. And our current disease model includes a lot of different things that you would expect from autophagy inducers, like viral clearance, bacterial clearance, clearance of lipid droplets in fatty liver, but also finally testing this idea that you can have autophagic cell death, and that will actually slow down tumor growth. We actually solved the NMR structure of this peptide, and we realized that it's helical. I told you I don't want to work on helices, but here we go. I found a new helix, and it's enforced by a novel staple that's completely different from all that helical stapling technology I told you before. And so here we have a cell-penetrant helix that actually gets into cells using a completely new type of chemistry. And the very last thing I want to tell you about is in dealing with this kind of middle space, right? This space where you can now target protein-protein interactions using constrained peptides, but these are maybe too polar to get into cells very well. We've come up upon this engineering problem of how do you actually measure how much of your peptide is getting into cells? Not getting into endosomes or getting into other cellular compartments, but how much is getting into the cytosol? And we've developed a brand new technique that allows us to do this in a quantitative relative manner. So you can take any exogenous molecule and actually figure out how much is getting into the cell. So we're doing this with peptides, with proteins. You can do this with viral particles, antibodies, if you don't want them to get into cell, to measure how much the cell is degrading your antibody, nucleic acids, any sort of therapeutic or biomolecule that you're actually have the question, how much of this is actually getting into cells when I add it exogenously? Such a simple question, and yet there is no good quantitative assay for this, and I think I've finally cracked that nut. So I'm very excited about that. And it just goes to show that that engineering background and that convergent approach, right, leaves you open to solve the problems that you actually encounter along the way instead of just shrugging and saying, well, there's no good tools for that, so I guess we can't measure it. So just to show what all the different fields that these types of science requires, convergence to me is not just a buzzword. It's the only way to solve these complex problems, both within my lab and with all my collaborators. These are actually the people who do all the work, of course. Tim Siegert and Mike Bird are a grad student undergrad who worked on the EPS-15 project. As I said, Tim Siegert works for a pharmaceutical firm. Mike Bird is now a PhD student at Scripps. And Leila Peraro, there she is. Leila Peraro and Yang Mei are working on the autophagy project. I'd like to thank my funders, federal funding and private funding, and you for your attention. And I'd love to take any questions. Yes. Yes, that's a great question. So the question is, have we looked at more than just kind of autophagosome generation to show that to show some of the downstream consequences of autophagy? And the answer is absolutely. I haven't shown it, but we've shown that we see turnover of proteins like P62 that are known to be turned over in autophagy. We've tested it in a mammalian cell model of Huntington's disease, where you just take, not neurons, but just HeLa cells express 102q Huntington, right? And they get these aggregates. And then the aggregates are very hard for the proteasome to turn over. And so they have to be turned over by autophagy. We can see clearance of these aggregates when we add our peptides. So we definitely can see that. Yes. Yeah, absolutely. So these are the disease models we're working on now, and this is an ever expanding list. Beth Levine, UT Southwestern, initial peptides have been tested in some of these models already, and those results are just starting to be published. So there is published work for that original cat-linked peptide that's 2.5 times as big and not going to be a drug anytime soon. But even those, we see in vivo phenotypes reducing some of these issues like protein deposition in the kidneys, lipid droplets, things that are known to be, the biology of these really suggests that inducing autophagy will help clear some of these aggregated lipids or proteins or organelles. And so we are now testing our peptides in those same models. Yes. That's a fantastic question. So the question is, do we test for stability in vivo? And in fact, chemists will point to this thioether here. If you've ever dealt with methionine oxidation, right, you know that this can be chemically labile. So these are relatively protease resistant. We've looked at that, but they do have a metabolic half-life. We definitely see effects at least for 6 to 8 hours. So we know that they're long enough to see an effect in terms of a normal dosing regime in vitro or even in vivo, where you look for effects, where you're going to dose every 24 hours or something. So the answer is way, way better than normal peptides, which will be great half-life in minutes. We know the half-life is hours, but medicinal chemistry can definitely improve it from there. Yes. Yes. Okay. Great. So that's great. So I didn't get to it. So what we see here is a small molecule control, and we have a nice sigmoidal curve showing that even starting at about 10 nanomolar, you can see cell penetration. This is a relative scale. We're working on calibration curves to make this absolute, so we can read out exactly the concentration. But it is highly quantitative and reproducible. And here we have our autophagy-inducing peptide that's about 100-fold shifted. And so you might say, Kritzer, your peptide's not very good at getting into cells if it's 100-fold worse than a small molecule. And what I would say is that peptides aren't supposed to be able to get into cells at all. So being 100-fold worse than a small molecule by cell penetration is actually a great starting point. The other thing I would mention is that right here at about 5 to 10 micromolar is where we start to see autophagy induction in vitro. And so that strongly suggests that if we can just improve cell penetration by 4-fold, we will actually improve autophagy induction by 4-fold. And so now we have an independent axis to improve the efficacy of peptide therapeutics. And again, you would think, well, don't drug developers improve on this axis by improving the cell penetration? And the answer is, it's very hard to do without a quantitative high throughput assay, which we have. Great question. So can this actually only, and I can't talk about the details because we're filing on it, but the paper will be out in another month. It's under review right now, so it won't be long before I can talk about the details. But it does not measure efflux. It actually just measures how much got into the cell in the first place. However, it does tell the difference between different organelles or cytoplasm. So there is no interference from being stuck in the endosome, no interference from being degraded in the lysosome. So. I'll tell you a little bit more about it. another time. But I can't, I hate that right now. You always promise that you won't do what your parents did, right? The wrong things. And so I used to be in the audience and be like, oh, I can't believe this person's presenting data, and then like I can't talk about what the molecule is or whatever. But I was really excited to talk about this. I'm looking for collaborators to test this in different areas, and that's why I wanted to talk about it today. And I'll be free to talk about the details really soon.
It gives me great pleasure to introduce the last speaker of the session today before our two lunch panels. And that, of course, is Bill Sellers. Bill, I think, needs no introduction for many of you. You know him, particularly if you're in the Boston area, from what was his former position as Vice President and Global Head of Oncology at Novartis, at NIBR, actually, Novartis Institutes for Biomedical Research. Bill purports to be retired now, but I can't imagine a scientific mind as fantastic as this one is actually retired. So I think you're going to hear some really interesting thoughts from Bill in his talk today. And the title is From the Identification of Cancer Vulnerabilities to Therapeutic Development, and it is not present. Oh, it's because you're asleep.
Thanks for inviting me, and I'll just say one other thing. I'm from a long family of Tufts alumni. My father, my mother, my sister, and most recently my son in 2015. And I'm sure you all know my son because you probably are all at the Tufts hockey games watching him play hockey. That's a joke because only family members were ever at those games. So I'm quite the reductionist. Even though I like high throughput biology, we tend to apply it in a very reductionist way. And the two questions at Novartis we were most interested in asking are, what are the genes that are required for the maintenance of the cancer phenotype? And I use the word maintenance specifically, despite the fact that it might be better to prevent cancer or stop its initiation or block its metastatic process. The 99.999% of patients being treated today are being treated because they already have cancer, and we need to try to eliminate it. And then the second question, which I think our first speaker, our last speaker got at, is how do we drug these? And I would share his enthusiasm for going beyond what is considered the sort of druggable set of genes as we know them today. So over the last, really about the last 10 years, and Dave Ruddy is here in the audience who's experienced every one of our transitions along the way of trying to discover cancer essential genes through the use of first siRNA and shRNA. Most recently, we've been using, like many others, pooled shRNA screens to try to identify those genes that are critical for cancer maintenance. And the way we do this is to synthesize large pools of shRNAs on chips, to clone those into plasmids, use those plasmids to generate lentiviruses. The lentivirus pool is then used to infect cancer cell line. That cancer cell line is then allowed to double over time, and at the end, we count the abundance of each shRNA in the population by next-gen sequencing. And so you can imagine, this is just a schematic diagram of how this might work. At day 0, we imagine the abundance of all shRNAs is relatively equal. Actually, it's not equal, but there's a distribution. And that distribution is going to change based on the effect the shRNA has on cell viability. So if, as in this middle red column, the viability of a cell is adversely affected by an shRNA, its sequence will be depleted from the population, and the abundance of the sequence in the next-gen sequencing output will go down, versus in green, where this shRNA would have an enhanced effect on cell viability, and its abundance would grow. And of course, the vast majority, really, we think, are not going to change because they're not going to have an effect on viability. Now, as I said, we've been doing this for a long time, and some of our first attempts were to use small shRNAs against a small number of genes, so 5 shRNAs per gene, 1,200 genes in 250 cell lines, and I'll just summarize that by saying that data was too noisy, even though it was a lot of work. Then, together with our colleagues in the Developmental Molecular Pathways group, we did a small pilot screen where we targeted 17 shRNAs per gene, really this being enabled by the sort of drop in the prices for oligonucleotide synthesis on chips, targeting 385 genes in the epigenome in 80 cell lines. And that data was really quite nice. We discovered BRG as a synthetic lethal with BRM, but the data set itself was too small. But the two experiments together led us to conduct something that we called Project Drive for deep RNA interrogation of viability effects in cancer, where we targeted about half the genome, 7,500 genes with 20 shRNAs per gene, and we did that screen across 390 cancer cell lines. So how do we take those 20 shRNAs and convert them to an output at the end? Of course, each shRNA has its own pattern of on and off target effects. And one underlying assumption is that the off target effects will be relatively non-coherent because they'll be off target, and that the on target effects would be relatively coherent. And with 20 shRNAs per gene, you'd be able to identify a coherence among sets of shRNAs that reflected the actual on-target effect of depleting a certain gene product from the cell. And so using an algorithm developed by the Broad Institute called ATARIS, we take the pool of shRNAs and create a gene score for each gene in that population for each cell line. And then that ATARIS score is used here in a statistical model to show and plot the effect on cell viability for depleting that gene. And here I'm showing you the concordance of BRAF viability effects with BRAF mutation. You can see the concordance is quite strong. So this is just a plot to show you what it's like to go from the 20 shRNAs and why you might worry about just using 1 in your lab or 10 or 5. This is all of the shRNAs against KRAS against every cell line. And I think you'd be hard pressed to go in there and pick out 5 that you might want to use as your definitive loss of KRAS experiment. But if you now convert that from each individual shRNA to the ATARIS score, and then color each cell line by whether it is or isn't KRAS mutation, you can see that the vast majority of cells whose viability is affected by KRAS are mutant for KRAS. This may seem sort of obvious, but at the time we did this in the literature, there were people going back and forth on whether KRAS dependence was uniquely associated with KRAS mutation versus other features of how you might activate a cell. So I'll show this. This can be somewhat controversial, but with this data, we can now go look at all of the publications of synthetic lethal effects with KRAS. So this is the data I just showed you now, shown as a little graph. And these are 5 publications reporting different genes that were thought to be synthetic lethal with KRAS mutation, all starting from a much smaller pool of shRNAs. And you can see in the larger data set, we're not able to recapitulate the dependence of cells on either the genes to the left on the KRAS mutation status. It's not to say the data are wrong in the paper, it's just that they're not reproducible across a large scale of shRNAs and across a large scale of cell lines. And I think these were largely false positives based on a small number of shRNAs being used in the original discovery. So here's an example of just one class. We've described sort of in our own work 4 or 5 different classes of genes that can come out of this large-scale analysis. The most obvious one is the class of genes that are oncogene drivers and hence have genetic dependence. And I'll just show you again BRAF, NRAS, PIK3CA, and then even FGFR2, which has only 2 or 3 cell lines that had FGFR2 alterations. The data are robust enough to pick those out as uniquely dependent on FGFR2. The second class of genes, and probably the largest class of genes that come out as specific eliciting specific dependence in certain cancers, are those that encode lineage-specific transcription factors. And I'll show you here MITF, which is the master regulator of the melanocyte lineage, something myself and Levi Garraway published on a long time ago, is uniquely required for melanoma in cancer. Similarly, SOX10, which is also in the melanocyte lineage, is uniquely required in melanoma. And in this case, because we also have the DNA methylation status for every cell line in the collection, we can specifically show that the reason SOX10 is required in these cell lines and not the ones shown in black is in these cell lines, the promoter of SOX10 is unmethylated, where in the rest of the cell lines it's all methylated and off. So they don't care about SOX10 because they've never expressed it in the first place. We're able to recapitulate some other findings that have been previously published, for example, the requirement of MIB in the hematologic malignancy space, and then work from Miles Brown that showed FOXA1 was a specific breast cancer, hormone receptor positive breast cancer dependent gene. So for the rest of this section, I'll just talk about a discovery we made coming out of this data set, one of the novelties we were able to publish. And this involved the interaction between the arginine methyltransferase PRMT5 and a metabolic gene known as MTAP. And we're calling this a collateral synthetic lethal interaction. And you'll see sort of what I mean by this. This was published recently in Science. The first author is Mavrakis. So the reason we were interested in this metabolic gene MTAP, methylthioadenosine phosphorylase, is that it's been previously known, and people have looked at this in the past, that MTAP is closely located near CDKN2A, or also known as P16, on chromosome 9. And because of that, and because P16 is the target of deletion in so many cancers, MTAP is co-deleted along with P16 in a high proportion of cancer cells. So shown here in orange is the frequency of P16 deletion, or CDKN2A, and shown in yellow is the frequency of co-deletion. So you can see there are deletions that affect P16 that don't affect MTAP, but a huge proportion of those cell lines or cancers that delete P16 also delete MTAP. So what does MTAP do? Well, methylthioadenosine phosphorylase is an enzyme in the methionine salvage pathway that metabolizes methylthioadenosine. Now, I'll get to that in a second. And as part of its metabolism of methylthioadenosine, it eventually regenerates methionine, which together with adenosine and ATP generates SAM. And of course, SAM is an important cofactor for lots of enzymes that require methyl donors, including arginine methyltransferases. So we thought or hypothesized that if you delete MTAP, you might first predict that MTA levels should rise in cells. And there have been some published reports of this that we wanted to also confirm. So we took a number of cell lines and by mass spec measured MTA levels in those lines that had MTAP/P16 deletion versus those that didn't. And you can see MTA levels rise substantially in cell lines that have a deletion of MTAP. And I'll just draw your attention to this pair here where we used CRISPR to knock out MTAP in a cell line that had wild type MTAP and showed that the selective knockout of MTAP leads to a rise in MTA levels. So it's not just this big deletion that's doing it. You can recapitulate the rise in MTA levels by a specific knockout of MTAP. So the question is, if you go back to this, if MTA levels rise, if this is cut off, would you become dependent on other enzymes in the methionine salvage pathway? This was the original question. And so we looked specifically in our data set to compare those cells that had an MTAP deletion versus those cells that did not, to ask, were their shRNAs selectively being depleted from the population of cells that had an MTAP deletion versus those do not? And you can see one of the enzymes in the methionine salvage pathway actually did score MAT2A, which if we go back a second, is shown here. And we could hypothesize that if you don't have this, you're now not going to even use exogenous methionine in this pathway, and now you're dead because you have no way to generate SAM. But what was really interesting and a big surprise to us was that the most statistically significant finding was that PRMT5 was highly selectively lethal in MTAP-deleted cells. So what was also interesting in this data set, and I think this goes to having used so many shRNAs and having what we think is a really robust data set, is that we're able not only to find PRMT5, but the cofactors that PRMT5 uses to execute its function. So first, PRMT5 is found nearly entirely as a heterodimer with the WD40 protein known as WDR77. And you can see one of the top hits also was WDR77. So we pull out the protein complex in this analysis. That complex, in addition, forms two additional complexes, one with rheokinase 1 and nucleolin, and the other with a splicing complex known, encoded by PICIN, and both rheokinase and PICIN were found as hits. So this gave us really strong confidence that this protein PRMT5 plus its complexes were selectively required for the viability of cells deleting MTAP. So even though I would say that data was so strong, I wouldn't have needed additional validation to be excited. We did go on and do stable lines with DOX-inducible shRNAs to show that you could recapitulate this phenotype. So shown here is a DOX-inducible shRNA to PRMT5. We turn on the doxycycline and we get a knockdown. If we then overexpress a cDNA resistant to that shRNA by virtue of having silent nucleotide changes in the cDNA, we completely rescue the phenotype, suggesting that it's an on-target phenotype. Now, I'm not going to show you the data in the interest of time, but in addition, we showed that in those cell lines we made that were isogenic, where we CRISPR knocked out MTAP, they became sensitive to PRMT5 knockdown, so we know it's specific to MTAP. We also showed that using the DOX-inducible shRNA in an in vivo setting in a xenograft, we could get loss of viability in vivo and regression of MTAP-deleted tumor xenografts with PRMT5 knockdown. So the question we had, of course, is why is this? It didn't really make intuitive sense at the beginning, but if you look at the metabolite MTA, of course, it's a metabolism product itself of SAM, and hence they're structurally similar. And in fact, there had been previous publications that suggested accumulation of MTA might lead to inhibition of the PRMT5 family. So that was a clue that we followed up. And the way we followed this up is to test MTA itself, this metabolite that's accumulating in cells, to see whether it could inhibit PRMT5 or other methyltransferases. So we have a large panel of methyltransferases we tested here. We first tested them with a SAM analog, which is an inhibitor called sinafungin. And you can see that the panel is active. That is, we can detect inhibition of many methyltransferases with this non-specific inhibitor. But surprisingly, MTA inhibited only PRMT5. So for some reason, this metabolite, though it's an analog of SAM or very similar to SAM, inhibits only PRMT5. So we went on to solve the crystal structure of MTA and sinafungin bound to PRMT5, and this wasn't working so great the first time, but I hope it works now. So this is sinafungin, a SAM analog bound to PRMT5, and you can see it's making contacts with this lysine at 333 and a tyrosine at 334, and this is the arginine substrate sticking into the pocket that would normally be methylated by PRMT5. So, yeah, like I said, not working that well. So that's showing what I just told you. And now we're going to switch to the MTA bound structure, hopefully. Yep. So in purple is MTA, so I'm sorry the movie's not working so well, but you can see that it lacks this extension on SAM, and in the presence of PRMT5, this glutamic acid shifts positions here, E435, and now takes the place of SAM in coordinating the lysine at 333 and the tyrosine at 334. So when MTA is present in PRMT5, you get this reordering of the catalytic domain. And what's interesting about this is that lysine and tyrosine are only present in PRMT5 in this methyltransferase family and are conserved between Drosophila and human PRMT5, but are missing from the rest of the PRMT family. So we believe that this explains the specificity of MTA, though we have not done the killer experiment, which would be to take, for example, PRMT1 and convert this to lysine tyrosine, and now show that MTA can be a PRMT1 inhibitor. So that would be a cool experiment to do. So the question is, can we show in a reconstituted system that MTA can actually act as an inhibitor of MTAP? And we did this by first knocking down PRMT5, as shown here, and then rescuing with a cDNA for MTAP. So now we've reconstituted the cell with MTAP. MTA levels should have gone down. PRMT5 viability is erased. But if we add exogenous MTA, we now confer susceptibility to PRMT5 knockdown. So both MTAP deletion and MTA accumulation are required for the sensitivity to PRMT5. So our model for this is that under normal circumstances, PRMT5 is active. Under circumstances where you delete MTAP, MTA builds up and you get a partial reduction in PRMT5 activity that is then rendered a lethal reduction by the shRNA that we have applied to those cells. In fact, if you completely knock out PRMT5 by CRISPR, cell viability is affected in all cells. So this is one case where the shRNA experiment might, in fact, replicate a small molecule versus the complete loss of function, which would have told you this is just going to be a pan-essential gene. I'm going to skip that in the interest of time and switch to the second topic, which is our work on discovering allosteric inhibitors of the SHP2 phosphatase, and this was also recently published in Nature. So another interesting profile can be seen here in the DRIVE data set, which is if you look at SHP2 and you now color code the cell lines by whether they do or do not have an RTK driven phenotype, that is, they have an amplification, a translocation, or a gain of function mutation in an RTK, you can see that SHP2 has preferential loss of viability, not exclusively, but preferential in those cell lines that we classified by this method as RTK dependent. Conversely, if you look now and color those by RAS-RAF dependent, again, it's not perfect, but there's a redistribution of the RAS-RAF dependent cell lines now being insensitive to SHP2. Now that was exciting to us because of the way SHP2 is thought to function. So SHP2 is thought to be inactive in its normal state, held inactive by two SH2 domains. Upon binding to activated phosphorylated RTKs, it's thought that this binding event reorders SHP2's inhibitory effects of these two SH2 domains, creating an active phosphatase, and that in still an unknown mechanism triggers RAS pathway activation. Now, the evidence that this might be important in human cancer comes from the fact that there are mutations in SHP2 inherited in kids with juvenile myelomonocytic leukemia and sporadically or somatically in some solid tumors. And those mutations affect primarily the SH2 domains or the interface between the SH2 domain and the phosphatase domain. And those mutations, unlike, for example, PTEN, which is inactivated by mutations, are gain of function activating mutations in SHP2, suggesting that SHP2 is an oncogenic phosphatase. Now, we're also excited by work from Todd Golub's lab, our own group, and by Renee Bernard's. And to sort of very quickly summarize this, all of these data suggest that feedback bypass of various inhibitors, either RTK inhibitors or MEK-ERK inhibitors, is mediated through either growth factors like HGF, FGF, NRG, neuregulin, or EGF. And Renee's work in particular showed that these feedback loops are SHP2 dependent. So we're struck by the possibility that SHP2 might be a, let's say, universal mediator of adaptive resistance to other, in terms of either ERK-MAP kinase inhibitors or RTK inhibitors. I'm gonna skip that in the interest of time. So we did the sort of typical thing that people do when they start drug discovery. They take the easiest thing you can make as a protein, which is the catalytic domain only. It's active, you don't need to worry about regulatory domains. And we did a screen and we got nothing. Or we didn't get nothing. We got the same things everybody else got, which are cysteine reactive molecules binding to the active site cysteine. Now, to the team's credit, they decided to continue working and make the harder assay, which was to make a full-length SHP2 in vitro. And activate this with a synthetic IRS peptide that had 2 phosphorylation domains, so that now the activity of SHP2 in this assay would depend on the peptide activating through the SH2 domains. And just to show you that how this worked is, if we take a measure of phosphatase activation and we now normalize that fold activation peptide versus no peptide, you can see the truncated protein is not activated further by the peptide, whereas we get a robust activation of the full-length protein by the peptide. And this formed the basis of a screening assay, which was first used in not an HTS, but a pilot screen. So I know 100,000 compounds might seem to many people to be an HTS, but in reality, that's the pilot scale we do prior to HTS. And what was kind of cool is in the pilot screen, we of course have a lot of hits shown here, and this just shows you how noisy screens can be. All of these hits were subsequently revalidated in IC50 assays. 4 of those hits validated, including one called SHP836, which I'll show you the structure of. So SHP836 is shown here. It had an IC50 out of the primary screen of 5 micromolar. You can see here, it does not inhibit the catalytic domain only. It only inhibits the assay in which we're activating SHP2 using the doubly phosphorylated peptide, strongly suggesting this is not a pure catalytic inhibitor. Using that assay, further optimization was done, not a ton, to get to what we're calling SHP099 in the publication, which is a 71 nanomolar inhibitor of the peptide-stimulated SHP2. So how does this work if it's not binding to the catalytic site? Well, obviously it has to have something to do with the SHP2 regulatory domains, and I'm hoping this movie works better. So shown in green and blue are the 2 SH2 domains that I showed you earlier, and flying in from the left is the small molecule, and you can see it binds in an interface between the 2 SH2 domains and the SHP2 catalytic domain, which is all shown in brown. Now you'll see that the positioning of the second SH2 domain, or the N-terminal SH2 domain, is such that there's a loop. It would be interesting if there's a loop from our prior speaker that in fact occupies the catalytic domain in this conformation and blocks access of substrates to the catalytic domain. And what's particularly cool is if you look at the interaction of this molecule, it actually makes hydrogen bonds with all 3 of the different domains, the 2 SH2 domains and the catalytic domain. And we're sort of calling this a molecular glue for SHP2. So does this validate the genetic experiment we showed in the shRNA experiment? We haven't profiled this against that full panel of cell lines, but in leukemia cells shown here, 70 different leukemia cells, those that had RTK alterations such as JAK2 or BCR-ABL were sensitive to SHP2 inhibition versus those that did not have such alterations. And similarly, in colorectal cell lines, which don't have genetic alteration, we're able to define EGFR dependence by sensitivity to the EGFR inhibitor lapatinib versus insensitivity. And you can see there's a pretty good correlation between lapatinib sensitivity and SHP2 sensitivity, where those cells that are lapatinib sensitive are SHP2 sensitive, and the rest that are RAS-RAF altered have no effect of the inhibitor. We've also gone on to confirm that this molecule is active. It has good PK. It's active in vivo setting. This is a head and neck cancer, which has EGFR amplification and is sensitive to erlotinib. SHP099 and erlotinib have a comparable level of activity in this head and neck cancer tumor. And then this is a FLT3 primary AML orthograph, which you can see is extremely sensitive as a single agent to the SHP2 inhibitor. I'm going to skip this in the interest of going to lunch. And just summarize this part. The SHP2 is an oncogenic phosphatase. It's required for oncogenic signaling, mediated through RAS pathway activation. SHP099 is a novel allosteric inhibitor of SHP2 that induces the SH2-mediated inactive conformation of wild-type SHP2. This will not likely work against the JMML mutations that I mentioned before. In fact, that's a likely resistance mechanism. We've seen activity of this inhibitor in multiple RTK-driven cancers, including those of both solid tumor and hematologic origins. I'd just like to acknowledge a gigantic team of fantastic scientists at Novartis who I had the pleasure of working with for the last 10 years and whose interactions I miss dearly, though I have loved retirement so far. So far, yeah, winter might not be such a great time to be retired. Including Dino Mavrakis, Rob McDonald, Mike Schlabach, Eric Billy, and Greg Hoffman, who led the whole Project Drive and the work around PRMT5, supervised by Nick Keene, Tobias Schmeltz, Francesca Hoffman, and Frank Stegmeyer. I mentioned Dave Reddy for all his work in the shRNA Drive project. And then on SHP2, Yingnan Chen, who led the biology, along with Ho-Man Chan, Matthew Lamarche, who was the lead chemist for the SHP program, Tim Ramsey, who oversaw our chemistry group in Cambridge, Nick Keene, Travis Stans, and really Pascal Fortin, who also recently left Novartis, who really was the, let's say, father of pushing for the assay that led to the discovery of the allosteric mechanism. So thanks, and I don't know if there's time for questions, but I'm happy to take them.
Let's go ahead and take a couple of questions for Bill.
Hi, very impressive talk. I have two questions regarding the PRMT5 project. So is the FDA treatment toxic to normal cells?
No, yeah, it's not. I mean, you would probably because you can't achieve enough of PRMT5 inhibition. And the other reason you know it can't be toxic is we have cancer cells that have high levels of MTA, right? So if it had negative selective properties, the cancer cells would never have evolved the co-deletion of MTAP. They would have avoided, what would happen is you'd get selective deletion of P16 and you'd avoid the MTAP locus.
The second question is regarding the inhibitor. So I remember Epstein also developed a PRMT5 selective inhibitor.
Yep. Mm-hmm.
You comment on this too?
Yeah, I didn't go over this because it often ends into a longer discussion. But so if you remember what I showed you, MTA is binding in the same site as SAM. So in the cells, you actually have SAM and MTA simultaneously. It turns out even though MTA is high, SAM is even higher. The Epizyme compound is a SAM cooperative inhibitor. It requires and binds cooperatively with SAM to PRMT5, which means once that happens, the relevance of MTA levels in the cell become essentially irrelevant. So the PRMT5 inhibitor acts more like a CRISPR reagent and in fact is a sort of null for PRMT5 and doesn't distinguish between MTAP deleted and MTAP wild type cells. So you'd say, oh, well, how are you going to make an inhibitor? Well, we believe you need an inhibitor that's not SAM cooperative. So the enzymology is more complex, but we want a SAM agnostic inhibitor, or you could imagine an MTA cooperative inhibitor would ideally take advantage of the high levels of MTA in the cell. And believe me, the reviewers had a hard time understanding this, I think because they're not enzymologists, but yeah, that's the enzymology. And in the paper, there's all the data around the Epizyme inhibitor. It's all there. Yeah.
Okay, one last question back here.
Bill, can you comment on whether CRISPR has completely supplanted this pooled shRNA screening, or the pros and cons thereof?
Yeah, so the answer is yes for us. We had some debate about whether we should finish the next 7,500 genes by shRNA, and that's when all the CRISPR work was coming out. We've published recently our comparator in the first 5 cell lines between large-scale CRISPR and large-scale shRNAs, and we did a lot of work to eliminate false positives, but we had no idea what the false negative rate of shRNA screening, and it's substantial. And so with CRISPR, you pick up a significant number of hits that you don't find by shRNAs in just the first 5 cell lines. CRISPR itself has one notable off-target effect, which is copy number gain creates additional CRISPR cuts, which lead to toxicity that is gene independent. So when you scan across an amplicon, it looks like every gene and every region of the amplicon is cancer essential. This is also true if you have a CRISPR that happens to, by chance, cut 8 times in the genome. It will be 8 times more toxic than something that cuts once. So that's the downside of CRISPR, but the upside is significant gain in lowering the false negative discovery rate. And the false positive discovery rate is also pretty low, too.
Okay, thanks. Thank Bill.
Happy now to introduce Dr. Gail Sonnenschein, who is professor in the Department of Developmental Molecular and Chemical Biology at Sackler and Tufts University. And we're very happy that she's going to speak on, as you can see, the topic, ADAM8, a novel target for treatment of triple-negative breast cancer. And also should mention she's co-leader with Dr. Urban of the Breast Cancer Working Group, which I think has been a fantastic translational working group model that's been meeting for how long?
A couple of years.
A couple of years now. All right, great. Thanks, Gail.
Thanks, Ed. Well, I want to thank the organizers for giving me this opportunity to speak about our work. We're currently trying to target a novel protein that we saw is expressed in triple-negative breast cancer called ADAM8. And so I'd like to tell you about a little bit about how we discovered and validated ADAM8, and then what our new efforts are in developing a treatment towards ADAM8 positive triple-negative breast cancers. And in the interest of full disclosure, one thing we found as we were going through this process is that it's not easy to get money for development of an antibody or an antibody-based therapy from the point of view of making the antibody. And to that end, we ended up setting up a company which is able to get small business grant, which is able to get money for development. But the goal of this company and our goal in our lab is to bring ADAM8-based therapy to the clinic. So I don't need to go through all of this. I just want to point out that about 500,000 women, mainly women, die of breast cancer worldwide each year, mainly from metastasis. And while there are receptors on some breast cancers, ER and HER2, which are used for treatment, triple-negative breast cancers lack these targeted therapy options. And so they're often treated with chemo and radiation. That fails to stop the metastasis ultimately, and also have severe side effects. And although TNBC is about 15% of incidence, it's 25% of all deaths. So novel targets, new treatment therapies for both TNBC and metastatic disease are urgently needed. And our work has validated ADAM8 as a novel target for treatment of triple-negative breast cancer. So this introduces ADAM8 to you. ADAM stands for a disintegrated metalloproteinase domain protein. It's membrane anchored, exists on the cell surface. It is non-essential under physiological conditions. And so a knockout mouse has no phenotype either with respect to development of the mouse or its lifespan. And we came upon ADAM8. We had some discussion at the lunch about figuring out how higher throughput techniques. And we came upon ADAM8 actually from a walk down, several walk downs from an NF-kappa B rel-B pathway, which we had shown was promoting the aggressive phenotype, particularly of ER-negative breast cancers. So we did several walk downs and come up with a number of genes and ended up focusing on ADAM8 because of these properties that made it more ideal for targeting. So we're gonna talk about two domains on ADAM8. One is a metalloproteinase domain. The other one is a disintegrin domain. And these sit outside the cell, and I'll show you data that these are therefore available to an antibody-based targeting scheme. And ADAM8 itself is made as a precursor molecule, as are most metalloproteinases. So it has a prodomain that inactivates the ADAM8. And once it dimerizes, it actually autocatalytically clips off that prodomain, and what you end up with is an active form. And it's a little separated in here, but they're actually dimerized. There's another processing step whereby the metalloproteinase domain can be clipped off, leaving a remnant form. And I'm only pointing this out because you'll see this in some of the Western blots, but it allows us to distinguish whether the role of the activity that we're seeing is due to the metalloproteinase of the DI domain. So it's functionally helpful to have that. So as I mentioned, we found this downstream of NF-kappa B rel B, and the work was initially started by Mathilde Romanoli, and then finished by Nora Minova in the lab, and I'll show you the paper, the data first from the paper, which validates it. So Atomate in normal breast levels in normal breast is extremely low, but if you look at invasive breast carcinomas, the levels go quite high, and this is true in 14 or 15 independent studies. But more importantly, if you look at the overall survival of patients, those patients with high Atomate have a poorer overall survival and actually disease-free survival than those with low Atomate. So it is not good prognosis to have high Atomate. So we have collaborators in France, Sophie Barrier-Nyon and Delphine Luzang, who did immunohistochemistry for us. And they looked at either the normal breast or triple-negative breast cancers. And in the normal breast, they asked 50 samples, all 50 were negative for Atomate, which is consistent with the RNA data. And when they looked at triple-negative breast cancers, they found that 34% were Atomate high. And by high, I mean anywhere between 25 and 100% staining. So not Atomate low, we don't count those. 34% are Atomate high. But more strikingly, when they looked at metastasis, and not just metastasis from triple negative, it's all metastasis. When they looked at the metastasis, half of them were high for Atomate. And you can see this panel from the lymph node, but also from the brain. So our lab asked, what is Atomate doing? And in the cell, we started with cell models. So we used a very traditional strategy of knockdown. So we can reduce Atomate using siRNAs. And when we do that, in the cell line we started with was MDA-MB231, which, as you can see, expresses quite a bit of Atomate, and it's a triple-negative line. So this cell line, when you knock it down, loses the ability for anchorage-independent growth. It loses the ability to migrate. It loses the ability to invade and do invasive outgrowth through matrogen. Interestingly, growth on 2D plastic was not affected. And this was true in not only in the MBA231 line, but in HS578T, SUM149. So it was, and also if we did a stable knockdown in the 231 cells of Atomate. So we asked whether, what are the effects of this knockdown in tumor formation driven by TNBC cells in an orthotopic model. And I think you'll see the results were not what we actually expected, but are even more striking than what we expected. So these are cells that have Atomate. They have SH control, so they've been transfected with an SH control versus the SH Atomate. And what you can see is they're growing for a while. They're very slow. It's palpable, small tumor. And then beginning on day 12, you can see this break. And all of a sudden, the tumors grow much more rapidly. And we had to stop the experiment because the tumors reached the maximum size for our IACUC protocol. And if you can see, the tumors are red. They're highly vascularized. They're very large. And when we looked for metastasis, we found that the brains of 6 of 6 animals had a TNBC metastasis to the brain. So all the animals were getting mets. In the case of the Atomate knockdown, we were quite surprised to see that they weren't growing at all. Each day, Nora and Mathilde would come in and say, still not growing, still not growing. And then at the end of it, never grew. So what the tumors look like, and you can see it right here, they're small, they're not vascularized, they're just barely palpable. And when you look for mets, 5 of 6 mice had nothing, and this one mouse had what is a relatively small met, and that's it in the brain. So we started out to say, well, what does ADAMate do in the cell? What is it causing this tremendous difference in both angiogenesis and dissemination? So those of you who follow, I'm sure most of the people in the audience know that following the initial tumor growth, when the tumor reaches a palpable size, it undergoes hypoxic stress. And as a result of this hypoxic stress, what you can see is that ADAMate can get induced. And this is done in cells in culture, but if you look at the mice and the tumors in the mice, this is staining for ADAMate. This is the control mouse where you see the angiogenesis. You can see ADAMate, and in the ADAMate knockdown, it really was very effective, and no ADAMate was induced. So what that says to us is that in the absence of ADAMate, tumors go into tumor dormancy. And that, or at least it looks like they're going into tumor dormancy, such that they can't grow at all. So what does ADAMate do that is causing this ability to grow? So the first thing we looked at was angiogenesis. And we did a classical assay for angiogenesis. That is tube formation. So you can take human and umbilical vein endothelial cells, and if they're grown in the presence of angiogenic factors, they can form tubes. And we took the supernatant from the control or the knockdown cells, and you can see the level of ability to make tubes, both looking at branch points and polygons, is much higher if you have the control versus the ADAMate knockdown. And we went on then to look at what was being released by the ADAMate treatment of these cells, or I should say the presence of ADAMate on these cells. And it turned out we can show that VEGF-A, angiogenin, PDGF-AA, endothelin, and placental growth factor were all being secreted, and we can map that to the MP domain using some transfections of remnant versus full. So then what does ADAM do, or how does ADAM promote the metastasis that we saw? So we did a number of assays that are specific for metastasis. And the first thing that has to happen for a tumor cell to metastasize is that the tumor cell has to adhere to the endothelial cell, and then it has to go through the endothelial cell layer into the bloodstream. So that's the first step. And we can mimic that in assays in culture. The first one is asking, can the cells adhere? And the next one asks, can they get through the endothelial cell layer? ADAMate positive, ADAMate negative. Doesn't adhere very well. And migration through the endothelial cell layer is grossly and gigantically impressed in the absence of ADAMate. And we could actually figure out why this was true, because to bind, one needs to have activation of a protein called beta-1 integrin, and that happens through ADAMate. And we can show on the ADAMate negative cells, there was no activation of beta-1 integrin. So we can map that to that phenomenon. And then in collaboration with Irene Georgiakoudi's lab, Mike Palmer, who was a student in her lab, Mike and our group collaborated to see whether tumor cells could actually get into the circulation from the tumors. And what he can show from his analysis was that there was a big reduction of circulating tumor cells if you didn't have ADAMate. And that occurred very early, actually at day 7. And if you remember, day 7 is actually before we really see that break in the curve. It is early event that the tumors cannot get through the blood into the bloodstream if you don't have ADAMate. More recently, we did a study with Ben Dake and Charlotte's lab, where Ben taught us how to do cardiac injections. So we went and took cells that were either the control or the knockdown, and we introduced a luciferase vector. And we can show that if you take ADAMate knockdown cells, you're seeing a profound difference, not just in the ability of them to metastasize and get out of the site, but actually their ability to colonize as well is also dependent upon ADAMate. So from all of those studies, we came out with this plan that ADAM8 plays dual roles, both in angiogenesis as well as adhesion and intravasation and extravasation that lead to metastasis. So our next goal was to figure out if we could target ADAM8. And to do that, we considered two strategies. And the first strategy was, well, first we knew from other work and the work I presented that we had to inhibit both the MP and the DI domains. Inhibiting one was not going to be sufficient. A lot of groups have tried to inhibit the MP domain using a small molecule inhibitor that can go into the pocket of the MP domain. And they could get it to go in, but the problem is specificity. That if they have something that goes in, so many of the MP domains are so similar, they lose specificity. So they have failed, including trying ADAM8. So we decided that we were going to try a different approach using antibodies. And so we knew that a monoclonal antibody, if it could recognize this region, could potentially inhibit both. And we were very lucky because, and this part of the reason why we picked the strategy, was there was a commercially available antibody that was made, and it was made to the outside domain of ADAM8, and it bound to the MP and the DI domains, so we tested it and found that it actually could inhibit both the MP and the DI domains. So these are assays for MP and DI domain. This antibody, which I'll refer to as MAB1031, could inhibit both. So we did a mouse experiment where we took now the control cells, only the control cells, and we put them into the mice, and we started treatment with either an isotype matched control antibody. So this is just IgG2b, the antibody is an IgG2b, or a dose of this antibody. And we could see that we can inhibit both primary tumor growth, and we also got a reduction in brain mets. So we wanted to look more thoroughly at metastasis, so we designed a slightly different experiment, which is called neoadjuvant. And that means that you're going to give your chemo or your treatment before you do surgery. And this is currently being used frequently, so we introduced the tumor cells. We waited till the tumors were palpable, and then we said, okay, let's add some antibody, and then we remove the tumor, and that way we can now allow the antibody to tell us whether it can inhibit metastasis better than if we just kept the primary in there because we were going to reach our endpoint too quickly. And the results were dramatic. So here's brain and lung. This is the control, and now we've let it go out for 5 full weeks. And you can see 8 of 8 mice have brain mets, and now we see a lot more of them. 7 of 8 mice had lung mets, but in the anti-ADAM8 treated, only 7 mice had nothing we could detect. 2 had something like this compared to that. 8 of 9 mice had nothing in the lung and had only 1 that had anything. So what it says is that the growth and dissemination of tumors derived from ADAM8 positive cells was clearly susceptible to antibody targeting. So this strongly supported the notion that we should further evaluate taking ADAM8 therapy into the clinic, and it said that MP and DI domains were critical. This paper was published. Matilda and Nora were the first and second authors, and we had a lot of collaborators that helped us get through this process. So part 2, can we take this into the clinic, is basically the question. And MAB1031 is not translatable for various reasons. So the first part of this process was the submission of a patent. So for those of you who haven't done, you need to protect your IP or you will not get any ability to move this into the clinic. So the school submitted a patent with Nora, Matilda, and I, and its claims are for prognosis, diagnosis, and treatment of ADAM8 expressing cancers. And I can tell you we're talking about triple negative now, but it's actually on pancreatic, lung, renal, hepatocellular carcinoma. So if we make an antibody, it could have a huge impact in the clinic. And so we claim that our novelty was that we need to functionally inhibit both. So we proceeded to isolate our own anti-ADAM8 antibodies. And I'm not going to go through all the details of this, but just to tell you, the summary slide is we have made 17 mouse monoclonals that selectively inhibit ADAMate, i.e. we have removed activities. Any antibody that has activity against anything similar to ADAMate has been deleted from our pool. And they're specific, and when we look at the activity compared to the MAB 1031, we're seeing, for the most part, in one or both, equal or better activity. This is percent inhibition. So we took 4 of these, and we tested them. And originally, we were going to do a smaller test in vivo, but we were advised by pharma that testing the antibodies in vivo in mice was critical. So we bit the bullet, so to speak, and we designed now a different type of experiment. It's called single-dose in vivo efficacy screening. And in our case now, we're looking at pre-existing tumors, no longer co-treatment because that's the gold standard. So we can show that IgG, ADP2 and ADP13 can cause about a 50% drop at this one dose we picked of 10 mg per kg inhibition of the growth of the primary tumors. ADP3 and 19 were somewhat less effective, so we dropped those, and we're currently doing dose response curves on these with both the MDA231 as well as the another cell line, SUM149. We tested this in a neoadjuvant model, in which we have now taken luciferase tag 231 cells. We had the tumors. They were treated for a couple of times, surgically removed the tumors, and then we went out an additional 6 weeks and looked to see from metastasis using IVUS machine and biophotonic with fluorescence, basically. And these are the data for the GI system, and you can see that these are control mice versus our antibody treated. And these 3, if you sort of focus in, 16 and 17 look like this in the scan. This one is our highest with the treated versus 19, and you can barely see anything. So what it says is that these antibodies are able to block METs to the GI system. We have similar data for liver, pancreas, reproductive system, and bones. In all those organs, the antibody reduced metastasis. So the biggest question you can ask on an antibody-based therapy, and actually the only important question to ask in some ways, is does it increase survival? This is the FDA standard for treatment. And so we did a survival experiment, and we were quite nervous initially. When you start seeing the mice going down. So this is a survival neoadjuvant experiment that was performed, as I told you before, except mice were treated first for 3 times with 10 mg per kg. Then they got either IgG1, this is an IgG1 antibody, or ADP13 for 12 weeks after they were surgically resected. So recurrence, our assay for recurrence is having a palpable tumor. So if you palpate it on day, say, 18 and you see something, you then wait and make sure it's starting to grow and that you really didn't mix up. But you go back and that palpable tumor is listed on day 18, the first day you see it. And then for survival, the endpoint was 0.9 centimeters cubed. At that point, we're very close to our endpoint permissible by the IACUC protocol. And if you look at this, it's actually, as I was preparing this talk, I realized how close these were to the data for the patient data that I showed you earlier. As far as high ADAMate and low ADAMate, it's about a 50% reduction, and that's what we're seeing here. So we're quite excited because this would indicate that ADAMate-based therapy would enhance survival for patients, or at least it's consistent with that. I mean, there are a lot of reasons why it might not be, but it's certainly consistent. We're now in the process of doing the same experiment with ADP2. So how do we get this into the clinic? So there's one really interesting discovery that was made by Sonia Das in the lab, and that is that Atomate actually regulates the expression of a number of microRNAs. And these microRNAs, 60 of them, when we started looking at what their roles were, many of them were involved in resistance to chemotherapy. So that was quite intriguing and led us to ask, so here's some of them, but actually there are data on more of them now. So it led us to ask, can Atomate be playing a role in the in protecting cancer cells from being killed by chemotherapy? So if you just look at this middle panel, adding antibody alone, so this data is with paclitaxel and ADP13. And if you take the cells and treat them with antibody alone, you get a little bit of decrease, but not much. And then if you look at a very low dose of paclitaxel, you're not getting killing. But if you add ADP13, you enhance that killing. And if you do it with a slightly higher dose of paclitaxel, where you start seeing some killing, the addition of the antibody really dramatically, 50% improvement in the killing is pretty good. So now we have our way of getting this, we feel have a way of getting this into the clinic. And that is that we can take patients who fail their first line chemotherapy, which is usually with doxorubicin and cyclophosphamide. And we developed this strategy with a lot of input from Jack Urban, who informed us that we weren't going to be seeing naive patients because standard of care is so strict. So we would be looking at patients that have recurrent TNBC, and we can select for those that have Atomate positive cancers. And then we can ask if we can ask a patient whether she would prefer to have chemo alone or chemo plus our antibody. And then we're not depriving a patient of quality of care. So the patient could take paclitaxel alone if she preferred, and we would predict that after a while there would be recurrent metastasis over a current period of time. Or she could take paclitaxel plus our antibody therapy. And given the fact that we find that we'll get enhanced killing of the primary tumor even alone from the antibody, plus delayed recurrence, plus inhibition of metastasis, we would predict and hope that we will see increased patient survival. So that is our strategy, and we're testing it first, obviously, in mouse model. So this is the group that has done the work in the lab. Nora, Stefania, Sonia are actively looking at the mouse, doing one preclinical mouse model after the other. I hardly see them, so it's nice to see a picture of them at least. Sonia is also working with Ninon on more of the microRNA story. Matilda started the work. We've had a lot of collaborators. Jack has been wonderful, and everybody, many people know Ann Marsak-Rothstein, who has helped us with design of some of the antibody work, and we thank our supporters, and thank you. And if there are any questions. Yeah.
So I'm just wondering, since you saw
I'm sorry? Yeah.
I'm just wondering, since you saw an effect on the tumor side,
Yes.
if that alone would reduce the tax therapy. So I was just wondering if you ever waited until the tumors were the same size as control to look at the metastasis.
So we haven't done it quite that way but we have looked in the mice, and it turns out that when we start seeing at least circulating tumor cells, it's way before you see any tumors actually growing. So in that case, we see actually a very substantial drop in circulating tumor cells at that point. And in the experiment where we injected the cardiac injection experiment, there are equal numbers of cells going into the heart. So those those circulating cells had an equal opportunity to colonize, and without ADAM8, we saw much less colonization. So we haven't done exactly what you wanted, but I think it probably suggests there would be a difference. I think that the question is whether, how late can we hit this and still see inhibition of metastasis? Yes, right. Well, we have removed the primary tumor, actually, in that case. So, yeah, that was with, in that experiment, the primary tumor was also removed. Otherwise, it would have been. Yeah, so we start the antibody treatment before we do the surgery. So we give them 3 hits with the antibody first, kind of trying to mimic what a patient, if she were gonna get a pre-treatment, would be doing. And then we do the surgery, and then we continue the treatment. And we've taken it, that was 10 weeks, but actually it's gone out a little further. We just had to sack those mice. But the ADP2 experiment is actually being run now. Yeah.
One more question?
The other is, while I agree with you that survival is ultimately the endpoint, I think, piggybacking on what he was saying, if you're tracking your mice based on reaching a certain size, you're not doing a survival experiment. Now, if you disseminate model of metastasis, you can actually do a survival experiment, because usually those mice will die, so you can measure increased survival in those. But if you're doing a size-based experiment, it's not a survival experiment. And I strongly consider that you might want to have either before or in parallel with the study that you want to do, which is very difficult, having developed drug in this area. It's going to be very difficult for you to show the activity of the antibody at that moment. Maybe it's more of the type of a window study where you actually are ultimately testing in humans your underlying hypothesis, which is this agent will decrease metastasis. So I am very concerned you're not going to get any of your survival endpoints. You're not going to get any tumor shrinkage based on the preclinical factors.
Well, we'll have to see, won't we? Basically.
On that positive note, she's co-director of the Precision Medicine Program, as I had alluded to, and also Chief of Academic and Research Affairs in the Division of Pediatric Hematology Oncology. So she is a pediatric oncologist, but I think fair to say precision medicine is agnostic of age. In fact, she will be starting a precision medicine clinic in our adult cancer center in a few months. So we've asked her to speak about future paradigms for precision oncology.
A lot of what I'm going to present is from a publication of the same name that came out in May. But what I would like to do is give you a clinician's view. Much of what was discussed during the lunch discussion was you present clinicians with all this genomic and proteomic information, and most clinicians, unlike the scientific audience right now here, haven't been trained. Disclosure, much of the research for this presentation has been done with CSTS Healthcare, which has funded the scientists that have done the mathematical analysis. I have to say that in clinics, and that was acknowledged during that lunch panel discussion, we have had great successes with traditional chemotherapy. And I hope you notice that, I don't know if this points, the survival rates are in the 90s for acute lymphoblastic leukemia in children, and Wilms tumors in the 80s, Rhabdo, not so great, but still better than it was in the past. Now, I put 2003 as one of the turnover events, which is when the 40 years of molecular research has actually trickled into the clinic with the approval of the first biologic agent, which was Avastin. The survival until then was actually quite low with most agents, and most of the improvements were truly supportive care. We still aren't doing great in some of the solid tumors. So while one can assume a linear relationship between dose of chemotherapy and cancer kill in leukemias, because of the stromal elements that help cancer cells survive in solid tumors, we have not had as much success with chemotherapy at maximum tolerated doses in tumors that are solid. And I have to say, and this is purposeful, 80% of our patients with PNET or Ewing sarcoma die on standard therapies. And we have to begin thinking that some of those 20% survival rates may not be due to our chemotherapeutic intervention. And those may be the the people or the patients that may benefit from targeted therapies. So I have to say that in 2003, with the introduction of first biological agents, have changed, and that more and more therapeutic agents are now entering clinical practice, and we do have to get used to how to use them. It is very difficult to keep up, though, with all the emerging information. I think it's much easier as a scientist. For many years, I have run a lab, and I realized that there's a way more reading happening in a laboratory and in journal clubs than there is in clinics. The mechanisms of these agents are entirely new, and the science that has created those molecular agents, it's entirely unfamiliar to oncologists. So to be fair, we have also changed our cancer model. We no longer think of cancer as a single mutated clone. We now think of it as a very heterogeneous population of different cancer cell types, and that there is an induction of stroma that's actually very protective of that particular clone that's emerging and involves immune evasion, activating many of the PD pathways, inflammation, and angiogenesis. And you've seen a great example from Gail about how to engage some of these activations. We also have to be aware that many developmental pathways can be reactivated by activation of the stroma. So if one thinks about these heterogeneous population, and someone during the lunch discussion actually said, well, it's a moving target and we keep chasing our tails because each time we apply an agent that's targeted, the new population emerges. And I agree. We can also select very drug-resistant population by using MTD because we kill those cancer cells that are sensitive to that population and emerge new ones. This is from a publication in Science Translational Medicine, and it actually argues that if you drop the paradigm of maximum tolerated chemotherapy, you may actually preserve the ecological system within that cancer that has both therapy-resistant and therapy-sensitive cells, and you can engage some of those inflammatory and immune defense cells. When you do maximum tolerated chemotherapy, you definitely get better tumor shrinkage. But that tumor shrinkage may be due to killing everything that can be killed and retaining everything that is going to emerge. So metronomic or adaptive chemotherapy, as Dr. Gadenby's paper had suggested, it may be a better approach. This is a case in point. I'm a clinician. I always take it back to the clinic. This is a child who presented with blast count. She has ALL, acute lymphoblastic leukemia, with MLL mutation. This is the one time that we do not do well with leukemia. We were able to reduce, and if one can go to 0 and there is no residual disease, our maintenance therapy will prevent recurrence. And that's where our 90% comes from. If we can eradicate the tumor up front, we're good. We've cured, we follow the child, and she does well. We don't do well when there is minimal residual disease that actually recurs. We're able to bring them up with much higher doses of chemotherapy, but then happens that once they relapse, they've relapsed with the most resistant of clones. And you get multiple rounds until the child dies of disease. This is the child that somewhere here may have benefited from genomic analysis, and lestaurinib, which is actually directed towards MLL mutation, was never used in this child. So I hope I suggested that there are some patients that would benefit in this particular case. I am a believer, I do admit, and I believe that it holds a particular promise in oncology because there is way more agent than in other areas of medicine. It is applicable to many other areas of medicine. And in fact, our rheumatologists and our gastroenterologists do a much better job of using these agents. So I also believe that there is an individual specific tumor genomic signature that is actually a conglomerate of those genomic pressures of the host and of the tumor. And that a biomarker-guided treatment is an approach that may not only improve outcomes, but contrary to what's believed, also improve the cost effectiveness of our therapy so that we don't apply futile therapies in these multiple rounds of treatments. I also believe that targeted therapy may be less toxic. So what do we have in 2016? We actually have a lot. So if this is DNA, RNA, protein, and the cell, we actually have a lot of ways of testing our molecular targets. We have genomics, we have traditional in situ hybridization, we have dot plots, we have PCR, we have gene panels, and we have whole genome sequencing. One of the points I was trying to make at the lunch discussion is that all those DNA panels and what I call a candidate-specific approaches will answer questions you already know. So if a basic science supports EGFR in breast cancer, you already know that. What you are looking for are molecular signatures you haven't anticipated. So you can also go for RNA transcriptome. And this is particularly important for those mutations that are developmental pathways that have been activated at RNA level. Most of angiogenesis is not due to DNA mutation. It is due to amplification of pre-existing developmental pathway. Even though angiogenesis is silent postnatally, it actually gets reactivated. Similarly, protein-based molecular targets, we have immunohistochemistry, but our labs in clinics don't have the antibodies. So we don't do them. We also can do Westerns, but there's no clinical lab that will do it in a CLIA-certified lab. And we have CELDI and MALDI, and there is no place to send your patient samples. So proteomics has not entered the clinics. And then there is metabolomics. There are phosphorylation patterns, also possible to do in any research lab. You can do a phosphorylated protein very easy, much easier than a clinician can. You can do methylation patterns. You can do lipid patterns and glycosylation patterns. We cannot, clinicians cannot. So we also have to realize that when we as clinicians send out a genomic analysis, it looks for these, it looks for these syndromes, for Leigh-Fraumeni syndrome, for Lynch syndrome. It doesn't, at best, it uses the candidate gene approach. But very few times you actually get an open-ended approach. And this is what Bill Sellers was talking about when you actually try to give bioinformatics information to a clinician. So the whole genome sequencing is a research tool. And it identified the most frequently mutated genes in a population, not in a patient. And it is fed, and we created all those different protein-protein interaction maps. For most researchers, these protein-protein interaction maps are daily event. For clinicians, we hardly see them. But this is what we actually have. We can use the specific patient-oriented clinical information, proteomic information, cytochrome information, metabolome information, and we can feed it into our tools, overlay it over the exact existing databases. We have GEO, we have TCGA, those are atlases against which we can analyze our specific individual patients. And we can look at protein-protein interaction network. There's many of them. They're all academically curated. And if they're cross-referenced between each other, you can very easily identify the energetic pathway of these different situations and of these different profiles, genomic profiles, and bring it back to the patient and inform the therapy. I'm going to try and make a small attempt as to whether or not we can do this and whether or not we can include it in the patient data. Because the analysis that one does in clinics usually is a handwritten note on my desk or on Andy's desk or on Jack Urban's desk. We don't actually put it into the EMR, into the electronic medical record. So I'm going to try to say that all these different requirements that precision medicine may need exist. So as I said, we need an EMR that streamlines all the different informational streams and that it becomes part of the medical record. We also have to have the interconnectivity of those data sets that Moonshot 2020 is trying to do. We have to cross-reference our data integrity. These are patient lives. And we have to develop a hierarchy of these genomic alteration in individual tumor sample, not for the population, for that particular individual. And we need to, because many of those biologic agents are chemotherapy sensitizer, lower our doses of chemotherapy. So this is an example of integrating a patient's RNA information. So we have published back a year and a half ago with the team of mathematicians a manuscript that indicated that if you use the patient's RNA information, this is done on TCGA patients that actually have data, and look for interactive molecule and calculate the complexity of the network as an energy pathway, then you can correlate the complexity of the network as calculated as Gibbs energy with the progressiveness of the cancer and the 5-year survival. We have done that for different cancers, and this is again published for more details. And then we looked at 4 patients of low-grade glioma and looked whether similar histology, this goes back to I think Clive's point at the lunch session of developing panels and my argument of looking for unanticipated mutations. So these two patients within the TCGA both have the histological diagnosis of oligodendroglioma. But calculating the complexity of the network had identified entirely different targets for them, one being an HDAC as the primary target and one having HSP8 as a target. This also activates HSP, but in a different energetic level. So this is an example. This is an example of acute myeloid leukemia, again from the TCGA, from the Cancer Genome Atlas. And it identifies these subnetworks. We call them energetic subnetworks within the large furball of protein-protein interaction networks. And you can see that there are 3 proteins that hold this pathway that are connected with a FLT3, a very known target in AML. And you can see that by inhibiting this, you not only thin this complexity, you separate the activating pathway. Whoever wants to talk to me about more about it, just in time frame, I'm happy to talk about how this is done. This has now been automated and computerized using computer tools. So this is another case in point from clinics of how the patient's profiles evolve. This is a child who's 11, diagnosed with glioblastoma in 2011 with a large parietal lesion, and she presented with left-sided weakness. She suffered hemorrhagic stroke, her hemiparesis never resolved, and she went on 2 sets of standard therapy. She went on the ACCN, which is a high-dose chemotherapy trial, in combination with vorinostat, which was the Arm A investigational agent. She was then followed with Avastin temozolomide, and I know there are not friends of temozolomide here, but she did quite well for about 10 months. So she progressed. So we went back this time and looked at her molecular profile. This is a Foundation Medicine panel that has about 600 different genes that have been considered by Todd Golub and Eric Lander, who are the founders of that organization, to be important, clinically important oncogenes. And you can see that this child not only has an F1, which we then found she has systemically, she also has BRCA1 and SOX2. So because SOX2 amplifies Wnt pathway, we had started her on combination of sirolimus and sorafenib with a low-dose etoposide. She did quite well for about 4 years. And then we found a newly enhancing lesion on an entirely different place, which didn't look like glioblastoma. It looked like different tumor. And I said, can we get a tissue? So we got tissue and sent it for reanalysis. And this was an important point. She still had the majority of her previous mutations, but she now had an entirely different set of a clonal proliferation. And look at the histology. So her histology changed. This is a classic H&E of glioblastoma with the sort of hemorrhagic regions, the blue round cell tumors with about 30% Ki67 and a very strong GFAP, which is a glioblastoma multiforme marker. Now, note that this is more of a neuroectodermal pattern, more PAT-like pattern of cell differentiation with a lot of more larger cells and a lot less hemorrhagic phenotype. The Ki67 is now in the 67%, and she lost her GFAP. This is no longer a glial tumor, and that is confirmed by the change of her phenotype. So this time around, we know that GLI is activated, So sonic hedgehog is therefore activated. So she was on ceritinib, trametinib, and sirolimus, and she's back to school and dance lessons for a little while. I don't know how she'll do ultimately. We are indeed chasing our tails. So going back to what we will need, and I agree that we will need a different clinical trial design, because if we want precision medicine and individualized therapy, we cannot have populational studies. But this is what the clinicians are faced today. So we get this furball of protein interaction networks, and we will also need what our air pilots need when they consolidate their information, what our nuclear stations need to coordinate their information. We need computers to analyze this for us. The only way we're going to do it is to automatically process the varied resources. We have a lot. Our scientists have been working for the last 60 years on these molecular networks. And these complex socio-technical systems that I'm suggesting will provide a guidance, a technology, individual and teams regulated mathematical or computerized algorithms that will allow you to do it, bring it to the tumor board, discuss it, and provide care. So this is our flow, and again, it's published. If we consolidate all the tests on a particular patient, submit it to computers, recommend pathway therapy, review in tumor board, provide therapy, collect the outcomes, which is something we do not do. And I hope that the infrastructure that Moonshot 2020 will provide will be possible to do, and then bring it back to inform the next decision. So I'm actually quite proud to be part of this initiative. That Floating Hospital for Children is one of the founding members, and I hope we do collect the 20,000 children molecular profiles, and that we will be able to do it. So this is my summary slide. I do believe this is a transformative time in oncology. I do believe that we will need a lot of we will meet a lot of challenges, and both in quality of our tests, in quality of our genomic information, in the quality of interpretation of that genomic information, and that we do need to collect our outcomes, not as a case one study, because unfortunately, all of the case studies out there are the positive case studies. They're not the ones that failed. They're all the good outcomes. And I'll leave it at that, and I'm happy to take questions.
There's something I missed in your talk. Can you explain how you classify cancers in thermodynamic terms?
So,
because I don't usually give free energy.
Right. So our premise is that cancers are not typically following the tissue of origin. They typically follow the biological events that have occurred in that particular population, and then ecodynamically suppress or enhance a particular population within the tumor. So if one assumes that a cell is a space in which everything has a certain amount of entropy, certain amount of energy, then if you suppress this, this will come up. So all you need to do within that protein-protein interaction network of that particular patient is to identify the highly energetic pathways. That is a method that the mathematician introduced me to that is actually preordained. You can calculate entropy, and you can calculate Gibbs energy. So that's the term the mathematicians use. We have retained it so that people know which formulation we have used. We have then been able to connect in real biological data out of TCGA that this Gibbs energy correlated with survival. So the the higher your Gibbs energy, the poorer survival you had. Yeah. And then we went on and connected, so there is another manuscript where we call it a Gibbs homology, which also, and that's coming out next week in OncoTarget. That incorporates not only the energetic, the physical interaction levels between the different proteins, that also connects the number of connections that the particular protein has. So there are some proteins that are hubs. Some of them were mentioned in the panels. So the RASes, the BRAFs, the ALK1s, they're all hubs. They're busy and they're very interconnected. And that information needs to be brought into the energy information as well, which amplifies your selection of the target. And you can calculate by inhibiting the different proteins, so all thousands of proteins are inhibited one by one, and it tells you inhibition of which protein actually provides you with decrease of that complexity. So that's not done manually.
Good question. In the interest of time, let's conclude this session. Thank you, Dr. Clement. So welcome back, everyone. My name is Andy Evans. I'm director of the Tufts Cancer Center. I'm a physician scientist. My expertise is lymphoma individually, but I have the pleasure of helping coordinate all the diverse and unique enterprises you see on the slide here. And I know many people are in the room representing each of these. And I think it represents an opportunity and challenges to coordinate all of these unique pieces. But I just want to congratulate Dr. Hines so far in just a fantastic inaugural symposium. Great job, Dr. Hines. Really, I mean, I've been involved in a lot of symposiums, and so far, so good. But I think it's also fair to say that TUSM is really the anchor to our cancer scientific research enterprises. But the term convergence is very apt, because how do we figure out optimal convergence on these unique enterprises? And as you heard also, I think the answer to the question I posed to the panel, that we can't be everything to everyone, and what are we really good at and what can we become great at? But I think there's other layers and levels of convergence. One is just amongst these research enterprises, but the other is how do we coordinate and really converge that science so it benefits the patient? And this is somewhat of a construct of all the moving parts to across the street, Tufts Medical Center, and where we have primary care and all these different specialties that need to really find ways, which we do, to work together with a lot of their community health and other payer partners, et cetera, orbiting around the patient. And then we have research feeding into that and vice versa. So and we have a lot of tools now, all the different therapeutics. And how do we navigate that? It's not easy, but I think we have the tools and the minds to be able to do it. And of course, it's going to be through translational research studies, whether it's bedside to bench, bench to bedside, and we have the talent here at Tufts to do it. And so I'm just really so happy to work here, just given the expertise and the passion that everyone has. So this session is precision medicine. I think like a lot of centers, we're trying to figure out, well, what does that mean to us? It's kind of like the word translational. It can mean different things. And we actually, about 6 months ago, formalized a precision medicine program or working group. One of the co-chairs is Dr. Clement, who will be speaking, and also Dr. Martel co-chairs it. But I won't read through this, but you can see at least at the outset how we defined it. And this group has been meeting twice a month, really kind of sitting around, not a campfire, but a table, and doing a couple different things. One with scientists and how can we better kind of connect the bridge between our research and patient programs, but also how we will also go through patient cases. And you heard about how you can do genomics on patients and what does it mean. And it's actually really difficult sometimes to understand what these outputs you get from a Foundation One or others. And so how can we help patients in semi-real time and also do cutting edge research at the same time? So at least that's our kind of big picture view, and we're again just really happy to be part of this. And what is the future? The future is truly precision medicine, personalized medicine, putting it all together, which is complex but doable. And can we get to a place where that pill has 5 different compounds in it, little nanoparticles, et cetera? I think, and it's not all about treatment, of course, prevention, diagnosis, et cetera. So I just wanted to give that quick overview of kind of what we're thinking on the big picture at the Tufts Cancer Center. But with that, we're going to go right into our first session, and I have the privilege to introduce Dr. Amy Yee. Dr. Yee is professor at Tufts University School of Medicine at DMCB and also in the Friedman School of Nutrition and Science and Policy. She's also president of AACR Boston Incorporated. And as many of you know, her goals of her laboratory are to understand the mechanisms of triple-negative breast cancer using biochemical and genetic tools, as well as concurrent informative preclinical models. And she really has a very exciting project and compound that she's going to tell us about. And let me pull that up. Title of Dr. Yee's talk is Wnt Signaling in Green Tea, A Tale of Breast and Brain.
Okay, thank you for the introduction, and thank you all for coming to this inaugural Tufts Cancer Symposium. It's been a great set of talks, so hopefully I'll continue in that vein. Okay, so what I'm gonna talk about is one of the projects in my lab, and we're focused on triple-negative breast cancer. So this is a breast cancer subtype for which there are few treatment options. And in part because it is negative, it's called triple negative because it's negative for many of the markers that mark breast cancer, ER negative, PR negative, HER2 normal. And it's part of the basal-like class of breast cancers, which constitute about 20% of all breast cancers. But 2 of the pathways that are well associated with triple negative breast cancer are the EGF receptor and the Wnt signaling pathway. They're not quite separate pathways since one does inform the other, and Wnt signaling has been shown to be responsible for resistance to EGFR inhibitors. In fact, this is kind of an odd cancer, and this is the only thing I'll mention about EGFR, is that there's overexpression of EGFR in triple-negative disease, but these cancers are completely refractory to the EGFR inhibitors. And the Wnt signaling pathway has been shown to be part of that resistance. Okay. So the focus of our work, we focus on the primary cancer, but we're really moving our focus towards metastases. Why? So 90% of breast cancer deaths are from metastases. And no matter how many years, I guess since Nixon's war on cancer, still 20% of breast cancers have metastases with little improvement in the many years. But the oncologists are pretty smart. They've gotten really good at maintenance of the metastases. So breast cancers go from bone, lung, liver, and ultimately to brain. So the non-cranial metastases are maintained pretty well by chemotherapy. The patients have a pretty good quality of life. But one of the consequences of that improved maintenance, an ironic consequence, is breast cancer patients, it's a good thing, are surviving longer. But what is happening now is that there are increased rates of eventually fatal brain metastases. Once breast cancer goes to the brain, the quality of life is exceptionally poor, with death often within months. So my thought and the thought of the field is if we can make some impact on brain metastases, we can extend the lives of breast cancer patients even longer. And my hope would be so that they die of something else, not breast cancer. But there's some challenges in that few compounds for chemotherapy cross the blood-brain barrier. And these patients with brain metastases, they have the tumors, but they have a poor quality of life because of other comorbidities such as seizures and cognitive deficits. And of course, it's a horrible death with the brain mets at the end.
Okay, so what is our approach to triple negative breast cancers and brain metastases? So over the years, what we've done is to refine a preclinical model of triple negative disease. You need a preclinical model. And I'm a biochemist by training, and the cell biologists accuse me of thinking of a cell as a box with a bunch of signaling pathways, but that's what I think of them as. So you need some pathways and genes, molecular framework and tools, and you need the brains to be able to choose from all the options that we have for investigation. So the outline of my talk is 3 small parts, and I'm going to talk about the preclinical model that we optimize to get brain metastases and seizures. I'm going to talk about a specific molecular network, I guess, with Wnt signaling and a gene we work on, HBP1, and the consequences to the metabolic regulation of Warburg effect. And lastly, I'm going to talk about a compound combination that seems to be very effective in the preclinical setting for treating brain metastases. Okay, so the approaches that we take to tumor genesis, you can use cells, you can use animals and specimens and bioinformatics. So we focus primarily on these 3. Okay, so we use a xenograft model in which we implant a triple negative breast cancer line, and we use a couple. This is the workhorse one. The cells themselves are tagged with GFP and luciferase, and one thing we do to extend the course of the experiment, but to optimize for metastasis, we do a surgical resection. This was suggested to us by Jack Urban sitting here as a way to make this model more like what happens to the patients in the clinic. We use the vast amount of tools available in the bioinformatic databases for analyzing gene expression. Okay. Oh, let's see if I can turn down the lights. Oops. That was the other way. Okay, so these are examples of brain metastases in the non-skin mice. So our cells are GFP tagged. So here's example of one. And in another example, sometimes the metastatic tumors get quite large. So here's some of the larger tumors. I think this one was almost the size of a hippocampus. And so we're able to get not only the primary tumor growth, but also the growth to the brain, whether it be the micromets or larger tumors. And what our breast cancer model undergoes, so here's an example of a mouse, is that like metastatic brain cancer patients, which are often diagnosed after a seizure or a massive headache, our mice also exhibit seizures. So right now, there's no current animal model for metastatic tumor-associated seizures. And so this gives us a venue ultimately to study the origin of brain metastases in an animal model. Okay. Oops. So, I was going to show you the seizing mouse, but I can't. So this is an example of a tumor-bearing mouse that underwent a seizure shortly before we sacrificed it. And this mouse's section was one of the brain mets that I showed you. Okay. So that's our model. We have a model that goes from primary tumor. The cells are implanted in the mammary gland and goes all the way to metastases to the brain. There are also metastases in the liver and in the lung. So the framework for much of our studies is the Wnt signaling pathway, which I'll go through pretty briefly. So the central part, it's a total signaling pathway going from receptor to activation of gene expression.
The core element here is the beta-catenin destruction complex. The key part is to keep in the off state, to keep the levels of beta-catenin low so it can't go into the nucleus and activate gene expression. So as such, this is called the beta-catenin destruction complex. Through a phosphorylation-dependent mechanism, triggers beta-catenin degradation. Now, when the Wnt signaling pathway is activated, say by binding of Wnt ligand to its receptor and co-receptor, is that the net consequence of that is to inhibit the beta-catenin destruction complex, leading to accumulation of beta-catenin. Beta-catenin moves into the nucleus and combines with left TCF transcription factors to activate target genes that are responsible for many parts of biology: stem cell maintenance, cell proliferation, and the Wnt gene expression programs also responsible for tumor initiation to tumor aggressiveness. So in my lab, we worked on an inhibitor of Wnt signaling. I have a little bit more to say about in a moment, called HBP1, which actually needs to go down a little bit more. It inhibits the transcriptional activity of beta-catenin left TCF. Okay. So the Wnt signaling pathway, so that are the details of the Wnt signaling. Oh, I forgot to mention one other detail, is that this pathway is under substantial negative control, whether it be by tumor suppressor genes APC and axin for colon, colorectal cancer, for liver, or negative control at the receptor level by SFRP proteins that sequester Wnt ligand away from its receptor, DKK proteins that prevent an essential receptor co-receptor interaction. Okay, so it's both positive control and negative control. But the Wnt signaling pathway itself is associated, as I said, with tumor stem cells and with metastatic disease proliferation, but it's also important for normal development. When aberrantly regulated is when the pathological situations come about. And in some studies, the Wnt signaling, high Wnt signaling, has been concretely linked to triple-negative breast cancer and to brain metastases. And in this paper, what I thought was remarkable is they used a Wnt signaling signature in a database of over 1,000 breast cancer cases, of which some were triple negative. And having a Wnt signaling cassette pulled out the triple negative disease with a very high percentage, a very good reliability. So substantiating that Wnt signaling is linked to the triple negative disease. One of the newer findings with the Wnt pathway is that it's one of the pathways that's linked to a Warburg-like program of metabolic rearrangement. So this probably goes along with the fact that Wnt signaling regulates proliferation, since the Warburg rearrangement is responsible for, puts a cell in a situation that favors proliferation. Okay, so in a brief slide, what is the Warburg effect? So the Warburg effect is a wholesale metabolic rearrangement. We know it most as glucose going to lactate in aerobic glycolysis. And there's a lot of work on the Warburg effect. I'm going to try to simplify it. So the differentiated tissues, they take glucose and metabolize it down to 36 ATP and water in an oxidative phosphorylation dependent fashion. So this doesn't leave any intermediates to synthesize the constituents of a cell for division, such as lipids, amino acids, etc. Whereas in a Warburg effect, a Warburg effect is often associated with proliferative cells. Glucose is metabolized to fewer ATP, and lactate is often a byproduct. But what this leaves is lots of intermediates that can be interchanged to make amino acids, and lipids and nucleic acids. And in the Warburg effect, there is usually a switch of isoforms of many of the pathways we had to learn in biochemistry. So it's really a complete metabolic rearrangement, usually for proliferative cells. Okay, so let me return to breast cancer. So one of the things that we showed in our lab was that this gene HBP1 is a transcriptional repressor, and it's an inhibitor of Wnt signaling. It's part of a certain class of transcription factors. And the biology of it itself, apart from Wnt signaling, is it's an inhibitor of G1 progression and effector of oncogene-mediated senescence. But what I'm going to focus on is this role as an inhibitor of Wnt signaling and the fact it's mutated or reduced in breast cancers with a poor prognosis. In particular, is that it is reduced in triple negative disease. So this is a clinical specimen screen versus triple negative breast cancers versus other types. So HPP1 is reduced. This is an in silico screen with essentially the same result. So HPP1 is low in triple negative breast cancers. So that is consistent with its possible function, its functional role as a Wnt inhibitor. When an inhibitor is low, you expect activation of that pathway. So we do see that, and what we did was we did an RNAi knockdown, shRNA knockdown of HPP, and we found a surprising result, but it's now fully consistent with the documented role of Wnt signaling in regulating a Warburg-like effect. Because what we found when we knocked down HPP1 was we found, this is actually, we have gone back to the microarrays, we found over 300 genes that were altered with a pattern of a Warburg-like effect. There were increased glycolytic genes, increased expression of the Warburg isoforms, decreased oxidative phosphorylation, increased pentose phosphate shunt, and a lactate in a HPP knockdown cell was also increased. So how we have been doing our metabolomics is with my department collaborator, Jim Balea, who is an NMR metabolomics expert. So we chose NMR to delineate the metabolic changes because we wanted to be in a position to translate our NMR results into new markers for breast cancer diagnosis. So how would that work? So right now we're doing all the biochemistry, but every breast cancer patient here gets an MRI. And what you can do in an MRI is change the sequence of the MRI so that you can focus on a voxel or a box of the tumor. And if you know the spectrum, the spectral area you want to look at, you can get chemical information in a non-invasive manner. So what we're trying to do is understand some of the NMR-based metabolomics as a prelude to designing new MRS markers. And our clinical collaborators are Jack Urban and Shetal Makim at the hospital. Okay, so to make a lot of work short, what we found was that there was, and my part of the lab does the gene expression, the protein expression, Jim's lab does the metabolomics, and what we found was that when HPP was knocked down, one of the patterns that was increased was an increase in lipids and lipid synthesis, and we pieced together an increase in citrate, and there were changes also at this level. So this is also consistent with the Warburg effect. There's an increase in FASN, fatty acid synthase, and an increase in one of the Warburg isoforms, HK2. So this was a lot of effort to map both the gene expression and the metabolomics. Okay, now, so now with all these changes with Wnt signaling, and we also agree with the literature in terms of the metabolic changes, can we do anything about this? So this is where we use our brains, and I have to say we were actually fairly lucky because we made some simple assumptions, but I guess we had the bravery to test them. Okay, so what we thought was that triple negative breast cancer, Wnt signaling has a large role. And so how can we block Wnt signaling? So what we would ultimately like to do from the pathway point of view is to increase the inhibitors. And we knew inhibitors like SFRP1 and DKK1 were under epigenetic suppression. So can we use a compound like decitabine that's a DNA methyltransferase inhibitor? The other thing we wanted was, maybe we're not as brave as others. We weren't interested in doing a compound screening library. What we wanted to do was to repurpose compounds that were already in clinical use. So decitabine is FDA approved for treating myelodysplastic syndrome and off-target for leukemias. And then another compound we chose was EGCG. This is a principal compound from green tea that we had shown independently for reasons uninteresting why we began this project, is that it, in high levels in cell culture, blocked Wnt signaling, and it did it by increasing HBP1 an inhibitor of Wnt signaling. So we're thinking of ways to induce inhibitors. So let me tell you a little bit more about the two compounds. So EGCG is epigallocatechin 3 gallate, and it's the principal compound in green tea. So some early epidemiology studies show that about 5 cups of green tea per day were associated in some Japanese studies with reduced breast cancer, and there are many phase 1 clinical trials with green tea. So these are the numbers on green tea, but I think the bottom line of what I'm going to tell you in a few slides is that we use attainable dosing for green tea. So if we proceed forward to a clinical trial, the patients will not be drinking 10 gallons of green tea. And we've been very careful about doing our preclinical studies this way. And here are some of the numbers that are associated with the maximum ingestion of green tea. So in our study, the concentration we use is this, and it's an equivalent of this amount, which is fully attainable in the maximum in terms of a human. And what we are currently doing is refining this more in collaboration with David Greenblatt to look at the plasma concentration in mice. So this, based on other people's studies, would be a plasma concentration of that amount, and that is fully attainable. Okay, so that's the green tea part. And green tea, of course, has very good safety. It's one of the, tea is the second most consumed beverage in the world after water. So we're pretty confident about this one. The other compound we use is decitabine, which is, we also call DAC, which is methyltransferase, a DNMT1 inhibitor. It is approved by the FDA for treatment of myelodysplastic syndrome and off-label for leukemias. It's an inhibitor of DNMT1. And the consequence to hematologic malignancies is a re-expression of epigenetically silenced genes. But there are still things we don't understand about epigenetic regulation, because not every gene is unsilenced with decitabine. So I'm not saying it's selective, but it's not a 100% demethylating agent. Okay. And the key point about decitabine is right now there's no known efficacy in solid tumors. So excuse me, here are the doses that we use. And just to make a long story short, the dose we use in the preclinical study is also in the FDA approved range. So we have 2 compounds in use in other diseases that we are seeking to combine. Okay, so this is an example of some of our data. We've done many mice, probably 50 or more at this point. And what we see is that EGCG, DAC together diminishes triple negative breast cancers. So this is actually by tumor weight. When we palpate the tumors, we see a larger decrease in the tumor size. But the exciting part to us is the following. It's because we have a model that recapitulates brain and other metastases. What we see is that its EGCG DAC treatment reduces the total number of brain metastases and specifically reduces the number of brain metastases. So this is one of and we learned this after we got this result, but both DAC and EGCG cross the blood-brain barrier, in contrast to many other compounds that are used for breast cancer. Actually, we learned it from some epilepsy experiments in our lab as well. So this compound combination differs from other options for breast cancer patients. Okay, so what's the mechanism of EGCG DAC? And we had to work through the data. So first, this is a tumor growth curve, and here's another example of untreated and treated tumors. And what this curve simply tells you is that only the combination works. Neither compound works on its own. And this is an attempt towards perhaps stratifying the patients. If we use an HPP knockdown tumor, one in which HPP1 cannot rise, the EGCG DAC does not have the same effect as a wild type. So HPP levels must rise. Okay. We showed that directly here using two different exons of the HVP1 gene, is that the HVP levels rise with EGCG-DAC treatment. And with EGCG-DAC treatment is that there is an inhibition of Wnt signaling. So here's a control tumor, and then here's one that's treated. There's a vast decrease in nuclear beta-catenin, whereas the individual combinations themselves have no effect. And furthermore, what we see coincident with the decrease in beta-catenin and the decrease in tumors and decrease in metastases is an elevation of Wnt pathway inhibitors, HPP1, DKK1, and SFRP1. So the treatment combination elevates Wnt signaling inhibitors. So whenever I would present this work, I was always asked, well, EGCG and DAC are not targeted compounds. So what else do you think it does? So in an attempt to answer that question, and we're still in the process of analyzing this, we did an RNA-seq analysis with treated and untreated tumors from our xenograft models. And for those of you that do the whole transcriptomic analysis, these are the kind of things that you get out of that. So we see changes, a decrease in cell movement, and we also see some changes in stem cell genes. So we went to look at the pathway, and it was a relief in that Wnt beta-catenin pathway was one of the top 15 pathways that was inhibited with EGCG-DAC treatment. We had done much of the biochemistry before, but it's gratifying that an unbiased analysis reinforced that. There are other things that we're looking at right now in the future, now and in the future, is that there are changes in the epithelial adherence junction. In fact, one of the work done by a very talented Tufts undergraduate, Molly, has shown is that E-cadherin is elevated in this triple-negative breast cancer cell, which normally would not have that. But so we're still investigating these things. But we wanted to hammer this Wnt story one more time. We use a custom Wnt array in part to avoid making qPCR reagents to every single Wnt target on that RNA-seq. We bought one array, and what we found with Wnt targeting is that 90% of the Wnt target genes were in fact inhibited by EGCG-DAC treatment. That's the same data. But the EGCG-DAC inhibited Wnt signaling, but it did one other thing that we have studied. And in my lab, we do a lot of metabolomics with Jim Balea using the NMR. And this is a summary of his metabolomic analysis. And these is a principal component analysis showing EGCG-treated tumors and control tumors. So they're two separate populations in response to the 50 metabolites that we looked at. So there is definitely a different population metabolically. And we found that EGCG-DAC treatment reduced FASN levels, a key enzyme for fatty acid biosynthesis, and HK2, a hexokinase associated with the Warburg effect, that both were decreased with EGCG. So EGCG-DAC appears to reverse aspects of the Warburg effect. And here is two NMR analyses for steady state metabolites done with proton NMR, and this one is a carbon, a C13 analysis, and this is a flux analysis in which we inject C13 glucose and take a snapshot of the metabolism of the tumor at that time. So what we found out of all this, to support the idea the Warburg effect is partially reversed, is that, for example, lactate is decreased with EGCG-DAC treatment as reflected by a decrease in lactate in the C13 analysis. And another one that is reduced is alanine, which is a readout for pyruvate. So there's a decrease in the alanine as well, and a readout for amino acid biosynthesis. So we're still continuing to look at this. And this has been something we've been looking at for some time, because these are two compounds, not quite some time. So these are two compounds that are also either in extensive use in clinical trial or FDA approved. So what we've been doing over the last month is really, last month or more, is trying to package the studies into a phase 1 trial. So my partners in this is Saria Jelapan, Jack Urban, Julian Wools. Saria is a neuro-oncologist. Jack is a breast oncologist. Julian Wools is a neurosurgeon, and David Greenblatt is a pharmacologist. So we are trying to push the basic science studies over to the translational spectrum and towards a phase 1 trial. So we've begun to configure a phase 1 trial, to which we will, I'm going to guess in 6 months to a year, administer patients with DAC and with EGCG. Okay, and these are my calculations on the tablets that we're going to use, and the rough equivalents into the preclinical and the human models. Okay, so to summarize, what I've told you briefly today is that HBP1 and Wnt signaling coordinate a Warburg-like metabolism, and that there's reduced HBP1 in triple-negative breast cancer relative to other subtypes, and that the HBP1 decreases, which lead to elevated Wnt signaling, have a widespread effect on metabolic genes with alterations in the lipid metabolism. And the mechanism of EGCG-DAC to inhibit Wnt signaling works through elevating Wnt inhibitors, and that the EGCG-DAC combination has much needed efficacy on brain metastases, also on primary triple-negative disease. And we think it works synergistically by elevating Wnt pathway inhibitors, but it also has an unexpected effect on attenuating a Warburg metabolism. So toward the phase 1 trial, our preclinical studies using doses that EGCG-DAC are using doses that are attainable in humans, we think that both compounds, this will have excellent safety, and this will have manageable side effects. And all the calculations and studies that have been done previously suggest that there'll be predicted favorable plasma concentrations. So we're excited about finally moving this compound, hopefully, into a clinical trial. So these are the people in my lab that have done the work. Jim Balaya's lab did the NMR analysis. Some of the studies at the early part were a collaboration with Gail Sonnenschein and Nora Minerva in her lab. Together with them, we tested, you know, I'm cognizant of Bill Sellers' talk with 100 cell lines. We did test one more triple-negative cell line. These are my clinician partners, hopefully to bring this to the patients. Okay, with that, I'll stop, and I'll take any questions. Thank you.
If Gail could come up and we'll load your slides while we take 1 or 2 quick questions. Anyone? I had one.
Oh.
So, obviously, the hope is it's 100% remission rate, but knowing that likely won't be true, are there any early hypotheses on whether based on the transcriptomics or other studies, biomarkers of activity or resistance? Are you going to go into the study? I know it's just a phase 1. Is there any kind of
intermediate
biomarkers at all?
Okay, so one of the things when you're, as I understand, I'm a PhD, I've never done a clinical trial. One of the things that if you, let's say you have no effect on tumors. So is it no effect because your dosing was incorrect, or is it really doesn't work? So we're gonna take a page from the Novartis playbook. So the Wnt signaling pathway has been difficult to drug, but there is one compound in clinical trials. And what they do is they take a skin biopsy and look at axin 2 expression. Axin 2 is a Wnt target gene. Skin happens to have high background of Wnt signaling. So if you have Wnt inhibition, then you should get a decrease in axin 2 expression. And we're going to pilot that here as well. But the Wnt signaling pathway, I think the reason it's been difficult to drug is I don't think an inhibitor that completely knocks out Wnt signaling is going to work, because Wnt signaling is necessary for normal maintenance. So.
Great. Any other questions? One more in the back, right there.
As part of the phase 1 study, if there is biopsy material present, do you look for HK2 expression? There's stuff published previously about HK2 being
Right. So,
It's just to make it might be something else if there is biopsy.
yeah, so I guess when we're thinking about this, we're counting on not getting biopsy material, because one thing we're targeting our phase 1 trial to is patients with brain metastases who are actually often excluded from clinical trials, and there's a lot of them. So most of the brain tumors aren't treated by resection. But you are absolutely correct. There are a few papers that say that HK2 is increased, which also suggests a change in the metabolism as the tumors go to the brain. It's a good question.
Great, thanks again, Dr. Yi.
So I'm representing AACR Boston, which is our neighborhood network of cancer researchers. So what our organization is, we are a 501c3 nonprofit, so feel free to donate to us. I feel like a presidential candidate, aacrboston.org. So what we do is we're intending to host networking activities, but we sponsor 2 named lectures in the Boston area. The Apfel lecture, and also to today's lecture, the Sager Memorial Lecture. So this lecture was endowed by Dr. Art Pardee, an eminent cancer researcher that many of you know, in the memory of his late wife, Ruth Sager, herself a cancer researcher and the first woman full professor at the Dana-Farber Cancer Institute. So Ruth, during her career, made lots of contributions to tumor suppressor genes, oncogenes, and cancer, and she was a very dedicated scientist. The story is that she was writing a grant and a paper in the last week of her life. So that's pretty amazing to me. So she's clearly driven by science, but she's also a very well-rounded individual with lots of interest in art and music, and a dedication towards training training students and fellows. Many of her trainees are spotted through science to today. So Art Pardee, who usually comes but is not here today, endowed this lecture in her memory, and it's to honor each year that we have it, an outstanding cancer researcher and an outstanding human being. So today in Steve Elledge, we have both qualities. So Steve is an exceptionally accomplished cancer researcher that many of you know his work in the cell cycle and DNA damage. And his work is characterized always by innovation and creativity and rigor. So we've enjoyed his many fundamental discoveries. So let me give you something of his background. So Steve graduated from the University of Illinois, summa cum laude, and he got his PhD at MIT and did postdoctoral work at Stanford. He started as an assistant professor at Baylor University and rose through the ranks, and in 2003, I think, something like that, so sometime in the early 2000s, moved to Harvard Medical School in the Department of Genetics, and he's been an investigator of the Howard Hughes Medical Institute. So Steve, as I said, has made many contributions to the cell cycle. So if you ever run into the P21 molecule, Steve discovered that. And he's continued an amazing career. If you look at his CV of over 300 some publications, the majority of them are in the highest profile journals in our field. So Steve is a member of the National Academy, the recipient of many prizes, the Paul Marks Cancer Prize, the Gardner Foundation Award, and the Lasker Award. But to give you an idea of the type of person Steve is, so we all appreciate his and you won't need to retaliate against me. It's nice. He's looking at me. We all appreciate his intellectual brilliance. There's no question about that. But I think many of us that know him well know him as a very solid individual and person. So I'm looking at his CV. Has all these amazing prizes, National Academy member and such. But then there's also a couple others that are just thrown in there. I don't know if he put them in there to see if I was really reading his CV or not, but 2 of them are the Mayo Middle School Hall of Fame and the city, and the keys to the city of Paris,
Paris, Illinois. So Steve was raised and born and raised in Paris, Illinois. And actually, I think that explains a lot. I think he told me once his favorite toy was a chemistry set. I think he said something about creating a stain on his mother's ceiling or something like that. But Steve embodies those great Midwestern values. He's a solid individual. He's a tremendous amount of fun to talk science with. And I know him reasonably well. He is a wonderful person, husband, father, son, brother, and friend. And in the spirit of Ruth Sager, Steve has a lot of different interests as well. He also has an interest in music, but maybe not the same kind of music as Ruth Sager. So Steve has a humongous iTunes library with the likes of Dylan, McCartney, and others in it. So, in the spirit of the Sager Memorial Lecture, we honor an exceptional cancer biologist and a great and a wonderful human being as well. So let's welcome Steve Elledge from Harvard Medical School by way of Paris, Illinois. Steve.
Thanks for that wonderful introduction. I do have a great interest in music. I went to the Desert Trip concert last weekend, so 2 weekends ago, for those of you who are familiar with that. So today I want to talk about some work that we're doing on trying to understand aneuploidy and its role in driving cancer. And I'll take you through some of the studies we published in the last couple of years, and then in the first third of the talk, and then I'll go into some of the unpublished work, hopefully soon to be published. So this is going to be a talk with not a lot of experiments. There are a couple of experiments in it, the first and the last. Two slides will have that. But it's an integration of a lot of different data sets, and this sort of gives you an example of what you can do with data that's mostly just sitting out there waiting to be looked at. We start with RNA high proliferation screens. We do cancer driver mutations, aneuploidy patterns, also focal events in the genome, and we end up with RNA-seq analysis and putting that together with patient survival data. So if we think about how normal cells are transformed into cancer cells, we think about the occasional deletion or amplification, but mostly we think of mutation in driver genes or gene silencing or chromosome rearrangements that convert a normal cell into a cancer cell. But what's missing from this is the elephant in the room, which is aneuploidy. And so aneuploidy was really the first hallmark of cancer, but people don't talk about it very much, and in part because it's difficult to think about. If you look at a cancer cell, they have lots of different types of aneuploidies. It looks like someone threw a hand grenade in the genome. It's really hard to interpret as opposed to focusing on an individual gene as a driver, RAS, or P53, something like that. So people have generally ignored it and don't talk about it very much. But about 100 years ago, it was as soon as the light microscope could discover or look at chromosomes, it was found that tumor cells had the wrong number of chromosomes. And this is Theodore Boveri, and he was studying sea urchin development, and he discovered centrosomes, and he's been credited with the chromosomal theory of inheritance because he found that when the chromosome number was incorrect during development, it completely messed up sea urchin development. Anyway, so in 1914, he hypothesized that the aneuploidy could be responsible for the characteristics of tumor cells, and in particular, he said that specific chromosome constitutions could be produced such that the cells that harbor it are given to unrestrained proliferation. So this is 100 years ago. And since then, there hasn't been a lot of progress on understanding how aneuploidy works, but we do know that there are specific patterns of chromosome gains and losses shown here by a heat map, and also recurring focal events that occur through the chromosome. So what we don't know is whether these events just happen to occur at a higher frequency and they're tolerated, or whether they're actually drivers of tumorigenesis. And there have been a couple of, I think, experiments that point towards them being drivers. One of which was is done by mouse modeling of spindle checkpoint mutants. So they have higher rates of aneuploidy and they get cancer. And also David Pellman did a nice experiment where he made a tetraploid cell and showed that that was more tumorigenic than the diploid version of that. So those point in the right direction. But how does it all work? And so we started this by doing a very simple thing. We were looking for, doing RNAi screens, looking for things that killed tumor cells. We thought, well, maybe we should also do it on a normal cell line, since we don't want to just kill every cell in the body if you find a target. And so we did this with human mammary epithelial cells. We did a very simple experiment. We put in an shRNA library and just said, what makes you proliferate better or worse? And so we got shRNAs that made cells grow faster, and presumably they're turning off an inhibitory gene, so we call these stop genes, and for suppressors of tumorigenesis. If you look in this group, they're enriched for tumor suppressors. So a lot of your old friends are in there. And then the things that drop out were mostly essential genes, but also some oncogenes. And so we call those go. So stop and go genes, what we were interested in. And we thought, well, maybe there are more tumor suppressors in here, some new ones that had been overlooked. It's sort of guilt by association. So we said, well, which ones would they be? And so we decided to look at recurring deletions, thinking they might be in those. And when we did that, we found that we did find a group of our stop genes that were in these recurring deletions, but they turned out to be there at a much higher concentration than we expected. And, we were looking for the sort of classical two-hit sort of thing. We never found a mutation over these deletions that might pinpoint which one of our genes was the driver. But it led to the idea that maybe it wasn't the sort of classical two-hit, that it was actually something more like a cumulative haploinsufficiency. And I won't go through all the evidence for that, but it's really a very, very strong argument that these deletions are actually working by cumulative haploinsufficiency. And we came up with this model we call the cancer gene island model for focal deletions and amplifications. And what we found, based on statistical arguments, are that regions that are focally deleted and are recurringly focally deleted in cancers are enriched for these negative regulators of proliferation, and they avoid the essential genes and the positive regulators, including oncogenes. And although from that study we didn't show this, but we have now, that focal amplifications are just the opposite. They're enriched in oncogene-like genes, and they avoid stop genes, tumor suppressor-like genes. So this is the model, and we were really interested in whether or not we could use this sort of reasoning to try to understand big chromosomal events instead of these small ones. And you can think about these parts of the genome as sort of like high-priced real estate. That's where the cancer cell gets the biggest bang for its buck, if you delete that. And if you take the whole arm, then you sort of have to take the good with the bad. So we were interested in trying to model this sort of concept of haploinsufficiency and triplo sensitivity, which is when you gain a copy of a gene and get a phenotype. We were trying to model that on whole chromosome aneuploidy or arm aneuploidy, which is a very frequent event. And we decided that it didn't work with the genes from the RNAi screen, probably because it's a very messy thing, and also because it only measures one aspect of tumorigenesis, which is proliferation, although a very important aspect. So what we did to get a better set of stop and go genes was to actually identify the driver mutations present in tumors. And to do that, we took advantage of the TCGA sequencing effort and built algorithms that allowed us to identify drivers mathematically based on the distortion of their mutational signatures relative to that of a neutral gene. And so we divided all the mutations up into different functional categories. such as null mutations, which are stop nonsense mutations, missense mutations, which change an amino acid, and you can actually use programs like polyphen 2 to predict whether they're deleterious or not, or whether they're benign, and silent mutations. And the idea here is that, for example, a tumor suppressor will have a high frequency of null and missense relative to silent mutations, relative to a neutral gene, okay? And it's really important that you use the silent mutations, because it turns out that the mutational background throughout the entire genome is not uniform. So different parts get mutated at different frequencies, and it has to do with heterochromatin accessibility and transcription-coupled repair, things like that. And so if you don't normalize it to the background mutation frequency, then all the giant genes look like they're tumor suppressors, and you might have noticed in the early sequencing forays, they predicted these giant genes were tumor suppressors, but they walked that back a little. So having done this, we built this algorithm to do this, and we also looked to predict oncogenes based on recurring patterns of mutations in particular domains, like think about RAS and things like that. So these oncogenes are often activated, or at least the ones you can detect by sequencing, by mutating a specific set of amino acids. And oncogenes have very few loss of function mutations, but they have a lot of missense in a particular place. So we did that, and we found a lot more tumor suppressors and oncogenes than had been previously predicted. And this is sort of what you expect mathematically. If you look at more and more tumors, you get a statistically significant number of genes that deviate from what a neutral gene would look like. And so this is just a heat map for the tumor suppressors and oncogenes mapped onto the hallmarks of cancer. And I don't expect that you can read these, but these are different pathways. And I'll just blow up a couple of these so you can just see what we've been finding. So over here on the left, we have a cell cycle regulation module with a lot of your favorite tumor suppressors and oncogenes in it, including the CDK inhibitors and P53 and regulators of P53. We have the DNA damage response and cohesin modules here. We also have, of course, all the growth factor response elements. This is the RAS, receptor tyrosine kinase pathway. And importantly, I just want to point out antigen presentation. Now remember, when the TCGA data set is only on primary tumors. So these people have not been treated with therapy yet. You can see there's an active signal for the immune system to respond to cancer, and that'll become important later. And also a lot of epigenetic regulators. We've actually now increased the size of the database. We've repeated this, and we're still trying to figure out what's going on, but there are a lot more events than even the ones I showed you. So how that's to be interpreted, I think will be interesting in the future. So in order to get at this question of aneuploidy and how it drives cancer, we decided that we had to give a score to each chromosome arm, because what we're interested in is trying to predict the frequency that you observe the loss of that arm or that chromosome across all cancers. And so the way we did it, we set up this chromosome arm score, which we call a CHARM score, for tumor suppressors, and it basically adds up the number of tumor suppressors across a chromosome arm. And you can't just add up tumor suppressors, because then, of course, the bigger arms are going to have more. So we realized that there's a penalty also for deleting something. And there's a proteotoxic stress event that occurs, and this is work of Angelica Iman at MIT. And so to normalize for that, we just divide by the number of genes on the arm. And also, we don't simply add up the tumor suppressors, because we know some are more potent than others. So we have to weight them based on their potency. And the way we do that is that we give them a score based on the distortion of the mutational signatures, which is what we think is just a sign of selection. And so the idea here is that if you think about the clonal expansion theory of evolution of tumors, you get a mutation in something like P53, and it causes a overgrowth of a clone of those cells, so they're outcompeting things. And each one of these cells has additional mutations that you can sample. And the more you have in this clone, the more likely you'll find something, and you'll get another mutation, and that'll then further expand, and then this basically takes the train to the tumor, and you detect it when you sequence it. And so, you know, in my mind, I think about cancer just purely mathematically. You don't have to worry about the cell biology of it and stromal interactions and all that complicated stuff. In terms of getting it to become a tumor, it's really a math issue. Now, curing it is quite different. Then the math doesn't matter quite as much. It's still important. But you can see here there are other tumor suppressor-like stop genes that are less potent than p53. This one only makes you a clone of 100. This is a log scale. But you'd say, well, you know, you'll never see these in tumors. But that's not true. You'll see them at 1% of the frequency that you see p53. And so the distortion of the mutational signature will be less, but it'll still be distorted. And if you sequence enough tumors, you will be able to find less and less potent tumor suppressors. And I would predict that you'll be able to find a mutation that causes a cell to duplicate one time if you sequence enough tumors. But these, of course, are meaningless in terms of therapy. But I think some people really want to get to that point. So we weight them based on the distortion of the mutational signature. We give them a score, and then we look at the correlation. And so this is the CHARM tumor suppressor score plotted against the frequency of deletions of these arms across all cancers. And what you can see is that it's kind of noisy, but we get a positive correlation. The p-value is pretty strong. I actually didn't expect it to be this good. It could have been 0 or even negative. Okay, so that looked pretty good. This is the more tumor suppressor-like you are, the more likely you are to be deleted. What about amplified? Well, if you look at amplification, it goes in just the opposite direction. The more tumor suppressor-like you are, the less likely you are to be amplified. And that makes sense because this is triplet sensitivity. This is, you've got something that restrains proliferation, you make a little bit more of it, it's not good for the tumor. These events happen, they just don't make it out, they don't take the train to the final tumor. Okay, so you can do that for tumor suppressors. What about oncogenes? Well, for oncogenes, we did the same sort of analysis. And what you can see is that there's a positive correlation. P-value is not nearly as good, but the more oncogene-like you are, the more likely you are to be amplified. And if you then look at it versus deletions, it goes in the other direction. And that means that these oncogenes, the more oncogene-like you are, the less likely you are to be deleted. And these are deletion of wild-type oncogenes. That means these oncogenes are having an effect in their wild-type state because they're regulators of proliferation and survival. And you know, these are they have some effect normally because they're part of a node that is involved in decisions of amplification, or sorry, of proliferation, not during development, or a stem cell. So now, remember, the oncogene, the way we predicted oncogenes and tumor suppressors is different, and they're orthogonal. So we can add them together. They're not dependent on each other. So we made a combined score where we looked at the tumor suppressor score, and then we subtracted an oncogene score, and also a score for essential genes. At least for deletions, essential genes are important. Because what we predict is that there's about 30% of all the genes in the genome are haploinsufficient. And probably all tumor suppressors are haploinsufficient because in order to evolve the way that we think about with respect to clonal evolution, the first hit has to have a phenotype or you don't expand. So, you know, the two-hit hypothesis really does work for familial cancers, and you see examples of it in sporadic tumors, but that's not the only thing going on. And then once those things have to have an effect on the first hit. But when you combine these three things, now you can see the correlation gets much better. And the p-value is really good, and the R value is about 0.75, which is great. So the more tumor suppressor-like you are, the more likely you are to be deleted, and the less likely you are to be amplified. The R value is not quite as good here, but we know that, that we're missing some of the oncogenes because not all oncogenes can be activated by mutations, and that's the only way you can find them in this particular model. And the weighting's probably not quite right because you're weighting them on the basis of mutation, but you're counting them with respect to how they behave as triplet sensitivity and things like that. So this is for chromosome arms. You get the same sort of event when you do whole chromosomes, and now the R value is 0.8, which means the R square value is about 0.65, which means that we can explain about 65% of the variance in the frequency of chromosome deletions here, based on the distribution of genes along chromosome arms. Not so well for amplifications, but for deletions, it looks pretty good. So this led us to this model we call the gene dosage balance model of cancer, where the actual distribution of tumor suppressors and oncogenes and essential genes along chromosomes predicts whether they're likely to be pro-deletion or pro-amplification. And this term score then helps predict the frequency that you see this various cancer karyotypes. So this is part of how, because the aneuploidy can be explained on the basis of drivers that are activated by point mutation, it argues very strongly that aneuploidy is driving cancer. Okay. So I wanted to touch on this dark matter of the genome, the stuff that we don't know about in terms of oncogenes. And these are genes that aren't mutated in cancer per se, but may still drive cancer by virtue of gene expression changes or by amplification. So how do we find these? And so we decided to look for these drivers of proliferation in cancer by doing a genetic screen. And to do that, we built an ORF library in a TET-inducible vector, and we barcoded it. And so we have about 30,000 ORFs that correspond to about 16,000 genes, and it took us a long time to get these. But basically, we barcode this vector randomly. We recombine the libraries in by gateway, and then we sequence, paired-end sequence, the ORF to figure out which barcode it's linked up to. You can then put it into a cell that has a reverse TET transactivator, RTTA, and then add doxycycline and ask, well, what happens to proliferation? And we've done this for multiple normal cell types, so we do it for normal cells. We don't do it in cancer cells. And this just shows you some examples. This is what we see in terms of the data that comes out. It's really very good. And since we did it randomly, there are multiple barcodes for each ORF, which is really helpful in showing the reproducibility. And this is just now looking at the things that are amplified, and all of a sudden you start seeing things that make a lot of sense. You're getting cell cycle regulators and tyrosine kinases and MYC and things like that. So that's good. If you look at things that drop out, that's also interesting. So you're starting to see a number of CDK inhibitors here. And we saw interferon genes, a real enrichment for interferon genes because they're very toxic to cells. And this is net log 2, so this is a pretty strong dropout of these genes. And this is just, the first 70 or 80 genes, but we have 16,000. It's a continuum. But there are a lot of new genes there. We took just the known cell cycle regulators from the 3 cell types we looked at. We looked at human mammary epithelial cells. We looked at fibroblasts, IMR90 fibroblasts, and then pancreatic, normal epithelial pancreatic cells, all of which are immortalized with telomerase, so normal. And you can see that they correlate pretty well. This just looks at the 2 different screens. For the cell cycle regulators, they look pretty good. But if you look at the whole data set, you can see here that if you do non-hierarchical clustering, the mammary epithelial and the pancreatic epithelial cells look a lot more like each other than they do like the fibroblasts. So that makes sense. If you, we have 2 libraries that we use together to make up this set. So library 1, we did all 3 of these cell types. Library 2, we only did 2 of them. The other cell type got contaminated, so we didn't get the data from it. And my students graduated, so oops. But you can see that one of the most interesting things here is that there are vast differences in the proliferation response of different cell types. So there's more overlap between HMECs and HPNEs, but between all three of them, there's only 3 that make this p-value cutoff of 0.01. But there's a lot more things in the stop gene category. So it's harder to make them grow better, but it's really a lot easier to make them not grow so well. And I guess that kind of makes sense because you're hitting core components of the cell, maybe overproducing something that jams them in an off state. But anyway, if you just look at these two, there's a lot of overlap here in the things that slow them down, not so much here. And then the question is, why are they so different? And what does that mean? And so we found lots of interesting sets of genes. So in breast cancer, we found this whole family of keratin-associated proteins that cross-link keratin. There's 2 subtypes of this, and these are very cysteine-rich proteins. And those are the ones that score, and the other subtype doesn't score. But you can see we're getting gene after gene that is enhancing proliferation very strongly in breast only. And if you look at this in the pancreatic cells, they do nothing. They're just about like the olfactory receptors. They have no signal at all. They have very little signal in IMR90 cells, but they're all very positive here in mammary epithelial cells. And if you start taking the drivers of the different cell types, something interesting came out when you look at the sort of GO categories, the gene ontology categories. And what you can see, this just gives you a heat map based on whether, if you look at whether they're more highly expressed in breast or pancreatic cells, you can see that you're actually picking up genes that are activating aspects of that are specific to breast cells here or specific to pancreatic cells over here. And so it looks like that you're triggering pathways that are really part of the identity of that cell type. And so it suggests that there's a sort of underlying genetic architecture in these different cell types that we're sort of revealing by looking to see what genes you can perturb to turn them on. So this is actually very interesting biology. You can't get to this very easily other ways. Oh, sorry, the wrong way. Okay, so if you look at the sort of drivers of proliferation and map tumor suppressors and oncogenes on them, you get some other really interesting things. So this is just a graph that says whether or not it's likely to be dropout or enriched in mammary epithelial cells or pancreatic cells dropout or enriched. And you can see that some of the main drivers of pancreatic cancer, such as KRAS and BRAF, are strongly enriched in pancreatic cells, but they do nothing in HMECs. And you don't see RAS mutations in breast cancer. Likewise, I mean, there's a lot of things in black here that either drop out or turn on in both, but you see these ones that are colored are different. So here's a tumor suppressor that's often found in pancreatic tumors, but not in breast tumors. And you see it starts to drop out here. And then here's some breast-specific tumor suppressors, such as GATA3. It doesn't do much for pancreatic tumors, but it drops out tremendously in breast cancer. So, so we're still seeing this sort of underlying architecture. What else is going on here? What else can we learn from this? So these genes, if we overproduce them, make you grow faster, we call them GO genes. And if they make you grow slower, they're stop genes. So it's the same sort of idea that I brought out earlier, just to simplify this. So if you look at the GO genes and look at cancer amplifications, you can see that the HMEC-specific ones, relative to the pan-cancer amplicons, are enriched just like the pancreatic drivers. So across all cancers, if you look at recurring amplicons, you see they're both enriched, so that means they probably have drivers. We're finding drivers here. But if you look at just the things in breast cancer, the amplicons, you can see that the genes that were found in HMEX behave much more strongly than those that were found in pancreatic. And the opposite is true. And if you look at pancreatic adenoma amplicons that are recurring amplicons, you see a greater enrichment for the genes that are driving this sort of normal pancreatic cell type. That's true for the GO genes. It's largely true for the STOP genes, with one exception. Okay, so here, both the STOP genes, and I didn't necessarily expect this to be true, but they're avoided in recurring deletions. Okay, so some of the STOP genes, I thought, oh, you're just messing up, metabolism or something. You're just throwing a hammer at things. But actually, if you're avoiding them in deletions, they're probably really negatively regulating proliferation. Sorry, they're enriched in deletions. So you see that the pancreatic derived STOP genes are much more enriched in general in pan cancer for whatever reason. But in breast cancer, they're about the same. The p-value is actually stronger for the STOP genes in HMEX and the recurring deletions. But this is one place where they don't look that different. If you go back now to the recurring deletions in pancreatic cancer, the STOP genes in HP&E cells actually outperform, which is what you expect. So what you're seeing is that you see enrichments in amplicons that are tissue-specific, or enrichments and deletions that are tissue-specific that follow the cell type that these driver genes were found in. Okay, so this brings us to the question of whether there's an underlying tissue-specific genetic architecture that determines which genes can control proliferation, which is what we think. If so, we would expect that it would be largely conserved in related tissues. So we did the same analysis, and we looked at tissues that were related to breast or tissues that were related to the pancreas, and asked if their recurring amplicons and deletions behave the same way. And so for breast-related, we looked at ovarian and uterine and endometrial cancers. And what you can see is that the HMEC GO genes are more enriched in ovarian than the pancreatic ones, and that's true in uterine cancer as well. For the STOP genes and deletions, there really is not that much difference. But when you go over here to, these are for the breast genes, they behave about the same. But remember, the pancreatic STOP genes were really good for some reason. So they're about equal there. But if you look over at cancers related to pancreatic cancers, such as the amplicons in stomach adenomas or colorectal cancers, now the pancreatic-derived genes are outperforming breast again. And that's true also in the deletions. Here you can see that those are much better. So this suggests that there's a relationship, an underlying relationship between these different tissues that we know are sort of related developmentally, and that the driver genes are more related. It makes sense. But this also predicts that then you would expect that their amplicons, which I think are as a result of these genes, the pattern of amplicons that you see in focal deletions would also be more closely related. And so when you do that in non-hierarchical clustering, you get the same thing. And so what you can see here is that if you just look at recurring focal amplifications or deletions here in this heat map, you can see that breast cells are closer to ovarian and uterine-derived cancers in terms of what their pattern of amplicons are, and the pancreatic is closer to stomach adenomas and colorectal cancers. So this sort of just argues that the drivers that you can detect in vitro are actually reporting on something important underlying in these cells, and that this helps drive the pattern of focal events in these cancers that are related. And so just another thing about finding these genes, the goal here was to basically find some of these other drivers, not the ones driven by point mutations, but by overproduction. And these are just the chromosomes. These are twice the size, just because they were getting so small, it was hard to read them. But what you can see here is that these are the recurring deletions or amplifications. Pink is breast cancer, blue is pancreatic cancer, and gray is pan cancer, but these are just the genes mapped on the chromosome. So these are all in the amplicon. So these are candidates for new drivers that we found, and there's about 140 of them. Of course, we found a lot more that aren't in these regions, but we think these are now new candidate drivers. So how important are these events in cancer? If you look at the number on average per tumor, there's about 1 oncogenic mutation, maybe 3 tumor suppressors, but there are lots of whole chromosome events, whole arm events, or focal events. And you have to ask the question, well, they're selected, but how important are they? What can they tell us about cancer? And so we were interested in trying to understand more about how these drivers are actually driving cancer. So we think it's, as I told you, that aneuploidy drives cancer by altering the dosage of known tumor suppressors and oncogenes and new ones as well. And we also think that once you start having aneuploidy, you probably are more unstable, so it's sort of a slippery slope, so it allows you to evolve more quickly. But are there other things that are important? So I wanted to get back to the question of how important these events are, and in order to do that, I wanted to look at survival. And to do that, we decided to give each tumor a score based on its aneuploidy, the degree of its aneuploidy. So we sort of added up these different arm events and whole chromosome events and focal events and gave a sort of SCNA level score. These are somatic copy number alterations to each tumor. And then we could start correlating the degree of aneuploidy with different things. So one of the things we correlate it with is drivers. What kinds of drivers are enriched in high SCNA level tumors? And so here we look at the number of drivers, mutant drivers, that are in the DNA damage pathway, including BRCA1 and 2, and things like that, but ATM and any kind of DNA repair gene. And what you can see is that the more drivers that are mutant, the more genomic instability, the higher the aneuploidy sort of signature is. Makes perfect sense. If that didn't work, I would have quit doing the analysis, right? So, but then we went on, we found something that was actually kind of surprising, which is that we found there's a particular type of driver that anti-correlated with this aneuploidy score. And these were mutations in the receptor tyrosine kinase RAS PI3 kinase family. And what you can see here is that the more drivers you have in this pathway, the less genomic instability that you see. Now, this is surprising to us because there's been this whole theory about how oncogenes drive genomic instability. And you hear a lot about it. It starts with the fact that if you overproduce RAS, you can do senescence and you see DNA damage in these cells, which we know induces senescence. But those are usually vastly overproducing RAS. And I think that when Tyler Jacks actually made the RAS mutant on the chromosome, it didn't do that. But people stuck with that sort of idea that oncogenes are inducing it. And then Bartek and Hal Zanetis looked in early tumors, and they could see DNA damage foci. And these are early, but not really early tumors. I mean, you can actually see them, so that's been going on for 20 years maybe by then. But you can see foci of DNA damage, H2AX foci, which is recognizes double-strand breaks. And so since they knew that oncogenes drove tumors and you could see damage, it must be that the oncogenes are inducing the damage. But no one's actually tested this. And the only tests where people have done any tests is if they overproduce cyclin E, which is we know messes up the cell cycle, they can see genomic instability. So I think that this is the largest family of drivers, of oncogenic drivers out there. And if oncogenes are inducing genomic instability by making DNA damage, you should see a signal here, and you don't. So I think that whole thing is wrong. Okay, no. Sorry guys, that's just the way it is. So we gave them, this is just another way that we weighted, gave the tumor scores. So then we wanted to ask the question, well, does aneuploidy predict survival? If we take these scores, do they predict survival or not? And so we looked in a number of tumors, and these are the 4 that we got a signal for out of 10 or 12 we looked at. So it's not everything, but you can see that aneuploidy, the higher your aneuploidy score, the poorer your survival is. So it's Kaplan-Meier plots for melanoma, low-grade glioma, uterine, colorectal cancer. So, and the hazard ratios are pretty good. Here's one that's 4.7, but there are others are around 2 or 3. But still, that's pretty strong. But we thought, to be fair, we shouldn't just do it for aneuploidy. We should do it for mutations too, because that's sort of what people generally think about in terms of driving cancer and driving survival. And so we made a mutation score also per tumor. And then we got something rather surprising, which is that the higher scores, the more mutations you have, the greater your survival in all of these. This is, here. So, and the hazard ratio is really quite good here as well. So, aneuploidy bad, which is, people have thought this before, so we're not the first ones, but mutations are good. So this is really kind of a surprise. And I know some people think that they're anti-correlated, but they're actually not. Mutations actually possibly correlate with aneuploidy, and we can discuss why that is. But it's been published otherwise, but those are all driven by the most hypermutated samples, and when you take those out, there's no anti-correlation. Okay, so here we have it. Okay, we think that these higher numbers of mutations are probably better for you because the immune system has a better shot at you. So just to simplify why this is, in case you didn't get there yet. So now we have high and low aneuploidy tumors. What else can we do with these? Well, we can compare their transcriptomes. So we did that, and we looked for things that, gene sets that were more highly expressed in high aneuploidy tumors, and what we got are cell cycle regulated genes. DNA replications, mitotic cell cycle, chromosome maintenance, all the things you expect. And so what this probably means is that higher aneuploidy tumors are probably more mitotically active in general. Okay, makes sense, not surprising. And other people have seen this in cell lines, but these are now in tumors. So we found something else, though, that was surprising. We found that there was actually a set of genes that are depleted, whose expression are depleted in high aneuploidy tumors. And these are things that are associated with the immune system. And so what you can see here is this is CD8 cells, interferon gamma signaling, NK cells, B cells. These are all much lower expression in the tumors than in more diploid-like tumors. So, and aneuploidy is somehow suppressing this. Of course, the tumors aren't expressing these. This is the immune infiltration. Okay, so it looks like the higher aneuploidy tumors have a lot less immune infiltration. So what's known about immune infiltration in tumors? Well, there was a paper published by Nira Conan last year that suggested that the immune infiltrate into a tumor positively correlates with the number of mutations. Made sense? We redid that analysis here with our scoring matrix here, and just looking at the immune signature, infiltrate, and mutation number. There's a positive correlation in 3 cancers: colorectal cancer, lung adenoma, and uterine cancer. That is statistically significant. Now, the R squared, the R values are very low, about 0.2 to 0.3, and the p-values are 10 to the minus 2 to 10 to the minus 3. And the rest of these don't have a signal. So, and that's basically what Neer found also. So I just want to point out these three tumors because now I'm going to show you what the immune infiltrate looks like with aneuploidy. And I want you to remember these numbers here on the next slide. So now we're looking at at the aneuploidy score and immune infiltrate. Of course, it anti-correlates for everything except lung or brain cancers. And the p-values are actually pretty amazing. Here's 10 to the minus 10, 10 to the minus 7, and the correlation coefficients around 0.4, really very, very high. And for the three in which the numbers of mutations correlated, you can see now that our R values are close to 0.35 or 4, and the p-values are much stronger for all three of these. And so it says here that basically that the aneuploidy score has a much stronger effect on predicting the immune infiltrate. And also for 10 out of 12 of these cancers, there's statistical significance. And this is just another way of looking at this data. This is a heat map for the false discovery rate, which is sort of the p-values of p-values. The darker you are, the better it is. This is now looking at GO terms, gene ontology terms for CD8 cells and NK cells and B cells. And this is actually looking at a signature for those cells exactly, directly, Tregs, NK cells, B cells, and CD8 cells. And you can see that everything except glioblastoma and low-grade gliomas score. Everything else has a very strong false discovery rate. Another measure of immune infiltration is immunoediting, and that is that if you have a certain number of mutations, you can predict the number of neoepitopes you expect to see based on what can load onto MHC. And if you have immunoediting, that means you have a lower ratio than you expect because you're killing off the cells that have neoepitopes that the immune system can recognize. The only place you can really see this in action is when you have a lot of mutations. So if you look in colorectal cancer, you can see this. And so we look to see whether or not the number of neoepitopes observed over expected ratio was reduced. So in low aneuploidy tumors, you see lots of immunoediting. So you only get about half the number you expect. But in high aneuploidy, you get a lot more. And if you just do this with the number of mutations, it really has no difference at all. So this is another measure that says that aneuploidy is affecting the immune system. So we can now ask what aspects of aneuploidy affect these signatures, the cell cycle signature and the immune signature. And the way we do this is we do a sort of machine learning-like program called LASSO, which is a linear regression model. It basically says if you have these parameters, how do you weight them to best predict something? So the thing that we're predicting is the cell cycle signature, and the things that we're asking it to weight are either the whole chromosome or the focal events. Okay, and so what you can see here is that the things that best predict the cell cycle signature are focal events. They have higher numbers, higher beta coefficients. That's a weighting parameter. But the arm level events are actually, they do contribute. So this makes perfect sense. So it's darker red on top than on bottom in general. But this makes sense because the focal events are picking up particular gene sets. And the whole arm is, you get the good with the bad, but you're still getting the arms that are more good. But it's a weaker effect. So this makes perfect sense, okay? You pick out the prize real estate as opposed to having to buy the whole block. Okay, so what about the immune infiltrate? Well, there it's quite different. In this case, you only see whole arm events, whole chromosome or whole arm events. This is the main driver. There's very little effect of the focal events. And so this argues that it's not particular genes that are driving it, but it's a property of a whole chromosome. And the property that we know about of whole chromosomes is proteotoxic stress. And so that's interesting. So then, you know, we set up just another analysis with a whole bunch of different parameters and asked the model to weight them, including mutations, to predict the immune infiltrate signature. And what you can see is that across all these different cancers, except for the brain cancers, the chromosome arm and whole chromosome score is dominating. And even in the 3 cancers where you see a signal for the number of mutations, the chromosome arm level is of higher weight. It's negative, but because it's anti-correlated, but it's more potent. And if the area under the curve is really pretty good for these, for those of you who are into that kind of analysis. I won't go into it. So we wanted to actually do an experiment to test this, and so we made we took a syngeneic mouse tumor model. It's largely diploid, so you can put it into a mouse, into a wild-type mouse, and it makes a tumor. And then we made them aneuploid by treating them with a spindle checkpoint inhibitor. So this is now a collection of aneuploid cells. They're heterogeneous versus the parental diploid. And what you can see here, if you just grow these in culture, the highly aneuploid cells grow much more poorly than the diploid cells. It's because they're aneuploid, they're sick. It's not good to be aneuploid. All from Angelica's work. But then we injected them into tumors, or into mice to look at tumor genesis. This is into an immune deficient mouse. This is a RAG1 and RAG2 mouse. You can see that, again, the aneuploid cells, tumors are much poorer in terms of how well they're, how the rate at which they grow. But now if you put this into a wild-type mouse, now they're basically the same. So the immune system is hammering the diploid cells tumors a lot more than it is the aneuploid tumors. And we think that they're growing about the same rate because these don't have the burden of aneuploidy, but the immune system's holding them in check, whereas the immune system has less of a chance at this, but they grow more poorly. So this supports it. What about the immune infiltrate? Well, if you take these tumors and now look at particular gene sets or different cell types in them, you can see that you just look at T cell infiltrates, that the diploids have a higher level of T cell infiltration. And this now looks at the CD8 to Treg ratio. I didn't point it out earlier. Tregs and CD8 cells are lower in high aneuploid tumors. They sort of work in opposite directions. But it turns out that you deplete the CD8 cells more than the Tregs. So the actual balance of what's left is even skewed. And you see that also, aneuploidy suppresses that as well. So we think that what we're seeing in vivo is real, and you can recapitulate it in vitro. Can these impact therapies such as immune checkpoint blockade? So we looked at that. We gave aneuploidy scores to patient tumors who underwent CTLA-4 therapy, and we plotted their survival. And it's just focused on the top. This is from Levi Garraway's lab, Van Allen et al. And you can see that on the left, the high aneuploid tumors survive and respond much worse than the low aneuploid tumors. And this is the top and bottom 50%. We didn't find the best part and separate it. And so the hazard ratio is about 2.2. You do the same thing for mutations. They're predictive, but they're not statistically significant. You can add them together, and it actually improves the p-value and the score for hazard ratio. And we did it on a second set from another set of but smaller number of patients. You get the same results. So, so I think we're getting late here. I think I'll skip my hypothetical explanation for why I think this works, because I don't really know. And so it's just a bunch of BS, but it has some pretty pictures, and we'll just skip right through them. I can. I don't even believe it, but I had one idea. I think. We have other data I'm not talking about yet because it's not ready. You know, so we think it's working through antigen presentation. I think that is probably what's going to be. We know that's important. And so the question is, how do tumor cells present antigens? And antigens this is now for class 1 MHC. Usually what happens is you get whatever is turned over. These peptides get pumped into the ER and loaded onto class 1 molecules, which then end up on the surface and present their epitopes to T cell receptors. So how do these proteins get chosen? One model, well, it's known that the cell is, like most of the things that go on MHC are things that are rapidly turned over during translation. So if you add cyclohexamide, it just stops it, you know, and so most of them are newly synthesized proteins. They're called DRIPs because there's this guy who thinks that they're defective ribosomal proteins because the ribosomes are inaccurate and then you don't fold right and you get degraded, but I don't believe that. I think it's too important to leave up to being random. I think there's a dedicated machinery, we just don't know what it is yet. But then there's also general turnover. So things that are stable but fall apart and get degraded. So those are things that have the wrong stoichiometry. Those don't get sampled here, but they end up getting degraded anyway. So it's a ratio of these two things, but this dominates until you have aneuploidy. And then you have thousands of proteins that are made at the wrong level. And so we think that this general turnover mechanism, and these are mostly wild-type proteins, and so there's flooding through the second mechanism, and it's competing with this sort of even sampling here, and that may throw the cell into disadvantage in terms of properly sampling its epitope. So thousands of proteins are affected. So this is sort of a flooding hypothesis. It might be playing a role, but I think there are probably other things that are at play. And when I tell my immunology friends this idea, they hate it. So, but you insisted I tell you. Okay. So just to summarize here, we think that the balance between growth-promoting genes and inhibitory genes along chromosomes ultimately determines the selective pressure that drives somatic copy number changes that you see in cancer. And this is cumulative gene dosage effects. And this model surprisingly worked. We think that aneuploidy is driving at least two hallmarks of cancer, enhanced proliferation and immune evasion. And we think that we've found that there's sort of an underlying genetic architecture in cells that are cell tissue specific that controls proliferation responsiveness. And that also then is responsible for part of the SCNA patterns, the focal amplifications and deletions that you observe in those tissues. And those appear to be tissue specific and shared among related tissues. The immune evasion is predicted largely by arm and chromosome alterations, and we're really interested in how that works. And we hypothesize antigen flooding might be a possible mechanism. But importantly, aneuploidy is a much stronger predictor than mutation load for immune infiltration, and it better predicts the outcome of immunotherapy. And if we can figure out how to undo that and somehow suppress the defect, maybe that will enhance immune therapy. And of course, we all know that immune therapy is the hottest thing out there. It's the best news that we as cancer researchers have received in our entire careers. And so we're all hoping it gets even better, which I think it will. So I'll just stop and thank the people who did this. The cancer gene island model was started by Nicole Solomini in my lab, and the cancer gene island and most of the computational analysis was done by an extremely talented postdoc, Teresa Davoli, who's on the job market now, and she's amazing. And she collaborated with Peter Park and Andrew Wu in his lab to do the sort of cancer gene dosage model and prediction of tumor suppressors and oncogenes, and with Hajime Uno to do the survival studies. But she found the connection between aneuploidy and the immune system, and I think that's going to turn out to be important. So Thank you for your attention.
So we have time for a few questions. Any novel slug genes that are predictive of immune infiltration in and of themselves?
Yeah, so the question is, have we found any of our stop genes that are that can predict immune infiltration? We haven't actually looked at that. What I will say is that none of the SCNAs, the focal deletions or amplifications, seem to predict it in our models. That doesn't say, I'm not saying that there aren't individual genes, because we know there are. And those are probably more sporadic rather than general, since that signal probably gets washed out in the sort of large-scale analysis.
Thank you for a great talk. You understandably look mostly at solid tumors. Have you looked at this in any of the hematologic malignancies? And if not, any reasons to think it would show differences?
We have not looked in those tumors, and I don't know what to predict about that. For some reason, we avoided those in our. I think we avoided those when we were doing the focal event analysis early on, just because we thought they would be less related, and we'd get a stronger signal from the epithelial cancer. So we focused on those and sort of stuck with that data set since then. But yeah, they could be quite interesting in and of themselves.
I have a final question. So your hypothesis at the end was that if you flood the proteasome system, then you decrease the immune infiltration because the molecules can't be displayed on the cell surface.
Well, they're competing. If there's if it's a zero-sum game or saturable, that you might disadvantage the more even sampling mechanism.
So proteasome inhibitors are used in some cancers. Are they used in solid tumors?
They don't work, but most of them don't work. Most of them don't work in solid. They don't even get in, I think.
They don't even get in. Okay,
Yeah.
So I was going to ask the question about the proteasome inhibitors and the immune checkpoint drugs. They would be expected to be opposite then.
They would.
But if they're not used in solid tumors.
Yeah, that's a good question. Because I think people are aiming to get some proteasome inhibitors that work better. You know.
That was very interesting. So I think there are no further questions. Let's thank Steve for a great talk, and I'll turn it over to the Dean's office for the summary. You could just leave this up. She's only going to be.
Okay. Well, you need to sit down.
Well, I seem to get to come at the beginning and then come at the end. I must say, one of the most exciting parts of my day was to catch just a bit of the final talk. I wish I could have been with all of you for the entire day. I hope you've really enjoyed your time here at Tufts, particularly if you are not here at Tufts all the time. I think this has turned out to be a terrific day. I want to particularly thank everyone who participated in the program. I think it's been really great. And I'm particularly grateful for the opportunity that our investigators had to showcase just a small part of the cancer research that goes on here. Before closing and turning this back to Phil for final announcements, I'm hoping you'll all join me in thanking the sponsors of this event again, particularly the Boston chapter of the AACR, Cancer Research Technology, and the Raymond and Beverly Sackler Convergence Laboratory, not to mention, of course, or leave out the School of Medicine, and particularly our Department of Developmental, Molecular, and Chemical Biology, which took a lead role, along with all the faculty and staff who worked on the program. So please join me in thanking these people.